perform in the proteomics right now. Once you've identified the targets, you want to validate them. Then two approaches. One is the triple quadrupole based approach known as MRM. When you are going to know which peptides you are looking at for your validation, you are selectively looking at those peptides, doing their collision and generating the transition list from those, looking at the intensity in the time frame, and use that information to validate your protein expression level. Or you can look at simultaneously the entire window, and you can do the parallel direction monitoring from the arbitrary based configuration. So these are the two approaches. Just to sum up, proteomics, we have two approaches mainly: discovery or targeted approach. Of course, one could always go functional to understand the function. But you have to first start moving from the discovery to validation, because nobody will accept your finding if you don't validate them. Fortunately, now the mass spectrometers can also do validation, as you have just talked about the MRM and PRM. So good workflow will revolve around these two approaches. Discovery approaches, now with the shotgun, label-free, or the TMT or DIA, they're all very powerful approaches for you to look at, identify thousands of proteins or even quantify the proteins, do the entire mapping of the proteome. Whereas now, if you have to dig deeper into a few peptides and proteins of interest, you can now take it forward and you can go to hypothesis driven or the targeted proteomics based approaches, which can be used for the validating your peptide list. In addition to the mass spec based approaches, which is the focus of the course, but I do want to just show two slides to give you a flavor that other technology also exists. We sometimes conduct only workshops solely based on these technologies. So a lot can be done on the target proteins, which I just talked, and the protein arrays field. I showed you a video in the beginning. Idea was that if you have entire proteome array chip when all the proteins are printed, and you want to understand the function of an unknown protein, you can probe it with the entire proteome chip and see where it binds. So then now you will have some interactors which you can give an idea that probably these are the similar protein which will have that kind of function, or which pathway they belong to, right? So these kind of things you do with the different type of array based approaches. Or you can always even look at how best you can now identify the kinetics of these protein and their binding with the other drugs or other type of biomolecules. So therefore different type of biosensor label-free technologies are also in the progress to look into these kind of interactions. So just to summarize my lecture, idea was to start moving from the gel-based technologies to the gel-free proteomics, and you have seen like how 2D guys moved with the mudpit and the ICAT technologies, leading towards currently being used most actively use iTrack, TMT or LFQ-based technologies, and now we have to validate those different type of approaches with the targeted SRM, PRM, etc. So the entire workshop will be revolving around the balance of discovery versus validation. While I think there is more focus and time for discovery approach in this workshop, but you have to be also prepared in future for the validating your peptides and the proteins. So that's kind of a broad overview of bombarding you with a lot of technology, a lot of terms. Of course, we have time to digest that and discuss more and more as we go along in the next 2 days. Uh, we do not know what is the level of each participant is, so it is best for you to ask questions, discuss with us more so that we understand like where we can help you or where we can talk in more detail, right? All right, thank you very much, everyone. One or two quick questions if anybody has. You have to ask question, otherwise we'll ask you question. Okay, don't hesitate. Yes. Can we move the microphone?
Good morning, sir. Can you please explain the difference between DDA and DIA?
Right. So while that could still be a very broad topic to talk, but as I briefly mentioned, in DDA you are only looking at few very abundant precursor, which is being, let's say, every millisecond, mass spec is scanning few ions which is coming from the peptides. So you are only selecting maybe top 20, top n, which can be read in that millisecond time, and you're only fragmenting those. So your entire intensity quantification will be based on that. Whereas in the DIA, you first try to build the library, which takes a while to build the library with high resolution for the entire M by Z region. And then you run your sample and try to overlay the spectra on that. So in a way, DIA is going to give you much broader coverage you're not biased from the few abundant peptides or abundant peaks, whereas the DDA is much quicker, but it will give you maybe less coverage and the less data as compared to DFT.
In DDA, we are getting less proteins as compared to DNA. Abundant.
No, not in terms of protein. Protein injection will be same. Amount of protein what you inject will be same, but the workflows are different. In one, you are telling mass spec to only look at the top n abundant precursors. In that millisecond, how many you can reach, right? So then, therefore, you will have the limited coverage. Yes. So after this, we'll stop because I'm already late. Yes.
I wanted to know in that orbit of that machine, there were three analyzers, the iron, the orbiter, and the
Quadrupole.
quadrupole. Um, so what's the function of that orbiter analysis?
Right. So again, like the idea for all of these different hybrid technologies are to utilize different principles of different mass analyzers. So the quadrupoles are treated as the mass filters. They are always same. They will only, let's say, you will take the top 20 most abundant, which will be queued up. Now, orbit trap, all the ions are going to move in that orbital with the going to separate it in the orbit trap configuration. So again, that is one of the principle like top or like quadrupole. When you are separating the ions in the orbitals of that orbit trap. So therefore, it is one of the way. It actually works with the something similar to the C trap and the ion trap kind of technology, but it resolves the ions very much with the good coverage. But then further, you can still utilize even ion trap technology in the same instrument. When now you are going to look at very selectively few ions which can be further, bombarded and then the collision. So again, here we have two different type of collision cell as well, HCG and we have the CIG. So idea is again how best you can utilize different principles as well as different type of collision to improve the coverage. All right. So there are a lot of terms as you must appreciate in the field, and it may not be easy for everything to go so fast the way I have taught. But let's kind of introduce the terms first, and as you go along, please do keep asking these questions. When you go to the mass spec and you will have more interactive time, don't hesitate to ask these questions again if it was not clear. And we really hope that you will be able to start doing some of these things on your own, right? And that will be idea after these 2 days. Thank you very much, everyone, for showing up here. And let me thank all the speakers here as well who have all come from different places and trying to make a good show for all of you to teach proteomics. Dr. Grambhal, next, your talk.
The next, you have that. Now we have next up, have Professor
hands-on से पहले हमको देखना है।
Registration lain.
आज दे दिए
क्लियर आ रहा है। हेलो हेलो বলছি
तेरा हो गया
नहीं एक लड़की बैठी है उससे मांग लेंगे इसको मांग लेंगे नाम बताओ नीता 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 वो एग्जैक्ट कोई नहीं है क्या नाम उनको मिल गया उनको अरे नहीं उनको पीछे वाले में उनको लिखना है वो कौन जा रहा है कौन जा रहा है नोट पैड दे भाई इतना मतलब है नहीं नहीं
So good morning everybody, thank you very much for such a warm welcome. It's a great pleasure to come to IITB again, and thank you also Sandeep for giving a very eloquent introduction to components that I will... Okay, that's better. To components that I will talk about. In a sense, translational sciences involving mass spectrometry are two sides of the coin. You have on one side the generation of data, converting clinical samples to useful information. And then on the other side you have the process whereby you do something with that data, how you extract knowledge from that data. And that's where techniques around statistics and machine learning and artificial intelligence come in, because that is how we start to interpret that data, try and find useful things. So this part of the presentation, and I'm presenting tomorrow as well, is about big data analysis for biomarker discovery. So what is a biomarker? Well, this is short for biological markers. It's a measure of a biological state. The state of a protein, the state of an individual, or the state of whether the individual has a disease or not. And it's a characteristic that's measured objectively and evaluated as an indicator of a process. Now these can also be molecular, they can be physiological, they can be measured parameters. So it's not just protein or gene.
So, sharing the screen. Oh, okay. Second.
Okay, so the next term that is important is big data, and this is used a lot. So what is big data? Well, it's data that is large. I hate the term big data because it does what it says really, but this big data can be hard to manage, it can be having multiple structures, you can have complex interactions. Imagine you have image data, sound data, proteomic data, and text data. And then you have to somehow integrate that. That's kind of the extreme end, but that's kind of the nature of the problem. In biology, it's data generated by measuring large numbers of molecules, possibly by high throughput technique. And it's representing the large volume of data in the human genome, transcriptome or proteome. Just to highlight that, imagine what complex organisms we are. 22,000 genes, potentially being up and down regulated, leading to hundreds of thousands of proteins, all of which are post-translationally modified, existing in a different temporal location, existing in a different spatial location, and all possibly interacting. That's how complex we are. 22,000 genes. So big data can be a challenge. There is what is the nature of this problem? Well, for precision medicine, and it was stated very eloquently by Sir William Osler, we were not for great variability amongst individuals, medicine might be considered a science and not an art. That's quite contentious. And that's because individuals all vary, they all are in different states. Medicine isn't defined by a standard nomenclature necessarily. So an example of that, diseases are not defined by a standard nomenclature. They can be defined by a location, so Japanese encephalitis, a symptom, hypertension, body part, breast cancer, the person who discovered them, Alzheimer's disease, and the causative organism, the dysentery, or the age group in which they occur. So there's no standard nomenclature there. So we don't define the diseases based upon the molecular biology, we define them based on various things. So that makes it difficult to have a standard taxonomy. Now in the post-genomic era, we have the potential to start to characterize diseases in new ways. But we generate a huge amount of data, this big data. Diseases are very diverse, and one has to search through potentially hundreds of thousands of pieces of information, pieces of data, to find a consistent marker or feature that addresses the clinical population. So that's the challenge of finding the marker. A solution should ideally identify a small set of markers. So if we look at 100 patients and we find 200 biomarkers that define disease versus healthy in those cases, that's not a valid signature. Every individual could have their own marker and it won't be generalized. One marker will not answer all questions. The complexity and heterogeneity of the population, a single marker is unlikely to be the sole answer. So, where do we use precision medicine? Well, precision medicine can be used for diagnostics. Does somebody have the disease? It can be used for prognosis. What is the outcome for this individual? Are they going to be at high risk or low risk in the future? Will their disease progress rapidly? And we can use them for predictive medicine. Will an individual respond to a therapy or not? Based upon their profile, what's the best way to treat this individual to get the best outcome? And that's really kind of where precision medicine is headed. And there are challenges in big data. The first challenge is the quality and replication of the data. We don't want samples to be degraded. In DNA, RNA, we can look at the RIN level, which tells us the quality of the RNA integrity. But data quality is really important in proteomics because samples denature very quickly, proteins denature very quickly. So you have to make sure that you haven't left the sample too long before fixing and freezing it and using it. When we're looking at expression, each expression measure, i.e. each protein or each gene, should have equal reproducibility. And this was one of the problems with the old expression arrays. People were finding genes not because they were the answer to the question, but because they were the best quality probe that stably measures them. So you have to be careful that you're not just selecting genes that are easy to measure. We have this adage, rubbish in, rubbish out. If you make rubbish data, if you put rubbish data into your algorithm, you will not find anything stable, you will not find anything reproducible, you will not find anything useful. So as well, what we need is a good sample size, sample number. I'm not going to dwell on that too much, but at the end of the day, if you can allow for cross-validation, You can split your data and look at seen and unseen set. That's also practice. The next challenge is one called recursive dimensionality. It sounds awfully Indiana Jones. And what it is, though, is where you have very large data sets, and the effect of a single gene enmeshed in tens of thousands of genes gets masked, gets taken. So what's happening is the dimensionality of the data means that any given parameter will have a small influence, or appears to have a small influence, even if it is actually strong. And it's masking the importance of things. So, we look at this about searching for straw-colored needles. It's hard to find those things when you're looking at large numbers of things. Another challenge is computational time.
Ball.
So if we were to develop a classifier in 6,000 parameter dimension, 6,000 dimension data set, and we wanted to find a gene panel of 10 genes, then, and we wanted to cross-validate that 50 times, then we would have to look at 3 times 10 to 47 different combinations of genes to try and find that 10. That could take a lot of time. So the advent of high-performance computing, the advent of using GPUs, is all speeding those processes up to help us get to an answer more quickly. We also have a challenge around generality of the models we look for. We want models not just to be focused on a small population, we want models that actually work for the general populace. What I mean by that is diagnostics shouldn't just work for the people who were used to make the model of the diagnostic. They should work for every individual in the population, or at least in a geographical region. And it should be broadly applicable to a population and not be constrained. And unfortunately, scientists have a tendency to try and control things. So that also is true in diagnostics. So for example, I had a PhD student, Gopal, who was looking at estrogen receptor positive versus negative breast cancer. He wanted to understand transcriptomic biomarkers that were predicting immunohistochemical status. And when he did that, he found a paper that was looking at estrogen receptor positive breast cancer in patients aged 55 to 58 who were HER2 negative, who had a grade 3 stage 2 tumor, had overexpression of Q67 VEGF. And they controlled the population that they looked at so much that it's statistically relevant. And in the UK, we worked out that could mean 4 individuals like that a year. So it's got to be generally applicable. Now, the number of genes often exceeds the number of cases. I mentioned that earlier. There's one study in tuberculosis, I'm not going to name the author, but they had 293 genes that explained the population of 120 individuals. And they had a sensitivity of 78% and a specificity of 76%. So that's not statistically valid. It's not parsimonious. Biology is non-linear. Look around the room. There are no people in this room who are purely made up of cubes. This becomes a challenge because it's non-linear, because genes are switched on or switched off. Interactions can add to further complexity, things like PTMs. The problem is here, linear methods will only identify things that are linearly related to them. So if you stay with a linear paradigm, you will not find non-linear features. So when you're analyzing biological data, you have to have something which is perhaps more non-linear in terms of its search space, when you're modeling things. It also, though, to some extent, extends to parametric statistics and looking at just size of changes to find biomarkers. There is a challenge in data mining as well, is that the same algorithm doesn't find the same answer if you look at different data sets. or the different algorithms don't find the same thing when they look at the same data set. And this is a challenge as well, because that suggests that the studies are underpowered, suggests that there are too few replicates, and that the algorithm is not finding the global truth, the ground truth. So what we've done is looked at individual data sets, but then compared them and identified concordant features between them. So if you start to adopt a concordance approach, you're looking for the same genes that come up in every data set, or come up in every algorithm in the same data set. And those tend to be more powerful, more robust than trying to find things which are not there. So how do we find biomarkers? Well, we can use a difference-based method. These are parametric, so there might be a differential expression, a t-test, fold change, and many people use volcano plots as a way of showing that. But we can also use things around the information content in a marker. So this is modeling based. How good is a marker at predicting a yes/no answer or a question? How good is a marker at predicting the outcome for a patient? And so this is about the information content, not necessarily the magnitude of change. A lot of biologists have tended to look in the past for the big signals, the big differences, but it's not just about that. What happens, for example, with transcription factors? So we can use artificial intelligence and machine learning in that space to start to try and understand the patterns in markers that relate to a given question. Now, if we're looking at difference-based methods, we have a problem in that complex data will always have a chance of getting something wrong, type 2 errors. We can find things that are not there. These are so-called false discoveries. So if we had a 5% type 2 error rate, and we used a p-value of 0.05 to select our cut point, that means 5% of our discoveries, i.e. the significant differences, are false. Which means, in a data set of 50,000 features in data, 2,500 things would be wrong. They would be misidentified as features, biomarkers, and we couldn't use them. So just random those. So we overcome this by correction for multiple testing. And there are two methods, there's Bonferroni and Benjamin Hochberg. So Bonferroni correction is a stringent approach. It literally divides the p-value for significance by the number of genes looked at, the number of tests conducted. So let's say we had a p-value threshold of 0.05, there are 50,000 probes. The new threshold for significance would have to be 1 × 10⁻⁶. So it means that we have that stringency. We just look, but unfortunately, that just looks at the biggest, most consistent changes. So there's a slight issue there. We go back to our parametric idea. Benjamin Hochberg does this too, but it's slightly less stringent, and it looks at the rank of the variable. So the higher ranked variables get more reinforcement, lower ranks get less reinforcement. So we've talked about a modeling-based approach. And a model is a representation of a real-world system. So often things relate to cause and effect. So a model might be if genes X and Y are upregulated, gene Z is downregulated, then the cancer is highly likely to metastasize. Simple logical model. But we can represent the system by numerical formula. So we can use a function to say what is the probability of a tumor metastasizing. And there's a term which has emerged recently, and you've heard of in vitro in vivo studies, and that's in silico. And this is where we start to model systems and model, identify new things by searching with computers, by creating these experiments, if you like, on a silicon chip. So that creates a new opportunity to rapidly identify things, because you can have one measurement of one biomarker in the lab takes so long, but in silico, if you've got the right data, it will take nanoseconds. So we can explore these models to understand the system. Just to illustrate a modeling idea, what is a model? Well, the first question I'll ask you is this an aeroplane? What do you think? Who thinks it's an aeroplane? Hands up. Right, that's good. So you've won a free holiday to London. You can get on your aeroplane and travel there whenever you like. The point is, this is a concept, it's a model. It communicates an idea, but it has a large error. The model is flawed. You couldn't sit on it. It doesn't have wheels, it doesn't have a pilot, it doesn't have windows, it doesn't have engines, so on and so forth. If we look then at the next step, is this an aeroplane? Well, yes and no, but it still has an error, but that error is now smaller. So now it has a representation of an engine, it has a representation of wheels, and so on. So you could say what type of aeroplane it is based upon that model. If we look at this, is this an aeroplane? Well, no. It's just a very big model. But it's big enough and realistic enough to fool people. Now this is the real thing, because it's flying. So there is zero error with this. But you can see there's a continuum of error moving from the paper aeroplane to the real aeroplane, where we represent the system in different ways. So scientists like models, and a model is a very useful tool because it's a way of communicating something, representing something, but also sharing ideas. So, how many people here have seen now a double helix in reality? No, but you can all probably draw one, because it's a model used to create an idea. And similarly, cell structure, Einstein's theory of relativity, which does have its errors, it's not a perfect model. Serum albumin structure, glucose structure, the immune system and immune function. These are all models that are used to represent systems, but communicate ideas as well. But they have their errors. So just to look at how do we use models as a quick introduction, how can we use models for precision medicine? So as an example, we have a mixed population here represented by a well-known UK sweet called the jelly baby. And some of those individuals will have one response to a drug, others different response. So we can profile those individuals here by expression arrays, and then we can use machine learning, here a simple neural network, as a decision support model to predict, by selecting key probes, whether an individual would respond to treatment A or treatment B. Now, we can use the neural network model to actually work its way through all those probes and identify features which predict. And that's called a combination of a filter and a wrapper method. It's a machine learning technique, where the model performance is used to select the features that make the model. So we have to define our error as well, and that's the difference between what actually happens and what's predicted. So we're trying to minimize that error. We go back to the paper aeroplane example. We want to try and get the minimum possible error, but and just to be aware we can transform error values. So we can, for example, root mean square transform into root mean cosine and so on. Now, if we use these in a machine learning context, we take data from a mass spectrometer, say. By using a combined filter and wrapper method, we can select features from that mass spectral profile and make predictions of a particular question. So, for example, with just off the room with Sanjeeva and Deep, we've been looking at medulloblastoma for diagnostic purposes from mass spectrometry of medulloblastoma. We use model performance to select the parameters based upon error, and these are potential biomarkers. And we then cross-validate that. So we look at the model with different randomized subsets, training on one set, testing on another, to see if our model has generality, see if our model can predict for new cases, because that's the way of validating in this model system, and that's analogous to kind of the false discovery elimination approach that is used for parametric methods. So we can cross-validate with this across multiple data sources, and that allows us to get robust biomarker sets. So thank you very much for listening. I'll be happy to take questions. But there is a rumor going around. And that rumor is that I've based my academic career on using not a human brain, but an artificial brain. And there is some evidence to suggest that is the case. Any questions?
Good morning, sir. It was really a nice explanation. I have a question on finding biomarkers. During the time of cancer or any other disease, a lots of protein get dysregulated. But some of the protein regulate expression of their downstream protein, and we can assume that act as the master regulators. So from the proteomics data, how can you find that which are the master regulators?
That's another talk, but most people look at biomarkers as lists of genes or proteins. But in reality, they are in a dynamic state. They are interacting with one another. They are interacting over time, interacting over space. So the way we look at that is if we make a model of how an enriched set of biomarkers interact with one another, we can find what the most influential genes are in the system, the most influential proteins, and what the most influenced proteins are. And those will be the master regulators. So if we look at, say, 250 genes associated with medulloblastoma, for example, and we find that there is one that is strongly influencing all of those other genes, then that will be certainly a regulator. If we were to look at it at a genomic level or a transcriptomic level, that may well be a master regulator. And if we look at that in a pathway context as well, we could also see what is regulating the pathway. But what we found is when we knock these highly connected features out in using siRNA or CRISPR, we actually change the phenotype of the cells. And we, for example, one study where we looked at proliferation, we could switch off proliferation by hitting a target that was highly influential in the system. Sorry, it's a long answer, but that's kind of the next step is looking at how things interact with one. Thank you.
So, thank you, Professor Ramahar. We are immensely grateful for the knowledge that you have shared with us today. So, thank you for sharing all this information with us today. With great admiration and gratitude, we would like to extend our felicitations to Professor Ramahar.
University of California, San Francisco, for his postdoctoral training and worked on synthetic immunology to address novel immunotherapeutics development. Leveraging his extensive proteomics expertise, he developed a new technology platform called structural proteomics to enable identification of protein conformation-based cancer antigens for CAR-T therapy development. Using this technology, he successfully developed a novel CAR-T therapy against acute myeloid leukemia. Dr. Mandal was recognized with the prestigious Michelson Prize 2019 for his contribution to the
Then I
field of
think you will not
synthetic
be able to use
immunology.
the
He also
pointer.
received the abstract of
Yeah,
the
pointer you cannot use.
from the American Society of Hematology. Recently, Dr. Mandal has returned back to India and joined Intest Pharmaceuticals as a
Use the
principal
pointer,
scientist.
but you have to use the
So,
keyboard to change the slide.
we welcome Dr. Kamal Mandal. With his round of applause.
So, good morning, everybody. Thanks to Professor Sanjeeva for inviting me here. This is the first time I'm here as a speaker. All my prior visits to active on the weather students. So, yeah, it's a pleasure for me to be here. I'm going to talk about applications of proteomics technologies for cancer therapy development. So, for effective and safe cancer therapy development, how do we actually do it? We should be able to discriminate the cancer cells versus normal cells. And how do we do that? We need cancer markers. The cancer markers are proteins that are present on the surface of normal cancer cells, and that's not present on the normal cells. And that's how you can specifically discriminate the cancer cells and kill them in a targeted way. And that's how we can be alive. And that's what is called a therapy. And the therapy should be only to deplete the cancer cells. In case of cancer, the infections you want to deplete the pathogens. So there's a severe dearth of cancer-specific markers, and that's a question that we were addressing in my postdoc. So I joined my postdoc in the lab at UCSF back in 2018, and there we hypothesized that can we expand the toolkit of cancer marker discovery and maybe go beyond protein expression-based approach and find protein conformation-based cancer antigens. The same protein present in the normal cells and the cancer cells, the cancer cells display some unique conformation, and that unique conformation can be targeted for immunotherapy. So before I go into the proteomics part of my talk, let's start with a brief introduction of what is immunotherapy. So immunotherapy, by definition, is nothing but tricking the immune system to make our immune cells act efficiently against the disease. It could be a pathogen or it could be a cancer in our case. So in case of cancers, immunotherapy is started with PD-1 inhibitor, checkpoint inhibitor, for which Professor Holmes was awarded Nobel Prize. That was the antibody against PD-1. And antibodies are regularly used for cancer therapy these days. Trastuzumab against breast cancer is one of the most popular ones. But recently, CAR-T therapies has gained a lot of traction, pioneered by Professor Carl June. CAR-T therapy is basically engineering the T cells, but in principle, we can engineer any immune cells, and that's why broadly we call it engineered cellular therapies. So just a brief introduction of the working mechanism of immunotherapy. Typically, it's antibody-based, and where we know that antibody is the class of biomolecules that can bind to its cognate antigen with a very high affinity. And that's how it can be specific for the cancer antigens, right? And just leave the normal cells untouched. So in the Fc domain of this antibody, that could be effector cells. Effector cells could be macrophage or NK cells. Could be macrophage or NK cells. No. You have to share the screen. Okay. In engineered cellular therapy, I mean, this is antibody-based approaches are definitely in clinic and it works very well. But to further advance the field, what people have done is that you take the variable region of the antibody. It's the variable region of the antibody that determines the specificity of the antibody against this cognate antigen. And it's just these two, light and heavy chain of the variable region, and put it in an SCFD format, and you display that SCFD on the surface of a therapeutic cell. Therapeutic cell could be a T cell, and in that case it's called CAR T. If it is NK cells, it will be called CAR NK, and for macrophage it's called mCAR. So essentially, what we're doing is engineering the therapeutic cell so as to display this antigen binding domain with the antibody on the surface of this therapeutic cell, and helps it to recognize the cancer cells more effectively, and then deplete it or kill the tumor cells in a specific manner. But whether it is antibody therapy or engineered cellular therapy like a CAR T therapy, the clinical range is still limited because of one big reason, and that happens to be dearth of cancer-specific markers. So all these modalities still needs cancer-specific markers. So we're we hypothesize that can we come up with a technology with which we can find cancer protein confirmation-based cancer antigens, and we ended up using Proteomics technology, basically merged cross-linking mass spectrometry with surface proteomics to come up with a new technology called structural surfaceomics, and with which now, even though the cancer cells and the normal cells might have the same proteins present on the normal cells and cancer cells, but the cancer cells might display unique conformation, and we can engineer an antibody for this unique cancer-specific antigens, and then put that into a separate format. It could be a antibody-based approach, and again, antibodies could be reconstructed into a engineered cellular therapy-based approach. So here we developed this new technological structural surfaceomics by integrating cross-linking mass spectrometry with surface proteomics. What we do here is start with cells and treat them with a chemical cross-linker and freeze the 3D structures of the conformation, because we want to retain the 3D structure before conformation, because in our case, 3D structure is our biomarker is based on the 3D structure of the protein. So once we freeze the 3D structures of the protein conformations, then we label the cell surface proteins with the biotin so as to allow the enrichment of the cell surface protein. Because mass spec has limited capability, you want to enrich for the stuff that you want to see, and you don't want to deplete the stuff that you don't want to see. And then this is followed by pull down for the cell surface proteins, and then we do regular trypsinization of the trypsin digestion, and we run this plasmid peptides into the mass spectrometer. We get plasmid peptides, and these plasmid peptides we map onto the tissue structure and that's how we infer the difference between the protein conformations in normal cells and the cancer cells. I mean, in principle, this approach could be used to address any disease, including cancer. But in our case, we used AML, acute myeloid leukemia, as the model system to implement this technology for the first time. AML for a simple reason, because it's high unmet need for AML. It's a disease which doesn't have any FDA-approved CAR T therapies yet. 5-year survival rate is very poor, and it's really this dearth of good therapies against AML. So this was our workflow. I used 2 different cross-linkers. Cross-linkers are small molecules with which you can freeze the 3D structural conformations by using. So the cross-linker that is used these days are NGS chemistry based, which is lysine targeted. We have used 2 of such cross-linkers, DSSO and FOX. DSSO is an MS cleavable cross-linker, and that allows us to do MS3 based mass spectrometry, and that's kind of an online enrichment, of the cross-linker peptides during analysis. And we also use another cross-linker called FOX, which has a phosphonate handle, which allows offline enrichment of the cross-linker peptides using IMAC purification. And we developed this whole workflow. It's a parallel workflow that the same samples are subjected to both of these workflows to get better coverage. And interestingly, what we see here, there's a radius of reactivity of DSSO is longer than FOX, and that allows us to have better coverage of crossing peptides. With both these cross-linkers, we get 2 complementary set of crossing peptides, and just to enhance the coverage. During the process of developing this technology, we also ended up developing 2 softwares, one called Wing, and that's to help the proteomics community to do MS3-based accelerated analysis, which is otherwise that's a bit difficult these days, till date. So our software will be freely available for academic purpose through Proteomics pipeline, which belongs to Institute of Systems Biology, Seattle, Professor Robert Morris. So we developed this tool with the help of Morris Lab at ISP, and this it's a part of the bigger paper, which is currently accepted in principle in Nature Cancer. We also developed another tool called alpha cross, and this alpha cross is to allow us to map the crossing peptides directly onto the structure by in an automated fashion. And this alpha cross was developed in collaboration with Professor Sanjay Srivastava's team. I think Deep, Sanjot, and Ayushi is in the audience here. So they were the part of it. So these two, one would be available very soon, and this should be available in near future. So we applied our workflow to AML, and what we found is that most of the crosslinks we obtained were within the radius of reactivity of the crosslinker. By that I mean the radius of the actual distance between the license and the digital structure were less than 20 angstrom. But we found 4 crosslinks here overlaid on this blue bar, which we see like way beyond the radius of crossing, radius of reactivity of the crossing. And that suggests that these two domains are much closer to each other than what appears to be here. And that also means that integrin B2, that's what this protein complex is present in active conformation in cancer cells. But in normal cells, we know that it is in inactive conformation. And by that, we mean that suggests that in AML, integrin B2 is present in active conformation. So we can, in principle, target this active conformation of integrin B2 to address AML and develop a therapy. Immunotherapy against it. So we went ahead and validated our observation with antibody with flow cytometry, and we tested it against 4 different central lines of AML, and we found that indeed it is true that AML has integrin B2 in active conformation. We also had native controls, and they were integrin B2, but it was not active; it was in inactive conformation. We also validated our observation in patient cells. We show here the data of 10 patient samples. So indeed, we are true. It was positive in AML cell lines. It was positive in AML patient primary samples. The next objective was to engineer an antibody against this specific conformation integrin B2. So we used the state-of-the-art technology cell display in collaboration with Dave Siddle's lab from University of Toronto. And we initially got 10 hits, and then we funneled it down for further validation. We obtained 4 hits, which was validated by flow cytometry. And then we did further validation to finally ended up with 1 CAR-T hit, which was amenable to be used in CAR-T format. And I'm going to explain this part in more details in the next slide. So how do we discriminate at which antibody binds to active conformation of integrin B2 and which binds to inactive conformation of integrin B2? So this is how we do it. Integrins gets turned on when we add manganese to the buffer. So when you add manganese, if your antibody binds to the active conformation, we should see the boost-in signal. So that's what we see here. For these 2 antibodies, you see, like in presence of manganese, we see boost-in signal. That is, we don't see that in these 2 antibodies. So that means these 2 antibodies binds to the total integrin B2, but these 2 binds to the active integrin B2. And eventually found that 7065 that works better in CAR-T format than 7341. So this is how we typically do it. Like if you have an antibody, we take the heavy and variable light chain, stitch it together to form an scFv, and this scFv is then put into a CAR construct which has intercellular domain as CD3 zeta and co-stimulatory domain. So basically that's the finite transistor domain that relays the signal and activates the T cells, and that's what leads to T cell tumor cell killing. Was that I mean, no project goes without hurdle, and this was no exception. But it turned out that integrin B2 is also active in activated T cells. And when you make CAR-Ts, you activate the T cells. What happened in the first time when I was trying to make these CAR-Ts that they were killing each other, and we were not able to manufacture these CAR-Ts. So that's where CRISPR came into the picture. And what we ended up doing is we knocked out integrin B2 with the T cells before we transfused it with the lentivirus carrying the CAR construct. And then we were able to manufacture the CAR-Ts. Thanks to Jennifer Dorner and Emmanuel Charpenter for developing this brilliant technology. And our grad owners were working great. Thanks to Brunello Library for that. And then now we are able to manufacture these CAR-Ts, and then we found a drastic survival advantage in mice treated with our CAR-Ts. And it was indeed as comparable to CD33, which is used as a technical positive control here. CD33 is only a technical positive control here. The reason I say that is CD33 is indiscriminately present in the entire malware lineage, and it's not a good therapeutic CAR therapy, because it will basically wipe out your entire malleability, and that's highly toxic. It's just a technical positive control. So, and the same thing we see with BLI imaging. The previous one, the data was in the PDAs. This was the cell lines labeled with luciferase, and that enables us to measure the tumor burden with BLI imaging. And then coming to the toxicity part, we did a single cell analysis analysis, and we found that integrin B2 abundance expression is pretty restricted within the hematopoietic system. And even within the hematopoietic system, we are targeting the active conformation of integrin B2, and that provides us an additional layer of selectivity for the tumor cells, leaving the normal cells untouched. And then for any therapeutic development, the target therapeutic development, or any engineered cell development, we have to ensure that it doesn't touch the hematopoietic system. Hematopoietic stem cells. Hematopoietic stem cells are C34 positive. C34 is a marker of hematopoietic stem cells, and with clonogenic assay, it showed that our CAR-Ts doesn't touch the hematopoietic stem cells. And same thing we also showed with flow cytometry analysis. So our CAR-Ts were non-toxic to hematopoietic stem and progenitor cells. And then for further toxicity assessment, we resorted to this mice, humanized immune system mice, where we inject the C34 positive cells, as I said, C34 positive cells are hematopoietic stem cells. So we inject human C34 positive cells in mice, and then we reconstitute the human immune system in mice. And then we trade these mice with CAR-Ts and show that is our CAR-T toxic to our human immune system. And remember from the previous data, we know that our CAR-Ts target expression is only there in hematopoietic systems. We don't have to care about toxicity elsewhere in the body. So we made this mice and challenged them with CAR-Ts, and what we show, and then we measured the human CD45 positive cell abundance pre versus post CAR-T treatment, and our CAR-T treated arm looks pretty similar to empty control, empty arm. And as a positive control here, we see, as I said, the CD33 is a bad target therapy for AML. We should see there's a massive decline in the abundance of human CD45 positive cells, and that suggests it's very toxic. It turns out that our antibody 7065 that we are using is also cross-reactive murine version of antibody, the murine version of activating in vivo. Typically, this doesn't happen, but it does happen. You have a chance that it might happen if you are using cell display platforms, like a synthetic antibody engineering platforms, and not the animal immunization based. So we took this opportunity to do CBC profiling of our mice treated with CAR-T, and it turns out that it's non-toxic. It turned out to be non-toxic there as well. So coming to the conclusion, we came up with a new class of cancer antigen, which happens to be protein conformation-based cancer antigens. We also developed the unbiased proteomics technological structural surface, which we can find such cancer antigens, and then we also identified active integrin B2 as one such target for AML, and we successfully developed the promising CAR-T therapy for AML targeting active integrin B2. So this work was highly recognized, and I was awarded Michelson Prize in 2019, which was widely covered by the science news outlets in US and around the world. And then that also made me the lifetime member of Michelson Foundation, and then I was also invited for the other prestigious talks. So that was about my postdoc talk. Now I want to move on to the current positions where I'm currently working at. So I'm now back to India recently, and I joined Indus Pharmaceuticals as a principal scientist. And here I'm working on clinical CAR-T therapy development for commercialization in India. So let me briefly talk about how we do CAR-T therapy. Like, how is CAR-T therapy administered? It's a personalized therapy. What do? You have the patient admitted to the hospital. You need to harvest the blood of the patient, and you want to isolate you need to isolate the T cells out of the blood, and then you need to genetically engineer these T cells with lentivirus. This lentivirus would carry the CAR construct. This CAR construct would be specific for the cancer, the tumor type that you're targeting. So let's say if it is B cell malignancy, typically it's CD19. So you want to, your lentivirus will carry the CAR construct against CD19. So basically now you have the blood, isolated the T cells, transduce it with lentivirus carrying the CAR construct against CD19, and now your T cells are engineered to be very more potent against the CD19 expressing cells. Now put this engineered T cells back into the patient, and then these engineered T cells will take care of your tumors. So that's how it is. It's highly personalized autologous format, and thus very expensive. So there are 6 approved CAR T therapies approved in US, 4 are against CD19, and the remaining 2 are against multiple myeloma targeting BCMA. BCMA is a marker of multiple myeloma, which originates from plasma cells. It's a plasma cell malignancy. So at Intas, our vision is to bring down the cost. The current cost of CAR T therapy is around $4 to $5 lakh. So $4 to $5 lakh just for the product. Hospitalization charges, clinician charges, that's additional. And trust me, that's humongous in the US. Hundreds and thousands of dollars per week, maybe more than that. We want to change that. We want to go indigenous, make this therapy in India, bring down the cost, and make it available to a wider population. We also believe that this will promote medical tourism in India. Patients from all over the world would be able to come to India, avail these therapies at a much cheaper cost. So that's the idea that we are pursuing at Intas, and we have several targets that we are pursuing different stages of clinical development. And in principle, cardiac therapy is basically an ex vivo gene therapy. You take the cells outside the body, genetically engineer it, and put it back into the body. This is ex vivo gene therapy. What is in vivo? There is also something called in vivo gene therapy, which we have several portfolios that it does. These are the various diseases that we are targeting, and it's in various stages of development currently we are working on. So the department's name is Cell and Gene Therapy broadly. I'm mostly focused on the cell therapy part, is the CAR T therapy development, but yeah, I also have some role in gene therapy part as well, especially in the molecular biology aspects of it. So with that, I would like to thank the funding agencies for funding my work at UCSF, and I would also like to thank Intas for giving me the opportunity and of which I'm currently a part of. And with that, I would like to take questions. Intentionally, my talk was brief because I wanted to take more questions. Yeah.
So whatever clinical trials you showed, it was on leukemia, blood cancer, AML, or so. What are the chances of CAR T cell therapy in solid cancers? Because we don't see much of the trials in solid cancer. So there are a lot of solid cancers which are very lethal, like the glioblastoma. So what are your thoughts on exploring the CAR T for solid cancer?
For solid cancers, CAR T is still a challenging thing. I mean, to be honest, or brutally honest, in CAR T therapy, the relapse rate is very high. And CAR T therapy is efficacious only for liquid tumors at this point. People are trying to make it work for solid tumors as well, but it's still a lot of academic work need to be done. The challenges that we have with solid tumors is that the tumor microenvironment of solid tumor is very immunosuppressive. Also, think of it, in liquid tumor, all the therapeutic cells and the target tumor cells, they are freely accessible to each other. Whereas in solid tumor, you have a mass of cells. So your therapeutic cell should be able to penetrate into the tissue to kill them. The exposed surface area is very limited. Third problem, which I believe is that the tumor heterogeneity in case of solid tumors is very high. So it might not be like single targeting might not be enough. They need multiple targeting. So we might have to go for digital targeting, and getting multiple targeting in parallel. So that kind of approach is we might have to take care of the immunosuppressive nature of the tumor microenvironment. That's one thing. And who knows? We'll see how the things unfold. Maybe T cells is not good enough. We might have to go to other kinds of cells like macrophage CAR, NK cells, engineered NK cells, engineered CAR T cells. And that's why it's the popularity today is called CAR T therapy, but in general, it's the umbrella term is cellular therapy.
So there is a question from online. So data asking can IPL also be targeted as we also change the conformation in active state like IPG?
Yeah, let's say that again.
Can IPG also be targeted as it can also change the conformation in IPG?
Okay, so integrins doesn't exist, like integrin beta doesn't exist alone. It always exists in alpha and beta pair, and let me show that picture. Uh, yeah, so integrins exist in alpha and beta pair. They cannot hold independently. So this is ITGL, and this is ITGB2. So when you say ITGB2 active, it's already associated with integrin alpha. And that's what it is referring to, ITGAL. Yeah, I think he has a question.
Good talk, very nice talk.
Thank you.
So, based on the conformation change as you see in the cancer cells and the normal cells, so in cancer cells, it is having an active conformation. So, what about in the inflammatory response as well as in the autoimmune disorders?
Autoimmune disorders and
of this result. What is the state of this?
okay, yeah. So let me make my best guess what I understand. Basically, you are talking about like what if some of the other cells in the body might also have some expression of active integrin B2. If that is indeed.
There's a possibility of conformation.
Yeah. So the T cells and see, nature didn't design active integrin B2 for cancer cells. Its purpose was what you're exactly saying for adhesion. So it's there in granulocytes, it's there in T cells, but it gets turned on. It gets activated when it has to. When the T cells themselves gets active. So it basically gets activated in a when there's like pathogenic threat or something. So yeah, so activated granulocytes, activated T cells, they would be at stake, but we believe that would be under a therapeutic window. Think about a cancer patient. Cancer patient, when the tumor burden is very high, the patient essentially doesn't have an immune system. It's really a bad scenario. So forget about any activated T cells. The patient doesn't have a functional immune system. So those kind of those population of immune system, those compartment of immune system would be under stake, but we believe that that would be within the therapeutic window. Also, activated T cells, activated granulocytes, when they get turned on, and that's when integrin B2 gets activated into the tissues, right? So basically, they will be around the site of infection, and site of infection and site of tumor burden, assuming that they would be spatially different. It's very optimistic that it should be therapeutically tolerable.
So, Kamal, I know there are a lot of questions for questions and queries on online as well.
Okay.
I request you to reply that when you get time after the session.
Sure.
Let's thank Dr. Kamal for excellent talk.
Taking these small basics and build further from here. And maybe in some time we'll invite you for this kind of event and you teach the explanation of thank you. So next 2 hours, let's be mentally ready to be very panicked, and kind of crowded environment to work. What we are going to do, we are going to show you a small way of sample preparation. A small step we are going to show it here, and then we will take you outside where the tea is being served. We'll move on with the bioscience department visit. So there are four groups have been made. Okay, group A, B, C, D. You might have seen. I hope it will inform you already which group you are in. So there will be group A and B will stay here for the sample prep, and group C and D they will move to bioscience department. So let's Cyclone will now and give. All right. So Hector is he's a PhD student. He's almost finishing PhD and quite well experienced in proteomics. He's going to take your session to start right now for the sample prep, and then we'll split and make the group outside. So just few minutes of announcement and demonstration here, and then we will go out for tea break followed by hands-on.
Stay back here to go to BSc, and then post lunch we'll be swapping these batches again. Okay, it will already before lunch, so we'll be swapping the groups again. Expect the groups to move to that area where you will be guided by four mentors. That would be Ayushi, myself, Orgo, and Amrita. So these groups would again be split into groups of 15 each. So assuming that there are two batches here, A and B, A would be further split into groups of 15 and 15. B would be into 15 and 15. And there are four workbenches where you will be working on. The first workbench would be where you load the samples onto the gel. The next would be where you would quantify the samples using Bradford assay. Again, the third would be a table containing a gel assembly, and the fourth for quantification. So one table for first part of 15 students of group A, the second for the rest of 15 for group A, followed by B, 15 from B and 15 from the last 15 of group B. So I'll give the presentation in a while, and I expect the groups to then move and get going with the steps.
Is internet in
Okay, so one part is that when you take any cell lysate or any tissue or any well cell sample, right? First of all, you want to make sure that you are lysing the cell, right? And the cell is break open and you want to release all the contents out, right? So, and that's what requires, some buffer, the detergents, as well as many time, either physical lysing or chemical lysing, right? That takes a little bit, kind of conversion procedure of doing sonication or freeze or different ways of, you're lysing the cell. So what I'll do now quickly, I'll show you at least, one of the way of doing the sample prep.
I will share this.
On the gene level.
Yes. If not, then I'll just share this. Screen produce.
Hello everyone. So for the success of any proteomic experiment, sample preparation is the most crucial step. Sample preparation involves protein extraction, protein digestion, sample cleanup, and final mass spectrometry. Depending upon the experimental objective and type of biological sample, different protein extraction protocol can be used. Different biological samples could be bacterial pellets, cell pellets, or tissue samples. Today we will demonstrate you the protein extraction method for a tissue sample. Here we have already weighed amount of tissue. For this, we will use urea lysis buffer, which is 8 molar urea. Appropriate volume of this buffer is added to the tissue. It is watered for a few seconds and spilled. After addition of buffer, the next step is homogenization, which ensures that tissue is completely light in the given buffer. So for this, now we will use sonication. For this, we are using probe sonicator. All procedure of sonication should be performed on ice. So after this sonication, all of the protein will be extracted into the buffer. After sonication, it should be made sure that whole content is uniform and nothing is left without lysing. If any chunk of the tissue is left, sonication step can be repeated. Now the next step is to centrifuge so that we can get the clear supernatant. Centrifugation can be done for 15 minutes at 8,000 times gravity at 4 degrees. After centrifugation, tissue debris are settled down and clear supernatant can be collected. Now, the sample supernatant should be collected in a clean, fresh abundance. This supernatant has all the proteins of this tissue. So this is the collected supernatant that have all the proteins. After getting these protein samples, now they can be taken for the downstream process such as protein quantification and quality check, or they can be stored for further use.
All right, so this much has been done for you, what we just showed you. All right, so these lysis have been done. Supernatant is what we have frozen now and will be provided to you in the tubes. This part, I just gave you the background, what has been done. What will be given now, section and the team will distribute you some tubes. Most likely we have to make the group of 2 at least, a pair, when people can do the pipetting or the other activities together. All right, section we cannot take one. So now you got a lysate from where you want to quantify, do the quality checks, followed by digest the protein and then under mask, right? It is all shortcut, very fast and much simpler way of doing proteins, right? Okay.
Yeah, so once again, I request everyone to please take a look at the group that you belong to. And the group A and the group B can listen. Yeah, so groups A and B can stay back here. Groups C and D can proceed for bioscience department. Okay. Okay. All right. So we'll just wait for another 10 minutes. The introduction session is common for both the for all the groups. Sorry. So the introduction session is common for all the groups. And once we are done with that, the respective groups can then disperse to the department. Yeah, so these are the four mentors that you should be aware of. Amrita, Ayushi, Orgo, and myself. The session briefly would talk about quantification, followed by SDS page, that is loading the gels on the proteins on the gel. Then plotting a standard curve. I'm sure that many of you would have plotted a standard curve by now, but then there would still be a few who do not know what a standard curve is and the use of it. So we just thought of briefly touching upon that. Then interpretation from the SDS page gels that you would be running, followed by a theory on in-solution digestion. So quantification, the very term quantification, I mean, the first thought that comes to your mind would be, why do you need to quantify proteins in the first place, right? You would eventually be injecting the sample into a mass spec, which would give you some quantitative values. But even prior to that, you need to be aware of the amount of starting material that you are starting the experiment with, right? So that is why quantification is very important. And once you and you can quantify these proteins in a number of different ways. The one that we are going to talk about today is Bradford assay, which is a very commonly used technique for quantifying the samples. And then there are different colorimetric based assays, as you can see in the slide. For example, you would have heard of Bradford, the BCA method, right? The Lowry assay. Depending on the starting material and the sensitivity that you are looking for, you can choose the method of interest. I personally choose the micro BCA approach, which helps me quantify very low concentrations of the samples. Today, you would be using a relatively less sensitive technique, that is the Bradford assay. So here the basic principle is that the Gomasi G250 dye, which is present in the Bradford reagent, when binds to the protein, it forms a protein dye complex, which under acidic conditions would change from brown to blue. So the absorbance is measured at 595 nanometers, and you will see that the Bradford that we give you would be brown in color. As soon as you add the Bradford to the tubes, it would turn to blue. So we would then also be loading 5 micrograms of the given protein sample onto a 12% SDS page gel. The idea here is to separate the gels the proteins based on their molecular sizes. And I'm sure many of you would be aware of the fact that when you use SDS page, you are using denaturing conditions here. The proteins would have a net negative charge, and then they, in the presence of an electric field, they get separated. The proteins which are smaller in size would be found at the bottom of the gel, and the ones which are larger would move relatively slower. And then this gives you this is a way to separate your complex mixture into much less complex components. And this also has use in mass spectrometry, which I'll just briefly touch upon in a couple of slides from now. Then standard curve. So standard curve is is a curve which helps you determine the unknown concentration. So you take a series of standard samples for which your concentration is known, and you plot them on a graph. Okay, and then you try to extrapolate the OD absorbance that you get for the unknown sample to find the amount of protein that is present in the sample of interest. One common mistake which most of you would be doing here is that you make a standard graph on day one of your PhD careers, and then you continue using it for the next 5 years or 6 years. This is something which should not be practiced, and the standard curve has to be always made fresh for every set of experiment because the conditions vary from day to day and from minute to minute. So we will be giving you a set of standard values which we have acquired already, and you'll be supposed to plot a standard curve here just to be sure that you know how to plot a standard curve. At the end of the day, the samples that you will be loading here, the samples that you would be adding to the well 96 well plate here, one of our volunteers would go back to the lab and scan them because it was cumbersome to carry the entire unit, the spectrophotometer here. We thought of getting the plates scanned over there. So you will be shared values of the samples that you have loaded, and we expect you to give us the answers by tomorrow. Yeah, so just to briefly touch upon what can an SDS page actually give you, the information that it can probably give you is how good your sample, your protein extraction was to begin with. This is a representative gel where we took the 4 samples that we have used for your experiment as well. You can see lane number 1 is U87, which says 5UG. The last lane is also U87, and we suspected that so we just wanted to see what will happen if you heat the sample and just load it. It did not contain urea, so we could heat the sample. And you see that there's no major difference. So, I mean, we just wanted to see whether there's any difference, and it doesn't seem to be the case. The next 3 lanes are for 3 different cell lines. And we have loaded 5 micrograms. So the immediate observation that you can probably make is that each of these cells has a different profile to begin with, right? So throughout your research career, you would be expected to know how your sample of interest normally looks like on a gel. And if there's any deviation from that, then it's your first indication that something was not right during the extraction procedure. Also, each of these bands can individually be cut, okay, and they can be processed. This process is called in-gel digestion. So this is one way of introducing a sample into a mass spec, wherein a complex sample can be made less complex and can be injected into a mass spectrometer to get information pertaining to that particular region of the gel. So when you cut one band, it's highly unlikely that you would get only one protein. It could be many different proteins which are very close as far as their masses are concerned, the molecular weight is concerned. So this is the basic interpretation you can make from a SDS page gel. You can't definitely identify which protein those a particular band belongs to. This leads us to in-solution digestion. So once you are done all the loading and running on the gel, the next step for processing your samples would be in-solution digestion. So as the name suggests, this is different from what I just spoke a while ago. This is not in gel. This is in solution, wherein you directly process the sample in its solution form. So the first step involves the so here you have already, your buffer would already contain certain denaturing agents. Most commonly used is urea, and also a few detergents. Not all detergents are mass spec compatible. So when you decide your buffer of interest, you need to be sure that the right buffer is being used. The first step now would involve the reduction. That is, you break the disulfide bonds to open up the protein structure so that the enzyme that you are likely to use in the next step is able to access the sites for cleavage. So after reduction, to prevent renaturing of the protein, you need to alkylate a protein. So that would then keep the structure open for access for the specific proteases. So the final step in digestion would be adding a specific protease. The most commonly used protease in proteomics experiments is trypsin, but you can also use a few more proteases. This would again go on to increase your identification and coverage when you would get to know that once you start working in this field. So finally, once the proteins get digested, you need to remove any salts which might interfere with your mass spec run. So the process of desalting is usually recommended. And you can then inject the sample into the peptides into the mass spec. You may or may not quantify your peptides prior to injection, but it's always advisable that you know what is the amount of peptides that are being loaded onto a mass spec. So this is just a brief overview of the procedure, and I request sir to take over.
All right, so we are definitely running a bit late on the session. What we will do now, all of you can have a tea break outside. After that, those who belong to group A and B will stay here. Those who are in the group C and D will follow some of our volunteers and will walk to the bioscience department. All right. How much time you can save in this moment in transit is what going to ensure the success of today's hands-on. All right. So please be quick. Everybody can join outside for tea and then you can move to the DSB. Half of you. Remaining half, then you can take over and tell me. Yeah. All right.
So the batch is having the group A or group B.
So batches, what you have is already having information for group A or B. Yeah. From there you can identify which group you are. For people who are online, we will show a few things online in the meantime to show you some videos of the sample prep and live sessions, but the audience here will join outside. Thank you.
If there are few people who have not received the pre-printed batch, that is,
this yours?
for whom we wrote the link, they can also be uploaded in group A and B.
Yeah, from the graph. So,
Okay, that's right.
Who's going to take us to the bioscience?
Yes.
so Professor Graham is also going to that. So,
So from group C and D, those who do not wish to take tea, of course we would like you to take tea, but if those who do not wish to take tea can actually right away then from there go to the bioscience department. So
you can take the first batch.
Ankit is here. He will take first set of people and Megha is there as well. I think she will take the final people. Okay. Those who do not wish to go outside, but rest can join here then leave. So up to you.
Sure.
Yeah. Yeah.
Welcome. Very good to see you. Finally. Come on.
Thank you.
These will serve as a
Ongita.
Everyone, not so alright.
Today you're off for the You're online.
Actually, I got some slides I did talk with you over the email regarding please leave that part. I'll just tell you. It was so that's fine. I'm just fine. It's lots of it was getting from I was in actually some of them so yeah, it's like
You are at the oh yeah. So I
shows you that when we talk about a complex task, something like looking at proteome, right? But it shows you that when we talk about a complex task, something like looking at proteome analysis, which means we are looking at a large number of proteins to be analyzed, and potentially all the proteins to be analyzed from a given organism or given organ or given cell. Then I think we have to come up with different extraction strategies. So the first circle here shows that whether you can have a plant, you can have a cell, you can have any type of human sample, any other bacteria, any variety of sample type. You first want to come up with novel strategies for protein extraction. And once you have extracted protein and you have removed other contaminants, which is essentially nucleic acids, lipids, carbohydrates, salt, then you want to separate the proteins. And for protein separation, which is the next step, then you have electrophoretic methods like gel-based proteomics approaches, including the one-dimensional GS page or two-dimensional electrophoresis or different in-gel electrophoresis type of technologies. Or you also have gel-free methods multiple dimension protein identification technology, or various type of label based methods like tandem mass tag or I-track or isobaric tag for relative and absolute quantification. So again, after you have done protein extraction, you want to separate the protein and also you want to quantify the protein, and that's where a lot of things happens at this stage. And then, of course, in the process, you want to identify what these proteins are, which are looking interesting to you. Either they're coming from the gels, or coming after labeling the sample from the different labeled strategy like ICAT or iTrack. And then you want to identify them using mass spectrometry platforms, whether it is multi-top or ESIQ-top or Orbitraps. And then you also want to do a lot of bioinformatics work to search the databases to find out where is your protein located, and is that you're looking at the right protein. So again, database search and bioinformatics becomes very crucial for any kind of big data field, but more so for proteomics. Once you have done protein identification, now you know the set of proteins which you are maximum interested in, and you want to investigate them further to understand a given biological system. Then you want to move on to the protein characterization part. That's for different type of functional proteomics technologies like protein microarrays, different type of label-free biosensors like surface plasmon resonance, and other label-free biosensors are available, which are helpful for protein characterization. And now once you have narrowed down some of these leads and you are more keen into a very specific protein study, then you can look into the conventional organic chemistry and conventional chemistry techniques using the protein purification methods, using the X-ray crystallography, and do there more of the structural investigation. So I hope you are now slightly understanding the broader picture that for looking at the complex proteome, we have to do the series of experiments, and we have a variety of choice in the process of doing these proteomics-based investigation. But the first part becomes most crucial because there are too many steps and too many technologies which are linked later on. But to begin with, you have to start with the good protein extraction methods. And that's where I think, Dr. Jingre in the beginning of the inaugural talk mentioned in discussion about experimental design. And I thought to bring that point first up front, that your sample preparation strategies are many times linked to the experimental design. For instance, I have just shown one circle here talking about gel-based approaches. Let's say you are interested to take your protein extract and separate them in the electrophoretic method, and you want to separate them either in one dimension with a GS page, or you're looking at two-dimensional electrophoresis-based method. Your sample preparation strategy will slightly change because the buffers which you are going to use, they are going to be more compatible for the electrophoretic methods. And most of the time, we are going to use urea, thiourea, chaps, rehydration solutions, which can contain these things along with bromophenol blue, which can help you to track the progression of the protein movement in the gels for SDS page or 2D gels. But when you're looking at more quantitative approaches like two-dimensional difference in the electrophoresis or 2D dyes, then your experimental strategy again changes, because now you have to think about you need sufficient amount of protein so that your control and your test, they are going to have not only the sufficient amount of protein for running on the gel, but also you should make some internal standard, which I have labeled as IS here, that you need to make the pool of internal standard, which has to be labeled with another dye. And as we go in the the next module tomorrow about gels, we'll talk about what these dyes are of Site 3, Site 5, and Site 2 dyes. But here I'm just giving you an idea that you have to also think about which technology you are going to use to address your question. If the goal was to look at only 2D gel, you're going to look at very huge amount of protein, because each of the gel which you're going to separate on the 2D, you have to run them separately. And the dyes which you're going to use are primarily going to be Kumasi blue or going to be silver stain sometime. But still you need at least 250 to 500 microgram protein, and preferably even 1 milligram protein, which is of high quality. For doing dyes experiment, you can have even 150 microgram or 200 microgram protein, and even that is sufficient. But you have to keep in mind that how many internal standards you have to make. Accordingly, for instance, you are thinking about going directly with the shotgun proteomics approach, and you want to perform a label-free mass spectrometry-based technology. So now you would like to think about, in my experiment, I should have sufficient amount of protein. Let's say you want to compare 50 control and 50 disease samples. So first you should think about, like, you need to have sufficient amount of protein to digest for peptides. And then you should also have sufficient amount of protein from each of the sample so that you can make a small pool of that. And that pool has to be, made sufficient quantity so that periodically you are going to run them in the LCMSMS. Let's say after every 3 sample, you want to run one of your pool sample, which you are going to monitor over the time period, because LCMS runs takes time. Every sample is going to run 2 or 3 hours, followed by same time for the running the your blanks. So in every day, you're hardly going to run 4 or 5 samples. And if you want to compare now 100 samples, you have to go a couple of weeks time to just run one project. So given the changes happen and so much reproducibility issues comes in any technology, it's good idea to monitor the reproducibility of the instrument and also the intensities of these peptides so that you can use that for normalizing over the time period. So again, think about experimental design and strategies. That's very important, because your amount of protein which you want to extract from any sample type, you have to think these things ahead of time, that how much protein you need. And in this case, when you're looking at label-free quantification, you also think about what is going to be your internal standard, which you might want to spike in along with your sample of interest. Or sometimes people also use the retention time peptides, which is IRT peptides, so that you can monitor the chromatography, how reproducible it is coming on a specific time when the peptides are diluted out from the LCMSMS column. So again, many of these things may not be very clear to you right now, but it will become more and more apparent as we keep going in more detail about each of these technology and we talk about how that works. But I'm just trying to give you idea that it looks very interesting to think about. I want to do proteomics, I want to do quantitative proteomics, and I want to perform any of these experiments, but we have no idea that how crucial the experimental design is going to be, how crucial the sample preparation is going to be, and how much thoughts we should give in beforehand that is going to determine the success of our experiment. So for instance, now when we look into label-based mass spectrometry or quantitative proteomics approach using labels like iTrack or TMT. So iTrack and TMT, they both bind to the peptides on the N-terminal sequence, and you label them with these reporter ion-based the tags, which is tandem mass tag or isobaric tag for relative and absolute quantification. So once you want to quantify your samples, you have to think about again, how are you going to analyze the data? How are you going to ensure the reproducibility of the data? So a simple experiment for this type of workflow could be, think about a 4-plex iTrack experiment, right? And you have 114, 115, 116, 117. These 4 reporter ions tags, which you want to label your 4 conditions. So you have a control, and you got a time course experiment. The sample taken out after 2 hours, after 6 hours, after 24 hours, and now you want to compare all that with the control condition or 0 time point. So now you are looking at labeling all of those 4 with the reporter ions from 114 to 117, and you'd like to now compare them across. So while doing so, this is your one replicate. Now you have multiple replicates which you want to compare, and more your conditions are going to be increasing. So now when you want to cross-compare your one labeling reaction with other labeling reaction, again you have to think about how are you going to compare the intensities of one reaction versus other reaction, and how to cross-compare that. That's where making a pool which you can use in one channel is very important. So in this strategy, if you are looking at 114, 117, 4-plex only, you have to make one of the channel as the common reference pool, which you are always going to have a common sample which you are going to run. And that common sample, you are going to monitor the intensity of that reporter run of 114 in multiple conditions, so that you can cross-compare your different quantification of different conditions from one reaction to other reaction. So I don't want to afraid you that we are talking about many technologies right now, but I only want to convey you one single point, that there are many technologies in the proteomics field, whether you talk about gel-based approach, electrophoretic methods, or you talk about mass spectrometry-based methods, which is either label-free or label-based. In all of these, the considerations are on the experimental design, which is very crucial. Otherwise, you will do so much hard work, a lot of data will be generated, but you can't make it publishable, you can't make it of the conclusive quality. So when we talk about the sample preparation, given the audience who is all part of this workshop, we have very heterogeneous group of people who are coming from a variety of background, and we have to think about every biological sample brings their own specific type of challenge, very much uniqueness from that sample type. For example, plant cells, they're very harsh tissue. They have a lot of interfering substances. Plant leaf will have ribisco type of abundant proteins. Plant roots will have polyphenols and many other components. Many plant cells have a lot of pigments and colors. All those are going to be interference, which we have to remove if you want to prepare that sample to do the proteomic analysis. Fungal cells, again, is very tough sample to work with. Lot of proteolysis challenges are there. Bacterial cells have very high ratio of nucleic acid, which you want to remove before you move on to your proteomics work. When you want to look into clinical samples like cerebrospinal fluid, is very dilute sample which is having very high salt concentration. You want to first remove that, and also there is a trace of lipids which you want to delipidate. Then only your samples are ready. Your biological sample, which is very commonly used, is serum or plasma, which contains abundant proteins like immunoglobulins, albumin, and salt. So you have to remove all of these before your sample can be used. You are doing a lot of tissue culture based experiments. For growing those, you are using the buffer, you're using the salt, you're using the serum, and all those are coming as a part of your sample, which you have to get rid of, or doing proteomics. So I hope you got some idea that there are different types of samples, and I've just listed a few. I'm sure you can come across with your own sample type, which you are most keen to work with, and you will yourself understand that there are challenges of handling that sample. And this is where I think a user is best person who knows about their biological questions and biological samples. And you have to be intellectual enough to understand that what are the interferences which are going to come on your way if you want to prepare very clean and good protein sample, because everything else has to be removed from that sample except protein to take it forward. So let me focus a little bit more on the clinical proteomics part. That is something I think large number of participants are from clinical or veterinary background, and even we do a lot of work in this area. So when you are planning for any kind of clinical proteomics based work, even prior to starting your study, as I said, there are two things. One is what is your biological question, what technique you are going to use. That is first thing to decide at which technique you are going to be using, and how best you can address that with your sample preparation strategy. But also, when you are planning to collect your sample, you have to keep in mind that there are many analytical variability are there, and you want to first look at what could be pre-analytical variability as well. So, for instance, many factors which are going to affect your sample collection and sample storage, those are all pre-analytical factors. Even before you start your project, you are going to collect many samples, and the samples are going to be having the challenges of how best they were collected and how best they were stored. So there are too many factors over there only for thinking about this aspect. And there are, of course, many factors, some of those which we have no control, like intrinsic influences of gender, age, city, etc. And extrinsic influence also, like, your participants who are going to be enrolled in the study, what type of diet they have, what medication they take, what is their smoking or not smoking behavior, alcohol consumption behavior. So again, many of these things should be part of your clinical data record forms, where the participant, if they can disclose these information, that could be very informative, because you are, let's say, looking into something called metabolomics and analysis, and a set of participants are consuming alcohol, and then you are very happy that you see a lot of set of metabolites are very changing in those individuals, but those may not be influence of a given disease if you are investigating that, but could be just because of the intake of certain specific type of food behavior, right? So that's something which we have to keep in mind. If possible, as detailed clinical record forms or study design forms are made, that can minimize a lot of issues at the end when you're analyzing the data, because you'll always find there are some outliers. There are some individuals which are showing variability, and you have no idea, is that biological or is that because of anything unique to that individual, right? So that's where you have to keep many of these issues. Of course, there are many technical pre-analytical factors. For instance, how you are going to collect the sample, process the sample, and store the sample. Because many times you are handling the sample, which are biofluids, which are going to be degraded very fast if you do not collect them properly, do not process them quickly, and do not store them in good -20 or -80 type of conditions. Even sometimes the sample collection mode, like the different type of clinical sample, what is the posture of the patient when they are collecting the sample, or even something as simple which we even do not pay attention, that what type of containers and tubes which you are using for sample collection. You might be using, based on availability of the stock of the tube, one set of, sample of 50 patients you collected in one tube type, and that tube stock got finished, and now the next batch of the individual which are supposed to be controls now, you are collecting in a different type of tube, and now you are going to compare them for proteomics investigation, and you see, wow, big change in my control versus disease, and that's all coming because of the type of tube and not anything specific, what the biological questions, you want to address. Again, many of these things can be confounding, and these are very, simple things, but these are very crucial at the time of sample collection. So it's a good idea to make some sort of chart to think about do's and don'ts, and think about if you are planning for a bigger study, how best you can get the reagents and resource which is sufficient for that whole project, so that you don't have to keep changing your things in between as you go along. Otherwise, you'll have the problem of lot of reproducibility issues, which is not biological, but because it is of the pre-analytical factors which are creating those kind of variability. Many times, a lot of tubes, even the plastic quality is bad, and you will see, those components are getting shedding in the directly in your sample. When you are processing that in the mass spec, and you can see those traces, and those can be very confounding. Even people have shown some study of the cerebrospinal fluid measurements when they look at beta amyloid and tau proteins, and they found that they differed when they collected in the tubes of different type of material. So same sample when you collect in different type of tube, the behavior, what you measure for beta amyloid and tau proteins, which are aggregating very fast, you are going to see them very different. These are some experimental type of work which has shown that why it is so important to pay attention to a small thing like the quality of your tubes, the storage conditions, right? And again, I'm sure all of you are quite experienced in the field, but you have to keep in mind that multiple freeze are going to be very detrimental for any sample. So while you are collecting the sample, it's a good idea to make multiple aliquots of the sample, so that those samples could be stored in different boxes, and you are going to only handle a few boxes at a time, and not you are going to change all the time your level itself. And all of those are a way to actually ensure that there is no analytical variability, and you are trying to normalize those, you are trying to combat those type of variability. But how are you going to change these things? Like, think about some serum samples, some technicians, they have collected, and they kept it on the room temperature for 5-6 hours, which is very common. And some samples were immediately kept in 4 degrees. Some samples are kept in -20. Eventually, you got a stock of 100 samples, which contains all the 3 types. All of them have already got lot of variability to start with, and you are not going to at the end going to find anything which is very reproducible, because you have lost too many things in the statistical analysis while considering all these samples from the same type of patients. So I think your clinical collaborators, your student staff, your seriously that there's a lot of investment you are going to put in, a lot of time investment you are going to put in for analyzing the data and trying to make sense of the data, but that will not make any sense if these parts are not done carefully. The first 2 parts, what is your biological, for which technique you are going to take, which platform you are going to use, what will be the strategies in that platform. Second is your input sample type, how well it has been collected, and how well you have removed the variability. Again, just for a practical experience, what we try to do for any project when we start something of the clinical project, we do try to buy the reagents in bulk. We do try to buy the plasticware in bulk, and try to provide to our clinical collaborators the same reagents, so that at least that much variability can be reduced. We also do try to optimize the conditions in our lab here, and try to first mimic that in the collaborating hospitals. Then only the samples can be collected. But again, despite all that, you will have still some variability which you cannot avoid, given the nature of the samples, and you're talking about the patient samples, you're talking about the people which you have no control on, and you will have some degradation and stuff, but you have to avoid that as much as possible. Also, like if you're looking at clinical samples, good idea to reach out to the protein extraction and peptide preparation as quick as fast as possible. Because your raw sample, crude sample, which you are storing, whether it is serum or cerebrospinal fluid or tissue or any sample, is going to degrade. Whatever you do, your box is not open, but your colleague is going to open the freezer multiple times, and the freeze thaw will happen at least partially, and you will see progressive degradation of the sample stored for a long time. So it's good idea to, when you are collecting the samples fresh, good idea to process them and try to get the protein, followed by digest them to get the peptides. And all of that is ready for you to store them in your -80 freezers. Those will be highly stable and high quality samples which you can analyze over the month's time. So with this, I hope you have conveyed you two important points, that proteomics is not one technique and not one workflow. You have to think about which question, which technique to take forward. And second, as carefully you can do your sample collection and storage, that will be highly beneficial for your next set of activities. So coming to the sample preparation in proteomics, we are talking about variety of things here, whether it's, we are looking at, whole yeast proteome, whole organism, or looking at only specific, type of tissue, brain tumor tissue, or look at any specific, fluid, like, serum, plasma, CSF, urine, saliva, nasopharyngeal swab, variety of samples, or looking at different types of cell-based work. In all of them, sample preparation is crucial, most important, and very different. Very different type of strategies has to be taken forward, and as reproducible you can prepare these samples from different replicates, more and more meaningful data you will get from the technology which you are going to employ later on. So this workflow is going to involve multiple steps and not in the same order for every single sample type which I showed you, but you are going to primarily look into how best to lyse your cell or disrupt the cell, how to protect them from proteolysis, how well you can fractionate the complex sample to make it simplified fractions from, so that you can get the better sample output from those, protein extraction and solubilization strategies, removing the contaminants. Contaminants could be many things, your abundant protein to the salt, to the pigments, or for that matter, nucleic acid, lipid, carbohydrate, anything is contaminated other than protein. And you have to think about carefully how to make sure that those things are minimized or removed. And then, of course, protein quantification before you are ready for your next set of steps. So often you will use different type of precipitation procedures in proteomics workflow. Acetone is commonly used, when you can simply use 3 or 4 volume ice cold acetone, and which is really chilled at -20, and then you allow that for the protein precipitation with 1 to 2 hours of incubation. You do the centrifugation and remove the acetone, and that is quite a quick and efficient way of removing a lot of lipids and detergents which are present in your sample, and then you can proceed for that type of sample for further analysis. Or you can use TCA, trichloroacetic acid, for precipitation, very effective protein precipitant, and 10% TCA is quite efficient for precipitating the protein. If you incubate that in ice for 30 minutes, then you are going to get the protein pellet, which you can wash with acetone and ethanol, and you are going to have good sample recovery, so you will not lose much sample with this type of precipitation method. Often people use protein precipitation with TCA and acetone together. So now you weigh the TCA with weight, and then put that in acetone in solution form, make that mix, and then you are going to lyse your cells in 10% TCA with acetone, which also contains the dithiazolidine to break open the disulfide bonds. And then you are going to incubate that for 1 or 2 hours in -20 degrees. Then you are going to wash your pellet to remove the TCA, and you can wash with acetone alone, which contains the DDT. And in this manner, now your protein pellet is pretty clean, and that is very commonly used strategy for sample preparation. So just to summarize this part, I think you will often see, again, if you are not sure about what type of sample prep you should be using, you should try out some of these methods yourself and see, like, after extracting the protein from each method, when you run them on the gel, what kind of quality of the protein you can see. When you quantify the protein, what amount of quantity you can get from these type of strategies. And this is how you are going to develop a protocol. The protocol development of any sample type should be explored with variety of methods which you should be exploring simultaneously. Over the time period, what we have found that various type of projects, the cancer projects, they require tissue-based investigation, and given the complexity of these tumor tissue, followed by you want to minimize the steps required before you can perform the mass spectrometry analysis. I think urea lysis method is one of the efficient way of moving forward. And here, what you do, like you have the fresh frozen tissue, which you always want to go as minimum as possible. In our personal experience, you can go even as low as 5 milligram of tissue, which is properly frozen and snap frozen in liquid nitrogen. But if you can have 20 to 25 milligram minimum, that will be ideal situation. Of course, if you have 100 milligram, that will be really good to do sufficient protein extraction and store it for the future as well. But even if for 1 or 2 experiments, as long as you got, many times we get the, the sample from the little biopsy, very, small amount of tissue. But even that is possible to do as long as we can get even 4 to 5 milligram of tissue to start with. Now, in that, you can add the urea lysis buffer, which contains 8 molar urea, tris buffer, sodium chloride, magnesium chloride. Then homogenize the tissue with the sonicator and bead beating, followed by you can do centrifugation step, and then you can collect the supernatant, which is having a protein, and that can be taken forward for the next steps of quality control check. We had this workshop last year, the DST workshop only, where John Wilson from Profiti, he came from New York, and he was involved in the sample prep strategies workshop part. And I know he made a very nice comment that this entire procedure of sample preparation should be carried out in ice. It should be cold and fast, which are the cardinal rule of proteomics. So of course, when you're working on the proteins, you have to try to keep the things really tight. Everything should be prepared in advance, and try to do everything in the cooling conditions so that you can minimize all the degradation. Otherwise, those will be difficult for you to overcome in the analysis stage. Let me show you this brief hands-on demo activity of urea lysis method, how to prepare that.
Hello everyone. So for the success of any proteomic experiment, sample preparation is the most crucial step. Sample preparation involves protein extraction, protein digestion, sample cleanup, and final mass spectrometry. Depending upon the experimental objective, a type of biological sample, different protein extraction protocol can be used. Different biological samples could be bacterial pellets, cell pellets, or tissue samples. Today, we will demonstrate you the protein extraction method for a tissue sample. Here we have already weighed amount of tissue. For this, we will use urea lysis buffer, which is 8 molar urea. Appropriate volume of this buffer is added to the tissue. It is vortex for a few seconds and spin. After addition of buffer, the next step is homogenization, which ensures that tissue is completely live in the given buffer. So for this, now we will use sonication. For this, we are using probe sonicator. Whole procedure of sonication should be performed on ice. So after this sonication, all of the protein will be extracted into the buffer. After sonication, it should be made sure that whole content is uniform and nothing is left without lysing. If any chunk of the tissue is left, sonication step can be repeated. Now the next step is to centrifuge so that we can get the clear supernatant. Centrifugation can be done for 15 minutes at 8,000 times gravity at 4 degrees. After centrifugation, tissue debris are settled down and clear supernatant can be collected. Now the supernatant should be collected in a clean, fresh appendix. This supernatant has all the proteins of this tissue. So this is the collected supernatant that have all the proteins. After getting these protein samples, now they can be taken for the downstream process such as protein quantification and quality check, or they can be stored for further use.
That how simple it is, and this is one of the very efficient way of getting very high, protein yield from the sample type, especially the tissue type of samples. But often you will be using other methods as well, and so I'm going to give you glimpses of some other methodologies and a very short, glimpse of different type of methods which you can use. Not necessarily that the urea method can be only used for the tissue. It can also be used for the cell lines and bacterial pellets as well. But likewise, trizol extraction method I'm going to show you with the bacterial proteomics type of workflow. But it can be used for other type of sample, including tissue as well, right? So you can try many things. I think the good thing is, if you are not your lab has not worked the proteomics earlier, and you have no prior background and idea for what is the best workflow for doing proteomics in your lab, it's a good idea for you to first try out different type of extraction methods, see which one is the easier and also provides you high quality. Looking at the different type of quantification methods. The trizol method is one of the method which we found quite easy and good to work with, and many of faculty participants and students who are here might have already used trizol method for doing the RNA extraction. Then the phenol and chloroform which is used in the method is going to dissolve the lipids and polysaccharides and going to help in the phase separation for the extraction of the nucleic acid. Guanidine isothiocyanate is going to break open the cell membranes and inhibit the ribonucleases and proteases. Therefore, it has been highly used for the RNA-based for the denaturation. So in this method, we are going to use different phases, different layers, and that will be helpful for us to do sequential extraction. So think about, you got some, clinically important sample, and from same sample you want to perform the RNA, DNA as well as the protein-based work, and now you have very limited amount of tissue or sample available. So trizol in that way can be very good way because you're already assured of good quality of RNA which you will get. But from our experience, they've also got very good quality of the protein from that. So the first phase, which is the chloroform phase, that contain the RNA. The interphase contain the DNA, and organic phase will have mostly the proteins. So you can actually separate these layers, and with that you'll be able to get the same sample RNA, DNA, and the protein. Let me show you briefly how this experiment can be performed.
Culturing. Clean the laminar workspace thoroughly with ethanol and keep the master plate having the bacterial culture ready. Light the spirit lamp or Bunsen burner to maintain sterile and aseptic conditions throughout the process. Carefully remove a bacterial colony from the master plate and inoculate the autoclaved growth media. Incubate this inoculated sample at 37 degrees Celsius for 6 to 8 hours with constant shaking to allow the bacteria to grow. The turbidity of the culture, which gradually increases, indicates bacterial growth. Culture harvesting. Transfer the grown bacterial culture to a fresh tube
under sterile conditions. Centrifuge this tube at 12,000 rpm for 10 minutes, maintaining a temperature of 4 degrees Celsius. Transfer the pellet obtained containing intact bacterial cells to a fresh microcentrifuge tube. Wash this pellet thoroughly with phosphate buffer to remove any unwanted debris. Sonication. The resuspended pellet is sonicated on ice to enable the bacterial cells to break open so that its contents are released. Sonication involves the use of high-energy sound waves that are capable of breaking open the outer membranes of cells. All cellular contents, including proteins of interest, leak out of the disrupted membrane. Carry out the sonication procedure for 30 seconds with a pulse of 1 second at 40% amplitude.
It shows you that when we talk about a complex, something like we have not worked on, we start very unbiased way to try out different extraction methods. And that may involve urea lysis or trizol extraction, different way of TC acetone precipitation, or it could be a mixture of some of these. So variety of things we try, and then also look into the publication that from which method, what kind of number of protein people are able to identify. And based on those considerations, then we take one workflow forward. Of course, if you think about proteomics, what we have to see, what is, because then only you are doing something at the proteome wide scale analysis. So that's where, one method cannot be 100% sufficient to provide you all the knowledge, all the glimpse of the entire protein. And what we are talking right now is about global protein analysis. Think about possibilities where you are looking at only phosphoproteome analysis or any type of specific post-translation modification. I request everybody to mute themselves. Some background income. All right, I request everyone to keep themselves muted. So, what I'm saying that, whenever you are starting a new project, new protocol, it's good idea to try compare different methods. And sometime when we think about very specific objective like PTM analysis, then your preparation, sample preparation, enrichment strategy is way too different. So we cannot just employ one method and say we can do proteomics using that method for all type of samples and all type of applications. So we have to be very given that plasma represents a lot of protein leaching from all type of tissue to the blood, and then it is very easily accessible from the blood, and it is most commonly being used for all kind of biochemical parameter measurements in the clinical settings. So even a small volume of the leftover plasma can give us a lot of insight about a disease at the proteome level. Also, if you think about if you are that biomarker from the plasma sample, then that could be very powerful to get you an insight about that disease, and also it can be clinically applicable because the test could be developed from that. Again, lot of ELISA kits can be made easily looking for the proteins from the plasma sample. So plasma proteomics is one of the very attractive area to investigate.
ठीक है मैम ये बताइए कि हाँ
involving the UV lysis method, the measurements before you process those samples. And even any other biological sample for that matter should be processed with the extra caution and care. And while it is not within the scope of this workshop, but biosafety considerations, performing your study with ethical standards and biosafety is highly required, which I think should be discussed among the participants. And if needed, sometime we can also intervene and give you some clues about how to do that safely. We spent a lot of time recently about just planning for these experiments on the COVID because of the how to do it, to the best safety possible manner, and I still try to move ahead with these projects. So these will be the kind of challenges which you will face many times, and you have to see what is the end goal and objective. In the COVID project, we had no idea for what peptides and proteins they're going to look for for disease severity prognosis. So we had to start from scratch, and that's where we needed to do depletion mandatorily, because otherwise we're going to lose all the low abundant proteins. We have no idea for this disease that what can be potential biomarker. But after we have done these experiments, and now we know these are some set of proteins which are very interesting. Now the targeted validation could be done only on those peptides, and now we can also use the crude plasma, which is undepleted. It is possible some peptides we may not see them if they are low abundant and not enriched sufficient, but most likely we will be able to see most of the peptides. So please keep in mind about weighing your many of these things I'm talking is the real practicality, not just kind of lecture. But the practical issues are very important because your cost is very prohibitive, for doing your large number of samples. So discovery work can be done on the maybe smaller set of samples, 30, 40 samples, for which you can buy these kits. You can do the depletion, you can perform the discovery experiment nicely, and you have already taken care of all the pre-analytical variables, which I spoke in my previous lecture, and made sure that everything has been stored in immediately and process all the samples in a week time. So you have done really everything meticulous way. Now even these 40 samples, if you do depletion, they will give you very reproducible result. Now we have identified a set of potential candidates which you want to take as a lead forward, then you can come up with a new strategies for validation. You don't need to do depletion for every single thing. Validation strategies could be based on the ELISA-based test, if you know for abundant protein antibodies available, or it could be based on the target proteomics-based workflow. So I hope I'm giving you the balanced perspective of how to weigh your options and still move ahead with the cost-effective manner to do the large-scale proteomics projects. I'll just talk about another sample, which is an interesting sample we had never worked on, nasopharyngeal swab sample, right? And I think most of us had not even heard about this to do proteomics until the COVID came. And recently, because of COVID, when everywhere the nasopharyngeal swab or otopharyngeal swab samples were being collected for real-time PCR analysis, that became a sample of choice for us to think about, can we do proteomics on that? And now for doing this type of project, think about situation like lockdown, extremely labs are all shut down, and we are thinking to work on a sample which is very unsafe, very difficult sample to work with. And these swab samples are being collected in the a tube which contains viral transport medium, VTM. Now this VTM comes from different vendors having different compositions. It also contains sugar, it contains albumin. So now very challenging sample to work with. And this was a real test, how best to do something on this type of sample in a safe manner. So that's where I'll show you, this brief clip of initial processing of the swab, but I can have some discussion about how to take this type of things forward. So now in this situation, the first consideration was, before doing any experiment, can we inactivate the virus from the sample, right? And at the same time, we don't want to lose our
audio is missing.
Is that problem for everybody, or audio? I am speaking, right? So
no, sir, it's fine actually. Maybe it's for a few people, but it's, your audio is totally fine.
All right, good. So what I'm
still
showing
no audio.
First nasopharyngeal swab sample, we are keeping them in the heat incubator for incubation, so that virus can be inactivated. So as per WHO guideline and some Lancet papers, if you keep these sample at 65 degrees for half an hour, that is safe to work with. And now what we did, we kept the whole assembly, including our trays and including our racks, so that if there's any spillover or anything, it could all be inactivated. I am requesting all the participants, please keep your background muted. Okay, thank you. So this sample, as I said, the swab sample was challenging sample to work with, and first consideration was to inactivate the virus. Actually, we did the same thing with plasma as well on the viral, the COVID patient samples. So once this inactivation was done, then thought was how to get the protein out of it, because, and also we had to think about something which can be clinically applicable. We don't want to go with very long routes. For example, in the urea lysis method, once we go ahead in the next step, I will talk to you that we have to dilute that urea, because that is going to otherwise have problem in the digestion process. So in that way, we thought about take some of the organic solvents and try to precipitate the swab sample and see if we can get sufficient amount of protein from different type of organic solvents. I was talking to you about different precipitation strategies of acetone, TC acetone, variety of things we tried. And we thought to keep it as minimal step as possible to do this type of work. So I'm just giving you a nutshell here of sample preparation strategies. One for the plasma, which I spoke just now, that we did heat inactivation of the plasma, followed by depletion of the abundant proteins. And then we ran on the gel, SDS page gel, to look for the how the depletion has worked out as compared to the crude plasma or the undepleted plasma sample. And that looked like, quite reduced intensity, although not totally removed, but quite a bit of abundant proteins have been removed. And likewise, we tried the swab sample. And in this case, from the VTM, we tried 3 different methods. Now, this one is much more infectious sample. So we did heat treated longer and 65 degrees. And samples are in the acetone, ethanol, or isopropanol, 3 different organic solvents were used. And we had no idea that what is going to work out for us, because of there was no literature available for this type of sample to work with. And so we just tried independently, and thought was, even if a patient is in a remote area, somewhere in the rural area, and you want to collect the sample from the patient, one of the bottleneck was for the real-time PCR work, that the sample has to be collected and stored at the 4 degrees for doing the RNA-based work in the cooling condition. We were thinking peptide being, if you are measuring peptide only, you don't have to amplify the peptide. So as long as we can get the protein precipitates, then one could take that forward and do the digestion to get the peptides. So in that way, whether in the rural areas or for looking for asymptomatic screening, even if the patient, we give patients that, or individuals, these tubes which contains ethanol or isopropanol or acetone, if they can spit, or if their swab can be collected, those protein can be precipitated in the organic solvents, and those could be sent to the facilities to test out. And that is actually, quite attractive strategy, given the nature of the, the screening which is required, and also thinking about the safety of handling these samples. So, so we tried 3 different methods. And after doing these extraction methods and running in the mass spec, we realized that even the viral peptides, some viral peptides are more seen in acetone condition, and some are more seen in ethanol condition, some are more seen in isopropanol condition. So it means if we are looking at nucleoprotein and spike protein, membrane protein, replicase protein, various abundant proteins of virus, and we want to detect virus for detection test, one extraction method may not be best strategy. So, and given these are all very cheap organic solvents and very safe to work with, what we thought, precipitate the, take a small volume of swab, precipitate the protein, and after precipitation, then pool that, so that now we enrich the viral peptides, because now we get the more viral peptides coming from this same swab sample from that patient. And then proceed for the urinalysis, and followed by do the trypsin digestion and desalting step. So again, you can think about how to come up with the newer methods to work with. For something like this, we had no prior idea, no background, and we thought to explore some new things, so that in the process we can also develop some safe method and quick method to do these type of investigation. And this method actually already being patented. Now we have filed patents for both of these. And also one of the studies just got published. So I'm not talking right now about this work and what we did and what we got out of it, but just giving you a glimpse of it, that different samples require different strategies, and you have to try out new things and newer things to really make some, really insightful idea from that sample. Let's move on to some plant proteomics based sample preparation. This is one of the key area. I think a lot of agriculture scientists, a lot of plant scientists, food people, they all work on plant samples. And given the diversity of the plant and having variety of interference they have, it makes it attractive but very challenging sample to work with because of the nature of the lysis, which is very harsh lysis required. You have highly abundant proteins like ribisco if you want to work on the plant leaf and want to investigate the effect of infection, what pathogen is infecting a plant leaf. You have to remove those highly abundant proteins like what we did for plasma. So you have to do in that case the ribisco, anti-ribisco column you have to use to eliminate those proteins. Likewise, if you're working on the other sample type from plants, you have to remove the chlorophyll, you have to remove the carotenoids, very coloring agents, pigments, or you are looking into the roots. Those are, of course, you have to remove the mud and soil and stuff, but also you have to remove the phenol and other interference which are present in the root. So likewise, you have to think about the good extraction method for doing your plant proteomic sample. But I'll show you one method which works usually well for the plant proteomics type of workflow. So let's watch that demo.
Select the plant leaf sample of interest and weigh around 300 mg of the leaf on an aluminium foil. Transfer these leaves to a chilled mortar and carefully add liquid nitrogen to it, which helps in drying up the leaves instantaneously. Grind the leaves well using a pestle to obtain a fine powder. To this powder, add around 0.5 ml of lysis buffer containing trichloroacetic acid, acetone, and dithiothiotol, and grind it well until a fine paste is obtained. The lysis buffer causes the plant cells to swell and finally break open, thereby disrupting the membrane and releasing all its intracellular contents. Add another 1 ml of lysis buffer to the paste and then transfer the solution into a fresh tube after grinding thoroughly to obtain a uniform mixture. Centrifuge the sample at 10,000 rpm for 5 minutes and maintain a temperature of 4 degrees Celsius during the process to ensure that there is no denaturation of the proteins. Discard the supernatant and retain the pellet containing plant proteins along with various other intracellular components. Incubate the pellet at -20 degrees Celsius for an hour. Protein precipitation. Remove the pellet from -20 degrees Celsius and add chilled acetone to it. Mix the sample well. Centrifuge the contents at 4 degrees Celsius for 5 minutes at a speed of 10,000 rpm.
Work involving the UEL lysis method. And this is watch that demo.
Use the sample at 10,000 rpm for 5 minutes and maintain a temperature of 4 degrees Celsius during the process to ensure that there is no denaturation of the proteins. Discard the supernatant and retain the pellet containing plant proteins along with various other intracellular components. Incubate the pellet at -20 degrees Celsius for an hour. Protein precipitation. Remove the pellet from minus 20 degrees Celsius and add chilled acetone to it. Mix the sample well by vortexing to obtain a uniform solution. Centrifuge the contents at 4 degrees Celsius for 5 minutes at a speed of 10,000 rpm. Discard the supernatant and repeat the acetone washing at least 3 times to remove all plant pigments. Obtain protein pellet at room temperature. Reconstitute this pellet with rehydration buffer. Vortex the sample to obtain a uniform solution. Centrifuge the contents the following morning at 10,000 rpm for 5 minutes. Collect the supernatant containing proteins in a fresh tube and store it at minus 20 degrees Celsius until protein quantification is.
All right. So I think we have seen a variety of ways now how to do your different type of sample preparation. During this process, whether you talk about plant or human plasma or human tissue or bacteria or yeast, any of these samples, you will have a variety of interfering substances. For example, salt, you will see it all the time coming from the buffer. And very oftenly seen in the case of urine samples, plasma samples, or even cerebrospinal fluid, that are highly enriched in the salt, and you have to remove that either by performing dialysis or different type of gel filtration methods, or even when you are doing the TC acetone precipitation, sometimes when that removes it very easily, and you can do subsequent washing steps. So many of these are the ways to remove your contaminations, but you are going to see that. And if you are going to plan your two-dimensional electrophoresis type of experiment, this is going to be quite crucial, because your IPG strip or immobilized pH gradient strip, they will stop to process your sample for the isoelectric focusing to happen, because so much voltage will rise and then go to stop, and as a result it can't move in the electric field. So that is one of the important thing to consider if you are planning for the two-dimensional electrophoresis experiment. Or even if you're thinking about directly shotgun proteomics of using LCMS based approach, think about that you have the nano LC columns in this case, when we are using everything in the very nanoliter volumes. Now these kind of contamination can clog your column, and each column is so costly, like in the mass spectrometer, the major cost comes from the cost of these analytical columns. If they can if they are getting clogged because of your interfering substance, you're going to lose 1.5 to 2 lakhs rupees cost, just because of your sample was having some interference and some contamination, and could be salt or could be nucleic acid as well. So these things, if you can remove them well and do the desalting step properly, both at the protein level and at a peptide level, I think you are really safeguarding not only your project but also your instruments and your reagents, because these columns and these instruments are quite sensitive to these contaminations. And when you are looking into nucleic acid contamination, sometimes your protein solution which you got is highly viscous, and that's where you can get a sense directly that this sample is probably not the best sample to work with, and you need to do more of the nucleus treatment, DNA that mixtures to be incubated and again washed off. Polysaccharides are another challenge. Sometimes you will see different glycogen and starch. They are going to be coming along with the protein. They're highly bound. So you have to think about different ways of precipitation and cleanup steps, like ammonium sulfate or using the trichloroacetic acid based precipitation methods. Lipids, again, the different type of tissue sample when we work on the brain tumor tissue or cerebrospinal fluid. Variety of these samples are having high lipid enrichment. So delipidation with the organic solvents can be very important in that case. And then, of course, there are different detergents which you are using, like organic detergents. SDS is going to bind very strongly to the complex of the proteins, and that has to be removed before you do your in-gel. So again, you have to keep in mind that can you use some of the deuterionic detergents like Chaps, Triton X100. Those could be more useful for you to avoid the contaminations. And of course, as I said, the interference could be very subject to the type of sample which you are working on. There is an endless list to it. Lignins, polyphenols, tannins, alkaloids, pigments, variety of things from the plant samples which are interfering as well. But one more thing which is crucial here to think about along with all these interfering substances, the way we process our sample, right? Simple things like, you're wearing the gloves all the time and adding some sort of mask and the face shield and small, these kind of practices could avoid the keratin contamination, because, the things coming from the skin or hair fall, those are the ones which we see very, prominent and, the most abundant peaks in the mass spectrometers. And as a result, you are going to lose out your less abundant proteins. So if you can avoid those contamination, then I think your sample preparation is going to be really good, and you can make use of that for your further mass spec or other proteomics experiments. Now, given that what we are talking is highly complex samples, which is having thousands of protein to investigate. So, of course, problem is complex, so we have to deal with it, and let's see how best we can keep the environment cool and background less noise free to proceed to the next system, which is quite complex of handling the samples. So now what we are looking into, thousands of protein which we want to separate. And after all the hard work of extraction, now we have this tube which contains these clean samples with thousands of protein. But if you are going to take that entire proteome and proceed forward for doing your mass spec or other proteome experiment, you might lose the coverage, because proteome coverage will depend, upon, how best your instrument can really read those peptides and the ions coming from the peptides. So, fractionation strategies are another thing to consider while planning your proteome experiment, how to fractionate my sample. And that's what I was going to show you on the slide, that the different type of sample fractionation strategies are very important. One could even, as I said, even sometimes you do not have, these columns which you are buying from the commercial ways. You can use some authentic chromatography. You can get rid of the abundant protein from the peaks, and you can also, fractionate your sample using electrophoretic method. Of course, we do IEF for the 2D gels, but also you can use off-gel fractionation, which is known as liquid phase IEF, so which is another specialized setup from Agilent, off-gel fractionator. But that's pretty, easy to work with, where you can have your protein in a solution form, and you can separate them in the isoelectric focusing unit in the liquid solution form in the cups. And then you can collect, if you have used 3 to 10 pH IPG strip, now that all the cups are, for different pH range which you have provided, and you know that between 4 to 5 pH range, these are the cups where you can collect a solution. And likewise, you have, all the different pH, different proteins have moved into respective PI values. So liquid phase IEF or off-gel fractionator is another way of fractionating your complex sample, also based on the pH value. So you know that these protein fractions are for which pH range. If you're looking at some more biologically interesting protein of a specific pH range, you're only keen into the basic protein or acidic protein. This could be another interesting way to separate your complex proteome and fractionate that, and only run those fractions where you have maximum interest to look into your proteins of interest. Of course, conventionally people use a variety of other methods, including differential centrifugation methods, sequential extraction methods. All of them could really, make different layers, and you can separate your complex proteome mixture into different fractions, which can be taken forward for subsequent analysis. I think in the interactive session, somebody mentioned about the working on the algal samples. And so I just thought to show this slide that, this is the kind of project which we had not worked very intensely in the lab. This is a nanochlorobsis sample. And again, when you do not have good idea for the sample which you are working with, good idea to try out different things, so that like which method is going to work out well. So we had not worked on the nanopyropsis, so we tried out trisol extraction method, phenol method, TC acetone method, and very interestingly and surprisingly, we found that each of these methods are giving some unique number of proteins. All three of them are giving substantial number of unique proteins from each methodology. And the way each extraction method works, some is going to enrich your membrane proteins, some are going to remove lot of bound proteins, some may have done, the certain bound fraction. And likewise, you are going to enrich sometime, different transcription factors or kinases. So then you are going to have differential expression of different proteins and enrichment if you try different extraction method. So that you cannot use just a logic that is what I'm going to use. You have to try out different things and see where you can get your most of the proteins possible. So by looking into this type of data, and just recently we are doing a project on the breast cancer samples, and we are trying to optimize that what can be the best condition possible, and results are something very similar. When we are trying 4 or 5 different methods, we can see that there are set of unique proteins which can be enriched from a unique method. And therefore, if we can afford our time and effort to analyze the sample separately, or at least extract the protein separately from each method, then that is going to enrich our proteome. So many times, if you think that you want to claim for that you want to do a proteome-wide investigation, so your goal is to be as many protein as possible, right? For example, you read Professor Mathias' month's paper, and the goal over there will be to reach out to closer to the whole proteome in single shot run, right? They showed very nice papers on the yeast, for example, in single shot, 2 hour, 3 hour of injection with same sample, unfractionated, you can totally try to map the whole proteome. And likewise, so your goal ideally should be to map the whole proteome from a given sample type. So if that is a goal, then you'd like to employ different extraction methods so that you can enrich the protein from different methods. Then you can pool those proteins and make it one pool, which now contains the pool of the sample to take forward for your further downstream analysis for proteomics work. And something similar we did recently in the nasopharyngeal swab sample of the COVID patients as well. Just to summarize the first part of this presentation from the previous lecture and continuing now, is that the sample prep is of course most crucial step for doing proteomics. It's not only just you have to know how to process your protein extraction method, but also you have to think about which technology they're going to take forward for which type of question to be addressed. Accordingly, you are going to enrich your sample and you are going to fractionate your sample. You are going to remove the contaminants from the sample, keeping that objective in mind. And as I said, it's a good idea to think about all the biochemistry what you have studied. Be critical, be intellectual, be creative, and think about, read literature, but also be open to new methods which you can try yourself. Sometime your 6 months of investment of just trying out new methods can give you, it will be a frustrating journey to explore many things for 6 months. But after that, once you have reached to the deep proteome coverage with good reproducibility, then your projects will start flying very fast. And in 3 months, you can generate, publication quality, high quality data sets very easily. So that's something which I like to emphasize from the, the sample preparation part. While in the entire proteomics, there are 3 major components. One is the sample preparation part. Second is the instrumentation part. And third is the data analysis part. Most of the time, most of the people feel that the middle part, the instrumentation part is most crucial. And you always feel that, you may not have access to the best technologies and best instruments. So you should not do proteomics. But I think that's where there is a gap here in understanding. We should be thinking about only the first part and the third part more importantly or most importantly, because the sample preparation, if you can make a good sample, you can send the sample to different places and everybody will generate good data quality. And third part is data analysis, which you should be pick up from this course, how to analyze the data yourself. And if you can analyze the data yourself, then I think you can do justice to your sample because only you know your sample, you know your interference, you know your contamination, you know what is the artifact, what is the real signal, what is the correct database to be selected. All that cannot come from anybody, any facility or any other collaborator from that matter. You have to do that yourself. So first part and third part, sample prep, making good sample, and third part is data analysis, are the most crucial in the entire proteomics workflow, in my own personal opinion. Middle part looks very gadget oriented and cost oriented, but is actually the simplest part, because if instrument is good, facility is good, technicians are good, they will generate good quality data irrespective of different projects. But the sample intake has to be really crucial for that matter. Just to continuing on this, on the sample prep part, before we go to the more interactive sessions. Now you have identified that this extraction methods work very well for your sample. You want to know how much protein you got from each of the method type and what is the protein quantification yield from those methods, and then how the quality of these samples look like visually and also quantification wise. So Bradford assay is one of the commonly used methods. So again, given that all of you are at quite senior level, I don't want to make it again a basic course of my classes. So just to refresh you briefly here, but I'd like to show you more hands-on. So you are using different type of assays for doing quantification. Please quantification method, which is the best method. You can choose the quantification method the way you think is working well for your lab. Could be Lorry method, could be any other kit which you are using, BCA method. Anything is actually good, and none of them are actually perfect, right? They're all subjective. They give you some landmarking and some idea that okay, your protein sample is 100 microgram is around this, or this sample is 150 microgram. So they are giving you some relative idea, but they cannot be absolute that it is exactly 100 microgram, right? So that's where I think you can choose to select your own method for quantification. We are just giving you one for illustration and discussion purpose. So Kumasi Brilliant Blue based method, Bradford assays and modified Bradford assays are quite popular as well. And you are measuring the binding absorption max shift from the 465 nanometer to 595 nanometer when the protein binds. The red color dye change to the blue color form because of the hydrophobic interaction with the positively charged amino acids, and that's what you are measuring at the 595 nanometer. For doing that, we make the standards with the known protein like BSA, and again some of these things could please don't feel that we are trying to teach you something very new. You are already doing it. We are just giving you some recap and also giving you an idea that how you should be careful about some of these steps as you go along. So you should make some standard graph the way you have been doing for other protein assays, and choose any standard protein and make the known amount of those protein, their stocks, and measure their absorbance, and do the plotting, make the standard curve for the known amount of, let's say, BSA in this case, and the absorbance value measured at 595 nanometer. Calculate the R square value, and the graph should be as linear as possible with the good R square value, in this case almost 0.99, which is good. What we are showing you here some actual data for some samples. So take at least 2 absorbance for every sample, because you will find sometimes some variability. But if at least you should take the average of those. If you find one is very different than other, you should repeat one more time or 2 more times, because your protein quantification is very important to start with. And then depending on how much volume you have taken for quantification, you can make it per microliter, and then choose accordingly that how many microliter you need for different application for 50 microgram, 30 microgram, 5 microgram, likewise. Let me show you this clip of demonstration for the Bradford assay from the lab.
In order to know the protein concentration in a given sample, or to get the total yield of protein in a given sample, it should be quantified. Depending upon the type of lysis buffer used, various quantification method can be used. Today, we will demonstrate you one such method, which is Bradford protein assay. Bradford protein is one of the common methods used for protein quantification, and it is a colorimetric assay. Before performing the assay, firstly we should have a standard protein. The standard protein here we are using is bovine serum albumin, that is BSA. So here we have prepared serial dilutions of BSA, that is standard 1 to standard 5. And also one is blank, which will involve only the reagent. And here we have one unknown sample. Now to each of the tube, equal volume of Bradford reagent is added. After addition of reagent, now the tubes should be kept in dark for 5 to 10 minutes. Bradford reagent exists in two different forms. One, when it is unbound, it is in brown color, and then when it binds to protein, it becomes blue color. The intensity of blue color is directly proportional to the amount of protein present in the given sample. It is basically based on the absorbance shift, which from unbound form to bound form changes from 465 nanometer to 595 nanometer. So absorbance should be taken at 595 nanometer. Now after incubation, each sample is added to the plate. Preferably the sample should be added in duplicates. Now all the samples are added in the plate and then it is ready for taking the readings. For this we are using multi-scan geo. Plate reader. The first step is to set the plate layout, which includes blanks and all the standards. Also the unknown sample. The next is protocol, in which absorbance at 595 is set. Measurement mode is selected to be precision. And also blank subtraction is set. After getting the absorbance values, the values can be exported. So here we have the absorbance values for all the standards, including standard 1 to 5, as well as the unknown sample 1. So here we have absorbance 1, that is OD1 at 595 and OD2. So firstly, we will calculate the average absorbance. For all the sample, average absorbance is calculated. Similarly, for the unknown sample, average absorbance is calculated. The next step is to plot the standard curve for BSA. For this, sample amount and average OD to be taken. So here we have the plot for standard. Also, the R square and equation is displayed, and we can see that we have got R square of 0.7. Using the equation obtained, amount of the sample in amount of the protein in the unknown sample can be calculated. So for the unknown sample, we have to calculate X, and the absorbance obtained for the unknown sample would be as Y, as shown in the given formula. Since we have taken 2 microliter of unknown sample, so concentration would be half of it. Now we have got the concentration of our unknown sample. This is how we can get the protein quantified. Its concentration as well as yield can be calculated. Other than the Bradford method, there are several other methods such as BCA and 2D quant. So after getting the protein quantification, next procedure can be performed, such as SDS page or any other proteomic procedure.
All right, so once we now know that we got good yield of protein and we have sufficient amount of protein from the given sample, then we would like to check the quality, that how good the quality of the protein is on the SDS page. And many times you might feel that from the quantification you have very high values and you think you got very good yield, but actually in reality you may not have so much good yield. And we will talk more about the SDS page and gels tomorrow, but I'll just show this now lab demo about how to check the quality of your samples on SDS page gel.
Quality of the protein in a given sample can be checked using the gel. For this, one-dimensional SDS page can be used. Here we will demonstrate you how to cast SDS page and how to perform the gel electrophoresis. For casting the gel, we need the casting apparatus and glass plates. The glass plates, one is thick plate, another is thin plate, and suitable sized comb is also required. Glass plates are fitted into the casting apparatus so that the space created between the thin plate and the thick plate can be poured with the gel solutions. For performing the discontinuous type of STS stage, two different gel solutions are required, which is one is resolving gel solution and another is stacking gel solution. For these two solutions, composition is different, such as this could be the composition of the resolving gel and stacking gel. So the first step is to add the resolving gel solution inside the space between the plates and let it be solidified. After some time, now we can see that resolving gel is casted. Next, we have to add the stacking gel solution. Now the stacking gel is added. Because we need to create wells inside it, now we'll add the comb into the solution. Let it for solidification for some time. Now we can see the gel is cast. It is ready for performing electrophoresis. Now the casted gel is placed inside the electrophoretic tank along with STS running buffer. Comb is removed to expose the wells so that the sample can be loaded. Suitable amount of protein is loaded for checking the quality. The sample can be loaded in duplicates along with protein ladder so that the molecular weight can be estimated for the proteins. Now we can see that the sample is loaded and it is ready to perform the electrophoresis. After setting the suitable voltage, start running. Here we can see that electrophoresis has started. It has to be performed for around 1 to 2 hours. So the gel electrophoresis may take around 2 hours, and after that, in order to visualize the proteins, it should be stained and de-stained to remove the extra stain. As the sample run would take some time to complete the electrophoresis, we are showing one of the demo gel to show how the samples look after electrophoresis. As we can see here, that clear bands in each sample are seen, which shows that the quality of the sample is good to take forward. So this is how to check the quality of the sample and to decide whether to take it forward or not. After getting the quality, it can be either gel-based or in a gel-free manner.
All right. So I think this kind of image speaks a lot about samples, right? As this page, when you see something like this, well, it shows that all the proteins of low and high molecular weight, they're very well resolved in the gel. So the extraction method has worked out well. But if all these samples we are loading, let's say 10 microgram of protein in all the wells, then I think we have somewhere wrongly quantified our protein, misquantified. So again, the gels can give you a lot of information after looking at the images, and you can make a lot of sense from that to see like, is that really good for taking forward, or you have to redo extraction method or redo the quantification. As I said, I can make a lot of observation here that some of these extraction methods have worked very well, but this quantification is not good. This quantification is not good. There is no protein here. So either the quantification is poor or this method has not worked. So accordingly, we can either re-quantify or try out newer methods as well. So quality control checks are very crucial to make sense whether we are going ahead or we should take a step back and just try out new things. I think in the sample preparation part, if you are having a lot of patience and you can keep taking the backward step couple of times, you will do good justice to your projects and to yourself for doing good proteomics work, because more and more time and effort you put in for this step, you'll be better prepared with your samples for the downstream further analysis part. So this is one of the way I think you should be looking at your gels. But also I would say that, many times you're not sure whether your protein may contain some nucleic acid contamination also. So same protein solution could also be done on the agarose gel, and that is not something which you will do. Otherwise, you will only run the nucleic acid on the agarose gel electrophoresis, But if you run this sample also, protein sample on agarose gel, and you see there are some smear or there are some contamination coming from the nucleic acid trace, then you know that, okay, this could be the what value you see in the quantification that may not be truly just because of the protein. Sometimes even the nucleic acid can also be binding to these dyes, and they are giving you some artifactual values, which apparently looks very high to you, and you feel you got too much protein on it, but you may not have sufficient protein from the given sample. So doing a QC check of the nucleic acid contamination is of course important. You can always say that, okay, I have used some nucleases and that has been removed. Then that is fine. Then you are sure about it. So these are the things which looks very simple, which every lab does. But I think when you are thinking about high throughput experiments and high gadget instruments, before proceeding those, we have to do a lot of work into this part to make our samples really good to take to the next level of analysis for mass spec or other type of proteomics workflow. So once you think that you have got good protein yield, like you have good concentration of the protein, sufficient amount of protein, you got 200, 300 microgram protein you have to at least to work with. So now you want to go for the next step and you want to find out what should be my next step for the proteomics. So for doing that, can we think about what is our goal? Are we going to do the gel based or are we going to do gel free proteomics? So again, for each one of those methods, there is a different way to move forward. If we are thinking about just the simple protein identification from the SDS page, then that should be done with the in-gel digestion. But if our goal was to look at identifying a large number of protein and their quantification, then that should be with the in-solution digestion. Again, we'll talk about more about going for in-gel or in-solution digestion when we go to the next steps for the mass spectrometers. But right now, I want to just make you prepared to think about the sample prep. But when I said, it is just not only about protein extraction. It is starting from knowing your sample type, interferences in that, quantifying the protein, quality control checks, and also then proceeding for the in-gel or in-solution digestion. So let me now show you one lab demo for in-solution digestion.
For the mass spectrometric analysis, in order to identify the proteins in a sample, protein digestion can be performed in a gel-free or a gel-based manner. We'll show you how to perform the in-solution digestion, that is gel-free procedure. So now, after quantification, suitable volume of the sample is taken for digestion. Before doing the actual trypsin digestion, we need to process through the two steps, that is reduction and alkylation of proteins. For reduction, here we are using TCEP, that is tris carboxyethyl phosphate. Appropriate volume of TCEP is added. The sample is properly vortexed and spun. After addition of TCEP, that is the reducing agent, it should be incubated at 37 degrees for 1 hour. So after getting incubated at 37 degrees, the samples would be reduced. So after incubation at 37 degrees, reduction is happening. That means all the disulfide bonds are broken within the proteins. So now it's time to alkylate the sample. For alkylation, iodoacetamide is used. During the alkylation procedure, alkylation will block the thiol groups so that the disulfide should not be reformed. After this, the sample is properly vortexed and spun. To perform the efficient alkylation, the sample should be kept in dark for 10 to 15 minutes. So after completing the steps of reduction and alkylation, the next step is addition of trypsin for the dilution. So before that, the step is dilution, which means we first need to dilute the molarity of urea that was present in the initial sample. So the diluent is used for diluting the sample. Appropriate volume of diluent is added to the sample in order to dilute it. Before addition of trypsin, make sure that the pH is 8 so that it can work properly. Now the next step is the addition of trypsin that could be added in the ratio of 1:30 to 1:50, which means that one part of the trypsin can be sufficient for 30 parts of protein. The sample should be vortex properly and spun. And finally, for optimum protein digestion, the samples need to be incubated at 37 degree for around 16 to 17 hours. After 16 to 17 hours of incubation, the protein sample is completely digested, and then the sample is dried in order to get the peptide sample. These dried peptides can be taken further for mass spectrometry. For mass spectrometry, the peptide sample should be properly cleaned. To make sure that the sample is clean of various salts or other contaminants, the sample can be performed through the cleanup steps. For these steps, we use C18 columns. So these columns can be made using the C18 material within the tips. And then the sample is passed several times through the column. This column works on the principle of reverse phase chromatography. During several steps of passing the sample through the column, the peptides would be bound to the column, and then finally the peptide would be eluted. After the peptides are bound to the C18 column, they can be further eluted using different gradients of acetonitrile. And then the elutes is again vacuum dried, and the sample can be taken for mass spectrometry.
After this,
Yes.
Then I will take 10 to 15 minutes to later will have that.
All means infinite knowledge, or essence of breath, life, everything that exists. Always refers to the science dealing with the use of technology to explore the roles of various biomolecules that make up the cells of an organism.
The Human Genome Project and the first draft of the Human Proteome.
What happened?
Are two crowning achievements by the global omics community thus far. With an aim to understand human biology at the cellular level, with concerted efforts of the best scientists from around the globe, the Human Proteome Organization proudly announces its most ambitious project yet, the Human Proteome Project.
The goals of the Human Proteome Project are two. First is build a parts list. Describe at least one product of each of the approximately 40,000 protein coding genes. And to identify and characterize the functions of the post-translational modifications, splice isoforms, and sequence variants of those individual proteins. The second is to embed proteomics, integrate proteomics, the genomics, epigenomics, algorithms, the integrated omics approach to really understand biological networks, pathways, drive normal biological processes and pathogenic processes in various diseases.
The Human Proteome Project's bold vision is to understand what it is to be human in strictly molecular terms.
We have 50 teams, 25 focused on individual chromosomes and their biology, and another 26 focused on particular diseases, biological processes, and what we call resource pillars of antibody profiling, bioinformatics, and mass spectrometry.
Chromosome-based Human Proteome Project, CHPP.
A primary goal of chromosome-centric Human Proteome Project, CHPP, is to identify and characterize proteins like mass spectrometers or antibody detection of the missing protein. To this end, we use various omics technologies, including RNA sequencing, siRNA, and upfront bioinformatics tools and resources.
The chromosome teams have been very active, and in fact, the Indian chromosome 12 team has performed interesting work on glioblastoma.
Yes, the chromosome 12 referred to HPP, along with the 4 other Asian countries.
The chromosome 12 coded genes and proteins are implicated in brain tumors and neurological disorders.
So we're very excited to be part of the Chromosome 10 project. We decided that the best way to accomplish the goals of this project is by doing Chromosome 15, Chromosome 16, Chromosome 19.
The teams are not only focusing on identifying proteins which play a role in the cell, but also on developing new technologies.
Collaboration is key to success in our endeavor.
Biology, disease-driven, Human Proteome Project, HPP.
Biology Disease Initiative as part of the Human Proteome Project started a few years ago with really the goal to be able to facilitate the use of proteomics into the broader community of biology.
Last time, Proteome Project. Proteome Project. Liver. Kidney and neuron.
HPP aims at expanding our understanding of the human proteome with a focus on changes accompanying the different stages of the diseases.
The Human Brain Proteome Project aims at identifying molecular mechanisms and biomarkers involved in neurodegeneration and other brain-related disorders.
The Human Liver Proteome Project aims to define a comprehensive and dynamic human liver proteome and identify novel proteins relevant in liver biology and pathology. The cardiovascular initiative aims at elucidating disease mechanisms and identifying candidate therapeutic targets to provide clinically useful diagnosis and risk prediction.
The Kidney and Urine Proteome Project aims at understanding kidney functions and pathophysiology of kidney-related disorders. Human Infectious Disease Project aims to understand host-pathogen interactions of several important human infectious diseases.
We have invested together highly placed vaccine or any started in infectious disease. Currently,
Resource Pillars, Human Proteome Project.
In addition to BDHPP and CHPP teams, we have 3 resource pillars: affinity-based protein capture, mass spectrometry, and knowledge-based bioinformatics.
This project began with the Protein Standard Initiative, PSI. In fact, Proteome Exchange makes it possible to upload the primary data set with raw spectra from mass spectrometry, place the data in dry or SRM for targeted proteomics studies.
We have several goals that knowledge-based pillar. The major components of the knowledge-based pillar are XPROT, which is the primary knowledge-based protein proteome project.
So the peptide atlas that we produced that links to systems biology provides the groundwork for a lot of identification by mass spectrometry.
Human Proteome Project has developed common metrics and guidelines that allow open and rapid exchange of high-quality data. It is now building strategies to discover those proteins where only lower stringency is necessary.
A crucial component also of the knowledge-based pillar are a set of guidelines which we have developed. Coordination with Proteomics Standards Initiative.
Finally, I want to mention there are sets of proteins particularly informative for the biology of one or another disease, diabetes, or aortic aneurysms, or other conditions where our colleagues throughout the life sciences research community could make very effective use. Sets of proteins that are readily analyzable, quantifiable by proteomics.
We're in a position where we can identify the missing proteins and take the regions of the genome and show that they encode the proteins, as well as define the complexity of the transcript of them and show which ones also encode proteins.
HPP has a lot of momentum right now. I want to see us have huge impact on all the other communities. The genomics community, the metabolomics community, the microbiome community. Together we will be able to combine all this information and have huge impact on biology and medicine. We are proud of the progress of the Human Proteome Project, and we are proud of the engagements throughout the community that the HUPO has stimulated.
Ladies and gentlemen, welcome to the workshop. Our next session is about to begin. May I request you to kindly mute your microphone and wait as we prepare for the session. Thank you. Ladies and gentlemen, welcome to the workshop. Our next session is about to begin. May I request you to kindly mute your microphones and wait as we prepare for the session. Thank you.
All right, welcome back everyone. So we have finished the toughest part, which was the kind of movement and doing some bit of the sample prep. Just to recap, I think what we have learned so far, essentially some concept of proteomics. And we then try to show you the lysate, which you can take forward. Of course, each of you will have your own projects of interest, right? You'd like to learn different things. Just 1 second, I think we should be doing the video as well. Yeah, so you will have different samples for your interest, which you would like to pursue. Therefore, we can't tell one best sample strategy, preparation strategy. Nevertheless, like, the lysis with the urea, that's very robust, can be good starting point. Many times we then try trizol, we try TC acetone, different methods can be tried, right? But that depends on your working of the plant, yeast, bacteria, human, whatever sample type. Even within human, you have sometimes tissue, blood, CSF, variety of sample. Every sample comes with its own challenge and uniqueness which we have to work with. What we thought, at least we give you flavor of something as basic as talking about protein quantification and the qualitative analysis of the samples. So the Bradford assay, you have done it. Our team has taken the plates. They will do the reading with the multiplex reader. Additionally, the SDS page, you have run some samples for the quality check, which overnight it will run, like run few hours, then overnight is staining, and tomorrow you will see the results. So few things will be done parallel, right? You have been given at least some demonstration of the mass spectrometer. While it is highly curious to look at the mass spectrometers and how they operate, but truly the black box, that's what you see over there, right? Nothing you have idea how it operates or whatever you do, except that you put your sample there and then that sample has to be analyzed. So it's good to go back to the basics and read about how the different operating principles work for different instrument type. Nevertheless, as a biologist or as an experimental person, all you will do, like this sample which we are preparing, you might have been shown over the auto sampler, where you're going to place your peptides. And that's all. Rest whole operation we will do from sitting here. We will take control of the system and software. We will show you how we are running the sample live, and that data will be acquired and you will be analyzing your data. So actually we are going to do everything in the real live way, that you will be given the live data for your analysis. But this much part is only what you do in your wet lab on the bench, right? Rest everything which you can do from anywhere. There has been a lot of time cases, while I'm in US sometime and we are looking at our sample, or they are traveling, my students are traveling somewhere, they're still analyzing the data. So these things are very common. So you can then try to place more emphasis on the next part, which is most important, like how you can now acquire the data and analyze the data well, right? So the demonstration was brief, which I understand, but I hope you got flavor of it. You're still welcome to come back to the lab in the evening post 6 p.m. session if you want to learn some more or want to see something more or discuss something more. We don't want to hamper your enthusiasm, but given the time constraint, we try to keep everybody on track. So thanks for cooperating and walking in the rain and coming back on time. So what we are going to do now, you have done the quantification, but how you're going to calculate it. Again, it looks very simple, but it's a good idea for you to do it yourself. We will be giving you values in some time, but right now, Sachin is going to show you demonstration how we can take the standard values and some unknown values and plot. So you can go through the step, and when we will provide you the data, you can then plot your own sample, and then tomorrow let us know what was your concentration, right? So that is the idea. So this is something which Sachin will now take over. Immediately after that, then we are going to start the second type of sample preparation. What we have done so far was the global proteome. It means that we want to break open the cell, every protein which is present there, and you have been given in the sample 2 different type of cell lines. So those cell line samples were what you will be analyzing the mass spec. But now next session will be about cell surface proteome, how you can enrich cell surface proteins, and as Dr. Kamal shown in his presentation, looking at more druggable target and which can have more relevance for the immunotherapy. Those kind of things you can pick up from these kind of cell surface proteome. So that next session, some presentation and the some demonstration will be done for that activity. Once we have done both of these, it means the global and cell surface proteome. Now you got at the end, you have prepared the proteins, right? The protein is global from all the proteins from the cell, all these only enriched protein from the surface. In either way, you have to digest the protein now. So once their session is done, we will show you the in solution digestion. Again, that in solution digestion will take you a good 1 day time to do it. We will do it in a fast track way. Like, the incubation step, we will fine tune and things like that. But we'll still keep some sample overnight for you to run for tomorrow. So this is a plan. So many things will be happening parallel. It means your gel is running, your quantification data will be given to you. You will be doing some in solution which will be done overnight. And tomorrow morning we'll show you some mass spec live run and the data analysis for that. That's the plan. So Sachiran, now you can come and talk about plotting the things, maybe just 5 minutes, give a flavor of it. But we would like everybody to do these things at your home if possible, if not tomorrow morning. Also tomorrow when we finish the workshop, we will conduct some quiz. We have already elected all of you. There will be some quiz, so it is a good idea to be prepared to follow each step properly with us. Yeah.
Yeah, so welcome back everyone. We just, so I'll quickly show you how to quantify, how to plot the standard curve. Yeah, so here on the screen, you see, I have values for blank to 6 standards. We usually don't take BSA about 12 micrograms because we've seen that Bradford doesn't follow a linear response beyond that. So what I do is I just copy these values and plot the graph again for you to be able to do it yourself. So I have with me 2 unknown values, and these are the ODs that I got after quantification. Right. So you see the OD for the blank, that is just Bradford alone, was around 0.32. And subsequently, you see standard 1 to 6. These are in increasing concentration or amounts of BSA, where it started with 2 micrograms and we ended at 12 micrograms. And the values, they do follow a linear response. Net OD is your observed OD minus the OD of the blank. Okay, so we take the net OD values and we'll plot the graph here. So you now add the trend line. And here set the intercept to 0, display the equation on chart and display the R squared value on chart. The R squared value usually gives you an idea of how linear the response is and how consistent your result is. And this seems to be a decently good graph. This is how the values in the real world look like. Unless you're an expert, you might not see anywhere, but I mean, you won't see 0.999. There are people who do manipulate the values as well, so I would strongly recommend against any such manipulation. If at all you feel that the standard graph, the curve is not that great, I would recommend that you re-quantify rather than trying to use such a graph, which would give you erroneous results down the line. So you now have the equation here, which is y is equal to mx plus c, right? So since the intercept was set to 0, you don't see c here. So y is equal to mx here. And with this value, so what we see here is the value of the y for the unknown samples. So
this would be divided by the slope is 0.0216, which is around 2.7. You apply the same to the next sample, it's 6.4. I don't usually report beyond 2 decimals. And while preparing samples for SDS page and even for digestion, I bring it down to 1 digit. So from this particular exercise, we were able to quantify accurately the concentrations of 2 samples, unknown samples based on the OD. And even without actually doing the calculation, you can still get an idea as to where your concentration is likely to be. Like for an net OD of 0.06, it's implied that it would be somewhere between these 2 values, right? So you would get a rough idea of where your samples are. So what we would do is we'll share a set of standard values for you and also your unknown values. And that is something and we expect you to plot this curve. Okay. And depending on the quantification approach that you have used, the assay you have used, the plot that you make may be different. Like for someone who uses a micro VCA, you are expected to plot an exponential curve. Okay, so the equation would be much more complex there. So it would require a little bit of math or maybe ChatGPT for that. So this brings us to the end of the quantification session.
So next we'll have the demo session for sample preparation for cell surface proteomics. For that we have Dr. Kamal Mandal and Mr. Rakul Nair. So Dr. Kamal Mandal you have already heard. Let me introduce Mr. Rakul Nair. So he has completed his BS in molecular and cellular biology at John Hopkins University in 2021. And current till June 2023, he worked as a junior research specialist in Dr. Arun Lab at CSF. And he's worked on several projects directed at the development of novel cancer immunotherapies for rheumatologic malignancies, which involve using mass spectrometry proteomics to profile the cell surface antigens of cancer cells to uncover potential therapeutic targets. So he will be starting his MD PhD training now at the Gallows College of Physicians and Surgeons, where he will continue working on designing the next generation of cancer immunotherapies and bringing these advances to the patients who need them. So along with Dr. Mandal and Mr. Naik, we also have our TAs, Ayushi and Amrita, to assist them. So over to you.
So which is the enter screen for you?
The output.
You will be staying in the PPT only. You will not swipe, right?
No, I will be. So.
There's some. Can you go to the Microsoft that PowerPoint? I think. Yes, fine. We have the this image. This one is the lesson.
Okay, hello everyone. Thank you, Sanjeeva, for having me. I'm going to be a graduate student, like many of you, so I'm very humbled to have been invited to speak about my experience at the WIDA lab. Today I'm going to be giving you a brief introduction, presentation about cell surface proteomics, so proteomics directed at identifying all the proteins on the surface of the cell. And then Kamal and I will be doing a demonstration of how that would look different from your normal proteomics preparation protocol. So just as an introduction, I think Sanjeeva and Kamal and Dr. Greb all have done a great job talking about proteomics. Just as an overview, this is the normal flow through. You start with cells, and then you lyse them to extract the proteins, and then you digest them into peptides, run them through PHPLC, and run them through your mass spectrometer. And this has served as a really powerful tool for identifying all the different proteins present in a particular sample. But it is limited that it only can read the highest abundance proteins. So in a particular mass background, you might get 5,000, 6,000, up to even 10,000 proteins. But that's not all the proteins in a cell. There's way more proteins in a cell. So basically, if you want to see the particular proteins present in a certain part of the cell, let's say the mitochondria, the nucleus, the cell membrane, then global proteomics or proteomics of the whole cell is not going to give you the information you need because just there's going to be too many other proteins present in the sample, and your proteins aren't going to be caught by the mass spectrometer. Therefore, so when you want to do proteomics of a specific part of the cell or a specific organelle, then you need to do enrichment before you run it through the mass spectrometer. Okay. So today I'm going to be talking about the cell surface proteome or enrichment of the cell surface proteins. So a little bit why we are in cell surface proteomics. As you all may know, cell surface proteins are very important to all the interactions with the cell. They're kind of the gateway between the cell itself and the outside world. So anything that goes in and out of the cell, how it interacts with other cells and the environment as a whole. And it's very dynamic. It's constantly changing in disease states and other conditions. And clinically important for us is that over 65% of drugs currently target cell surface proteins. So in terms of medicine, cell surface proteins are much greater represented than anywhere else in the cell as a place of interest. So profiling all these cell surface proteins of whatever cell type it can be very important for any clinical aspect. So today we're going to discuss a particular type of cell surface enrichment that we call cell surface capture. Oh, sorry, I skipped a slide there. So when we talk about methods for enriching the cell surface proteins in a cell, you have several options that you can try. One of the oldest methods that's been around for decades is selective ultracentrifugation. So essentially, you centrifuge the cell lysate at different speeds, and depending on the molecular weight of the molecules in a particular portion of the cell, they'll pellet or be in the supernating at different speeds, and you can experimentally determine which speeds you need for which organelles and molecules to pellet, and then you can take those different fractions for a mass spec analysis afterwards. A more recent development has been using NHS biotin to react with cell surface protein lysines. So you have a biotin with an NHS handle, and that can react with the lysines on the cell surface to chemically link a biotin to cell surface proteins that you can then pull out of the lysis with a streptavidin pull down with the streptavidin biotin interaction, and then wash away all your other proteins. And a recent development out of UCSF that's very exciting is the enzyme-mediated biotinylation of cell surface proteins. This is advantageous because it's a much faster process instead of doing a long chemical reaction. This is very quick. It takes a few minutes just to use an enzyme to link a biotin to cell surface proteins. And I can talk later about why some of these methods are more or less advantageous than the method we chose today to demonstrate. So the method we're going to be demonstrating today is something that is called cell surface capture. It was originally developed by Dr. Bernd Wohlsteed at the Institute for Systems Biology in Seattle, which is a big proteomics and mass spectrometry institute. So essentially, this is a method to enrich cell surface proteins by covalently linking a biotin to N-linked glycosylated proteins. So essentially, N-linked glycosylation means that these proteins have a glucose molecule covalently attached to them in a particular way, and then we can go in and use chemistry to attach a biotin to that sugar. And this is a good, you might be thinking, why should we target glycosylated proteins for cell surface proteins? This is because over 85% of cell surface proteins are glycosylated, and therefore this method can capture the vast majority of cell surface proteins. And it's a very robust method. So the overall principles of how this enrichment can work for cell surface proteomics is you start with your whole cells in culture. These can be cell culture, these can be primary samples that you got from a patient. And then you need the intact whole cells in a suspension. And then you chemically link biotin to these N-linked glycosylated cell surface proteins. These little black lines are biotin handles. And then after you lyse the cell, and then all these proteins, both intracellular proteins and cell surface proteins with a biotin linked handle are going to be in that lysate. You can use these black streptavidin beads to bind the biotinylated proteins. And then wash those beads with buffer, and all the non-biotinylated intracellular proteins are going to be washed through, and you're going to be left with just the cell surface proteins bound to the beads. And then you can do an on-bead digestion, where Sai-Chairman talked about in solution digestion or on-gel digestion. This would be an on-bead digestion, where you add the trypsin or your digestion mixture directly to the beads. It's going to chew up the proteins on the beads, and then you can remove that solution from the bead. So you're going to be left with just your digested peptides in solution, which you can then quantify and carry through the rest of the mass spectrometry protocol. So a little insight onto the chemistry of how this biotinylation reaction works. This is your protein with the sugar attached to it. That's the glucose molecule. So first you're going to use this sodium metapriodate molecule. That's going to act as an oxidizing agent. That's going to oxidize those two alcohols on the sugar to become carbonyl groups. And then you can use your azide to come in and couple the biotin to those oxidized sugars. And this is the biotin azide molecule we use. It's called biocide hydrazide. So that's going to be this molecule here that's going to come in and covalently bind to the sugar attached to your protein. And then this is the more detailed sample preparation workflow for our cell surface capture. So we begin with our cells in culture, and we wash them. We wash off all the media to make sure there's no extracellular proteins that are going to get labeled, and we pellet them. And then you mix in your oxidizing agent and your biocide hydrazide to get the biotin labeled proteins. And then you lyse them, and you're going to be left with these biotinylated as well as intracellular proteins in your lysate. And then you're going to add your biotin beads, which are in dark blue over here, to your column. And then run your, you're going to incubate your lysate with the streptavidin beads, which will bind all the biotinylated proteins. And after that's incubated for 2 hours, you're going to wash all the extracellular non-bound proteins off by washing through the column several times. And then you're going to add your trypsin mixture after you alkylate and reduce, as we talked about, the proteins. You're going to add your trypsin mixture to digest those proteins into peptides, which you can then desalt to remove all the salts that will clog up the mass spectrometer and carry it forward for mass spectrometry. So Sanjeeva and I published a paper discussing this protocol in Jove, which is accompanied by a video that kind of goes through the whole process, and you can see how the protocol is done. So I wanted to share that with you before we do a live demonstration of it here. That being shared.
So Teams. Yeah. Has it stopped?
Yes, I think it has stopped.
Very good thing. Can you open the link again, if possible? Yeah.
Can you just share the desktop screen? Is this your needs? No?
I don't think maybe if we close.
Yeah.
Hey, where is the entire screen for you?
What one, right?
No. He will show the video. There will be an entire screen. Or where is the the web where you are showing? Yes. Okay. So now can you open the Teams?
Yeah, share screen.
Can you just? Yeah. Now it's coming.
It's on the share screen. It's just not showing up here on the share screen.
Is a duplicate.
It's shared on the Teams. It's just not showing up. On
Right. So there's a duplicate system is there now. Windows P for special. No, it is showing over there. It's just not coming here. Yeah. This one?
Okay.
Can you check now? Yeah, I think it will work. Is it please? Yeah.
Oh, it's a separate. That's okay.
Go back. Yeah. Yes.
Audio. There.
No, just. Can you give the one? Can you just see? From our laboratory. Can you switch on this one? Can you on?
Yeah.
To begin, remove the frozen biotin labeled cell pellet from -80 degrees Celsius and thaw it on ice. Add 500 microliters of 2x lysis buffer to the pellet, mix thoroughly, and vortex vigorously for 30 seconds. Sonicate the cell lysate on ice and centrifuge the lysate at 17,200 G for...
Is coming with your system. So can we just start the hands-on because it's some issue with his system. It's coming. I think.
Yeah, we can start.
No, session को नहीं setting पर ना control हो जाएगा. Yes, just take this. Yes. First.
Sometimes I shared at your WhatsApp. WhatsApp is
Getting shared. Oh.
I think you can just you can just
with the Teams, open the Teams. Yes.
Do you have a standalone version of this video?
Like
Downloaded version?
No. Just Teams is somewhere.
I'm sure this video is very important.
Yeah.
I'm sure.
Teams. Teams is actually getting shared. Yeah. Background. I'm not very much sure over the Mac system. Mute.
Is it muted? Is it okay?
Microliters of 2x lysis buffer to the pellet, mix thoroughly, and vortex vigorously for 30 seconds. Sonicate the cell lysate on ice, and centrifuge the lysate at 17,200 G for 10 minutes at 4 degrees Celsius to pellet any remaining debris. While the lysate is centrifuging, add 100 microliters of Nutravidin agarose resin beads to a filtration column attached to the vacuum manifold. Apply a gentle vacuum and wash the beads by flowing 3 milliliters of wash buffer 1 over them. Remove the filtration column from the vacuum manifold, cap the bottom, and add the clarified cell lysate to the column with the beads. Cap the top of the column and incubate the lysate with the beads on an end-to-end rotor for 120 minutes at 4 degrees Celsius. After completing the lysate incubation, place the filtration column back onto the vacuum manifold and apply a gentle vacuum. Wash the beads by flowing 5 milliliters of wash buffer 1 over them. Repeat the washing step with 5 milliliters of wash buffer 2 and wash buffer 3. Once the final wash buffer has flowed entirely through the beads, remove the column from the manifold. Then add 100 microliters of lyse solution to the beads and transfer them to a 1.7 milliliter low protein binding microcentrifuge tube with a pipette. Briefly spin the tube to settle the beads and remove 50 microliters of the solution. Next, place the tube on a heat block shaker at 65 degrees Celsius for 10 minutes at 1000 RPM shaking. Resuspend the dried trypsin in the digest tube from the kit by adding 210 microliters of the resuspend solution and pipetting it up and down several times to ensure all the dried trypsin is resuspended. At the end of feed incubation, remove it from the heat block shaker and add 50 microliters of the resuspended trypsin solution to the beads. Incubate the tube at 37 degrees Celsius, shaking at 500 RPM for at least 90 minutes to digest the protein. Once the digestion is complete, transfer the solution containing the beads to a spin column and insert the column into a 1.7 milliliter low protein binding microcentrifuge tube. Spin the tube to separate the digested peptide solution from the beads. Next, add 100 microliters of the stop solution to stop the digestion reaction and acidify the peptide solution. Using a tube adapter, place a desalting column in a 1.7 milliliter low protein binding microcentrifuge tube. Add the entire acidified peptide solution to the column. Spin the column at 3,800 G for 3 minutes so that the solution flows through the column. Discard the flow through as the peptides are now bound to the column. Add 200 microliters of wash 1 solution to the column. Spin at 3,800 G for 3 minutes at room temperature, and then discard the flow through. Repeat the washing step with 200 microliters of wash 2 solution. Transfer the column to a new labeled 1.7 milliliter low protein binding microcentrifuge tube. Add 100 microliters of elute solution to the column and spin at 3,800 G for 3 minutes at room temperature. The peptides are now eluted off the column into the tube. Place the peptide solution in a vacuum centrifuge and allow it to dry overnight. Place the dried down peptides at minus 80 degrees Celsius until ready to quantify and analyze on the mass spectrometer. Proteome analysis was performed to characterize the enriched cell surface proteins using lipid chromatography tandem.
But only slice of money.
you have to be sure that what you're injecting in the mass spec are the stuffs that you want to see. Something that you don't want to see, you don't want in your sample. And that's where sample preparation, importance of sample preparation comes into the picture. So enrichment is a very crucial component of proteomics. So let's say if you want to see that if you are enriching the proteins which are in the nucleus, you want to enrich for the nucleic proteins. You want to see something like mitochondria, you have to enrich for the mitochondrial protein. Here, we are interested in cell surface proteins. The cell surface proteins are very attractive for pharmaceutical purpose. Like you want to have a pharmaceutical intervention for addressing a disease, you want to know what cell proteins are all there in the cell surface, on the surface of the cell. So if you want to am I audible?
Just you can look at the audience and then speak. So that's
the audience is here?
Only online audience is there as well. Yes.
Yes, I have to stand here. That's difficult. Having tilted so that I can
you can move on. Don't worry.
Okay. Better?
Yes.
Okay. So, basically cell surface proteins, they are just 2% of the total proteome. So if you just lyse the cells and put it in a mass spectrometer, only 2% of the total input material is what your proteins of your interest. So you want to enrich with the cell surface proteins and make sure that what you're injecting is mostly the cell surface proteins and you get rid of everything else. That's enrichment, right? So how do we do that? So you have to, again, you have to discriminate between the cell surface proteins and non-cell surface proteins, physical cytoplasmic proteins and nucleic proteins. So if you label the cell surface proteins something, so that you'll be chemically able to, by affinity chromatography, that I'll also speak about that later. So typically, the state of the art in enrichment proteomics or in biology, we make use of the structure-biotin interaction. Structure-biotin interaction is the strongest covalent bond that we know, right? So this structure-biotin based pull down is the most efficient technique. But there are some variations of it, like we use neutravidin, some like high capacity neutravidin, all those variations are there. But in principle, it's structure-biotin interaction that we can make use of for this kind of pull down. So let's say we have the cells. The cells come straight from your experiment, from your flask, and you want the cell surface proteins of these cells to get labeled with biotin. Cool, can we go to the slide where we have that?
I'm trying to share the slideshow right now.
Yeah. So we need that slide where you're showing the oxidation spectral.
Right. I'm trying to airdrop it.
Oh, I did get it done.
Yeah, well, Sanjay was working on it. Let me take more about the principle. So, how do we label the cell surface proteins and make sure that nothing else gets labeled? How do we do that? So here, there are several strategies that
Two more? Yes.
Target the license and all.
Oh, sorry, not that one.
So for just not for
download
membrane, it doesn't pass the membrane, and just make sure that the labeling is going. All those strategies are all there. Here, the strategy that we will be making use of is glycoprotein. Most of the cell surface proteins are N-glycosylated. Why is N-glycosylated? Glycosylated makes the proteins more resistant to any harsh chemistries. The cell surface proteins are exposed to sharing. Intercellular proteins, they are taking care of the pH, osmolarity, everything is very well maintained inside the cytoplasm. But something that is outside the cytoplasm, there has to be more, the PTM, like glycosylation is very So cell surface protein, 90% of them are glycosylated. So we use the glycan molecule as a handle to specifically label the cell surface protein. So what do we do? So glycans has glucose in it, right? The glucose derivative. So it has an alcoholic group in it. So what we do, we add this. Oh, you have gloves. Okay, so we add this reagent called sodium metapyrrolate. What it does is that it converts the alcohol. It oxidizes the alcohol into an aldehyde, right? And that's the first step. But before that, you have to make sure that when you harvest the cells,
it needs to be washed at least like 2 to 3 times before you actually start the procedure. And that's basically to get rid of all the remnants that comes from your media, because the cells will be growing in some kind of media, right? So what I'm going to do now is transfer these cells to a different view.
If we don't instrument, they can also check from
So 3 to 500 hours here,
yes.
typically for 5 minutes and 2 to 3 times of it. It takes 3 washes, but just to expedite it, we don't want to say here and go straight for dinner. So we want to cut short the incubation times and all the washing steps. Right now, I'm incubating it for 2 minutes at 500 hours here. Typically, don't go beyond 500 hours here for 5 minutes. And typically 3 to 5 minutes is enough. Just to extract it and make it for 2 minutes. And I'll do it just once now, but naturally would call it maybe 2 to 3 times. I prefer 3 times. And this is just to wash the cells, get rid of the carrier. Right, so you'll see a thick, nice pellet. And now let's assume that we did it 3 times. And I'm just trying to get rid of the supernatant. And now so all the resuspension happens in PBS. So I have PBS here. I'm going to just resuspend it in PBS. And we can gently pivot it up and down so that we get a nice suspension of it. All right, so now I have the cells resuspended it in PBS. Now I'm going to add sodium metabolite. It's an oxidizing agent. Will oxidize the alcoholic group to aldehyde group. So this is my vial of serum metabolite. If you need the exact protocol, you can share the exact protocol with all the details like what with stock concentration, what to the final concentration. But yeah, for now, in the interest of time, we're just demonstrating what do we add, how do we add.
So pretty straightforward. And these reagents are pretty cheap. It's not nothing very expensive. You can either use a gentle vortexing, or you can just use with a packet, you can up and down to make it. And this code in a disciplinary rotor. We don't have it here. We just, I'm sure like all of you have seen how end-to-end rotor looks like, which is fitted in. And this goes for 20 minutes. In that 20 minutes, you can have your breakfast, tea, breakfast, whatever you have in your schedule. So that day in the lab. And assuming that 20 minutes is over, now again, I'm going to put it in the centrifuge and get rid of the excess serum effect. So sodium hydrochloride is going to oxidize the glucose residues of the proteins, right? And surface proteins, because that's basically the cell surface proteins are kind of glycosylated. I mean, some of the cell blocking proteins are also glycosylated, but our next reagent that is impermeable. So even if some of these gets into the cell cytoplasm, although this also doesn't go back because it's in the chest, but that will take into the next step that it won't get back, although it might get oxidized. Right, so 2 minutes. Yeah. Yeah.
What's the specific? Just the sodium metabolite.
That's at this point, okay, I don't know. So I can look into it back to you. Or you can look into it right now. The other thing I know that the oxidizing agents are very corrosive, so you have to have that sweet spot of that it's still an oxidizing agent, and it's not toxic to the sodium hydroxide. That's also you don't want to add that to the
This protocol can be used for some other subcellular organ. We're studying membranes of some other organ.
If those proteins are glycosylated and glycosylated to be more precise. Because we are targeting the glucose residue. Sorry. All right, so your surface of the cells, they have glucose residues. Now they are oxidized. Now you want to get rid of all the oxidizing sodium hydrochloride. For that, you have to wash it 2 to 3 times again, so that you don't want any carryover in subsequent steps. Because that will neutralize the reactivity of the next reagent. So here again, in the interest of time, I'm not going for all the washes, but assume we wash it again. Basically, resuspend it in TDS and spin it down again, get rid of the supernatant and resuspend it again in TDS. Just wash it 3 times. And that's how you get rid of all the carryover sodium hydrochloride. Next, want to again resuspend it in TDS. Now we want to add 2 reagents here. Our glucose residues are now oxidized. We also install a bioclean on those oxidized residues.
Okay, so we would have to add biosaccharic hydroxide. So this is going to react to the aldehyde, and then we would have biotin installed on those glucose residues. Again, the exact concentrations and all we can share. Who is personally interested, just let us know. We'll share the exact protocol. And we need a catalyst here. Our catalyst is aniline. And we just need 1 microgram of aniline. Again, mix it well. So now this goes again in end-to-end rotor is what it is, refrigerated for 1 hour. And this is a good time for your, so you basically timed experiment such that this is your lunch time. And that's how I used to do it. Assume that 1 hour is already passed, but again, spin it down. So you look at that reaction, like the other reaction, we install the biotin. Biosaccharic hydroxide, yeah. So by this time now you have the cells here, and the cell membrane proteins, they have now biotin installed on it, right? The rest of the cells, rest of the proteins in the cytoplasm, they don't have biotin. Now your cell surface proteins are labeled with biotin. And now I'm just washing it, so I'll be washing it now 2 to 3 times again. Want to get rid of all the biotins, 3 biotins. So why? The next in this kind of cascade reactions, make sure that it's washed in every step, so that there is no carryover. What will happen? Let's say if you don't wash it properly, what will happen is that when we add the intermittent leads or the intermittent leads, certain meats will be already blocked by all those prebiotics, and you won't have enough space on your bacterial beans on your supervision beans to put it on the actual proteins of interest.
What was the
Catalyst.
for that?
Yes.
Do you want to show this? It's already shared. Which one? I would like to share something. It's nothing or not? Oh, I know it's good. I think we should keep talking.
We have converted the involved group to the SAI group. Now you're adding biocycling
On that.
on that.
Could you repeat your comments or questions and then answer?
So she's just cross-check from what she understood. Basically, she's asking that where the biotin is getting installed. Biotin is getting installed on the aldehyde. Aldehyde, we had sugar, right? And the glycosylated proteins. That doesn't have any aldehyde group. It has alcoholic group. We oxidize that alcoholic group to basically OH group to CHO, right? And we install the biotin on the CHO. What do you want? So now you have cells, the cell surface proteins labeled with biotin, right? Until now, we were supposed to be very handle these cells very carefully. We don't want to lyse them. We don't want to damage them. Because if the cells are dead, then it will be the reports in the cells, the reagents will pass through that and to enable the intercellular proteins of the cells. So until then, we need to be very careful about the integrity of the cells. The cells are alive, and make sure that you keep your eyes as many times as much time as possible. I was not doing that because we have to broadcast it on online. So, but yeah, now we don't care about that part. Now you can handle it as much as you want. We can next step is lysis. We lyse it. Now your cell membrane proteins are biofiltrated, right? So you lyse the cells, lipolysis, and then you add a stricter leads and pull down the membrane protein. That's the whole principle. So, so typically this is day one. This whole thing we do one day. If you proceed to the next step, you'd have to stay in the lab for night. So typically, what we do is that's day two. So this goes into biosafety. You can snap freeze it, put it in liquid nitrogen, and then put it in biosafety. And then you can proceed further whenever you feel like. Maybe next day or maybe after a month or so. Doesn't matter.
Yeah.
After adding the lysis buffer, it can be stored.
Good question. I haven't done that. You can do it and let me know as well. But in principle should be okay. But typically, like, why do you want to do that? Like, neither label cells is a good story in MaxSec. Next step is not just lysis, not just adding lipolysis. Once you add lipolysis, you need to sonicate it. Lysis cells. Okay? And Apul, you want to take it over from me? So I have my flight, and considering the flood in the city, I have to leave early. So from here, Apul will take it over for the rest. Thank you for your time. Yeah, I have some time. If you have any specific questions, I can come to you and explain it.
If we want.
That's a good question. So typically, so we have applied it on. Am I audible? No. I should be. Yeah. So we have applied it so far with the suspension cells, and suspension cells, they are single cell suspension cells, right? Basically, the team of our lab works in malignancies, so all the cell lines that we use, so all of them are suspension cells. So it's pretty easier in suspension cells because it's already single cell suspension. In case of solid tumors, we haven't tried yet, but basically what you have to take care of is that you don't want to use something, many promiscuous proteases like protein SK or any pancreatic, I don't know. There are some proteases which is used to typically for plant circuit. You don't use that. Maybe you can use something, I was discussing that with actually yesterday, you can use something like collagenase, just specific for collagenase. You don't want to you don't want to start a proteolysis of cell surface proteins. So if you just digest the matrix, that should be okay. That's something I have not came across anybody who was studying. We have done it once with the organoids. The results were not great, but good enough to get a grant. We got that grant. But that was a big grant. I think that's the work still in progress. Organoids, they're still, I guess, it's to be attached to each other. Cells that would be attached to each other compared to a tissue. Not just the tissue, like even for rim cells, you might have to modify and step ahead. Like what you cannot use trypsin. If you use trypsin, there basically you are digesting the surface protein. Then what you are looking for, digesting surface proteins. So you have to come up with that kind of optimizations. Maybe use EDTA, so that the development times, and that's how you can detach the cells. That kind of optimization. So same thing beyond surface proteins, surface results, you need more optimization. Yeah.
So in case of ended cells, trypsin is not to be used to for this work. Then scraping would be okay.
Scraping would be okay if your cells are fine.
Which one?
EDTA. We don't want. EDTA is one option, but scraping will also work from the, but you don't want to damage the cells. Scraping will end up damage the cells. If you damage the cells now, as I said, that the integrity of the cell membrane has to be maintained so that all the reagents doesn't have access to the intercellular proteins.
So what would happen if you label your proteins on the adherent cells? I mean, you don't have to only after lysis, you label them and then lyse them. Would that be a good idea?
So we label and then lyse anyway. I mean, you mean
that's what I'm saying for adherent cell. You just have to use much more
reagents. Yeah. So you mean label them and then detach them?
Yeah.
Yeah, that's an option. Yeah. But I mean, that way you would have access to half the surface area of the cells. The other half will still be inaccessible. So it's basically, you have to play with it, like, which is better. You can try first with scraping, and then as Sabis actually suggested that just before attaching, you can label them. And whichever works better, you just go ahead with that. These kind of optimizations are needed for anything other than suspension cells. Suspension cells. That made our life easier.
We've done some. We've done some adherent cells. You can use. There's also citric saline solution that works. That's gentle at detaching the cells, gentler than EDTA. It's a citric saline reagent. I can send you the exact composition of it. It's also a chelating reagent.
Yeah. Any more questions? Enzymatic biotin and NHS biotin. You mean the glycooxidation based biotin and NHS based biotin? That's a very good question. Okay. So, okay, you tell me. NHS biotin, what does it target? Okay, which amino acid has amine group? Lysine. And lysines are one of the most abundant amino acids in the protein, right? So, if you label all the lysines with basically you install biotin on all the lysines, what does trypsin have? Next to arginine and lysine. So basically now you're compromising the digestibility of your proteins because your lysines are now modified, right? That's one problem. It's not a catastrophic problem. It will still work, but it's just that it just compromises the efficiency of the whole protocol. So that's one reason that it basically compromising the digestibility of trypsin downstream. Because at some point you will digest the proteins with trypsin, right? And then we have peptides, and that's what you inject into the mass spec. You don't inject directly proteins. Mass spec doesn't read directly proteins. It reads peptides. It tells you what peptides are in your samples. Not you backtrack what proteins those peptides came from, right? All right. So the first problem is the digestibility with trypsin. You compromise the, you modify the trypsin. Second problem is that in pull down, what happens is that all the biotilated residues, they gets bound to the beads, right? Have you heard of detaching biotin from beads? That's an idea. So this is, as I said, that lysine is one of the most abundant. Is one of the most abundant amino acids, right? So basically, all the modified biotilated lysine-containing peptides, they will be bound to the beads. So what you will get, it will be all bound to the beads. I have tried that, and you have to trust me on that it doesn't work as good as this. Yeah. I don't have a very good explanation of why is that so, but yeah, I've tried those things, right? So yeah, I mean, so basically that is peptide-centric labeling. If you do peptide-centric labeling, so you lose all the labeled peptide, they're still bound to the structure in beads. There's a way to get rid of it. You get it back by heating, and there's other ways. I honestly don't like those kind of steps. That's my personal opinion. Others may differ from it. Yeah.
That's a good opinion because even if it does work, it never gets rid of all the things you want it to be.
Yeah.
I've done proteomics. It's just you start with lots of material and take into account all the losses, and then you can think about the protein.
Yes, yes.
Wondering how do you get to the rest of the 15% which are not Indian?
15%, which 15%? Try other method. This method cannot. It does, but that's like specificity bias. With this protocol, I would say, I mean, to be honest, just say 30% of the proteins are self-sufficient proteins, but those 30% represents 80% of the total iron chromatin. Basically, at the iron level, iron abundance level, you are enriching at 80%. But rest 20% is possible because the mass spec is so sensitive. How many? So in a typical self-sufficient protein experiment, how many self-sufficient proteins do you see? 1,000, 1,200. But your mass spec is still has the capability to see like 3,000, 4,000 proteins. So the rest of the proteins, it will detect. Although it's in a very trace abundance, it will detect them. But the total ion intensity will be low.
You talked about specificity.
So if you use a density, yes, the protocol, your non-specificity would be high. I don't think you can get. I haven't done it, but in principle, my opinion is that you won't get 80% enrichment with that, because this is here you pull down the biokinetic proteins, you show, and upper will show you wash with 50 ml, you have 3 buffers, so 50 ml per sample. So yes, you pull down only the glycosylated proteins because you install the biotics on the glycosylated proteins. So the washing step is so rigorous here that you get higher specificity. I'll give you an example. Like although 30% of the proteins would be self-sufficient proteins, the 70 would be the background. But like I personally saying, like integrins, they are very highly abundant on cancer cells. And I still, when I rank them, it still shows up at the top, like second most hit or third most hit. And otherwise, in a regular, cell lysate, self-sufficient proteins will stand nowhere in terms of abundance. Any more questions?
Yeah. Have you ever tried this protocol for nuclear surface protein?
Nuclear surface proteins, are they glycosylated?
Might not be. Plasma membrane proteins are almost all of them are glycosylated, but nuclear protein, not sure all of them are.
I would assume they are not.
How would you do it? Like you have to lyse the cell to reach the nuclear membrane. Right?
You can fractionate. There are fractionation ways to fractionate, isolate the nucleus, and then you can label it. But if they are glycosylated, I may be wrong, but in principle, I believe that nuclear membrane proteins might not be glycosylated.
There is no, as far as I think, there is no function.
Yeah, because they are not exposed to the extracellular environment. Glycosylation helps to make the proteins, more resistant to extracellular harsh environment. And that's for the same reason the cell surface proteins are also that have disulfide bonds. Right?
Is there any other way you can enrich the
nuclear envelope proteins? Okay.
So there is
yeah, please.
So there are ways of fractionating. People have been doing biochemistry for very long on nuclear membrane proteins. I think I was just collaborating. 3 papers came out of collaboration with Larry Juris at Scripps, and it's biochemical fractionation and how well you do. So it's old school biochemical fractionation. You need to play with lots of optimization. And finally, you come up with that. There are other ways. So we also applied what is called proximity labeling. If you have heard about it, it is Turbo ID, Bio ID, Apex. These technologies where you have protein biotinylate
interacting proteins in proximity, and you can then enrich. There are pros and cons for everything, but these are some of the ways that you can try to enrich for proteins in particular localized environments. And nuclear envelope can be one of those. Of course, nuclear envelope proteins have the complexity of being membrane proteins, and all those who have been following proteomics, membrane proteins are the hardest part going now. So, yeah.
Any more questions?
I have a question.
Yes, please.
So here we're talking about the bottom-up approach, right? Like, yes. Have you tried the top-down approach? Like, yes, like if you're in a hurdles or where are you with that? Rolling or is it still not to do in the way?
Okay, so just to get everybody on the same page on this question, saying that what we are doing here since morning is all mostly we are talking about whatever proteomics. When you start with proteins, digest something like trypsin, it could be chymotrypsin or whatever, and then you get peptides, and you inject the peptides, and you have to backtrack what protein is coming from. But in top-down proteomics, we inject I haven't done it, but we just inject the proteins themselves, and then you want to go.
I have not done it myself, but there are people in my lab who have been doing it, and I know a few. But it's going around. It's doing well, but there are lots of other. It means it's relatively in infancy, I would say, but no more in infancy. There are people whose labs are completely devoted to these kind of things. It's a very different approach altogether. Bottom-up proteomics itself is a huge field. It has its own dimensions. So labs are quite dedicated to this approach, and then multiple approaches can be difficult to follow. But in top-down, so top-down is something where you take the protein, and the two ways you can do top-down. The more native state is, of course, where it's not exactly native. Me coming from cell biology kind of background, native can mean very different. For biochemistry, it is native. So it goes usually into about 100 to 200 millimolar of ammonium acetate, and whatever is the state of the protein at that point of time. A lot of membrane protein has been done that way. Carol Robinson in Oxford, she's using some mild detergent to stabilize these membrane proteins and do things. For most of the other top-down, it's the whole protein. We are not concerned about the native structure of the protein itself. With native, you can do complexes as well, multiple protein complexes, because they are supposed to stick together proteins in the native state in a complex as naturally present. For top-down, you don't care for that as much, but the fragmentation might not be as nice as you can do with a peptide fragment. Also, the algorithms and computation that has been developed for bottom-up proteomics has been much more advanced. There are very few labs who are developing, hardcore top-down proteomics approaches in terms of computation, and that has a long way to go. So, somebody from my lab who was doing native and top-down was in touch with Neil Kelleher's lab in northeastern, I suppose. And they had some dedicated software, but he was basically interacting with the person who was developing the software. So that way it is still in infancy, even in terms of the analysis. But it's not something which you cannot do. It's just that it needs a whole different dedicated approach altogether.
I think you already mentioned the names. You can refer the Carol Robinson's paper, Albert Heck, and Neil Kelleher. Some of these are the key players, leading labs, which are doing the top-down. But it has much more robustly grown as compared to earlier. Now there are more software which can do analysis in a more automated way. But you have to do a lot of biochemistry over there to really understand the structure well, right? That's why it's not as easy or straightforward as doing the your bottom-up. Right? All right, yeah, good.
Yeah, I also think that most of top-down and native, it's going for a proteoform. So the different post-translational modifications, they can be labile and they can get off in bottom-up proteomics. So a lot of people who are doing this, they are targeting it for like different proteoforms. When I mean proteoforms, it basically means different versions of the same protein for different reasons, cellular reasons, including post-translational modifications. So, of course, there is a big advantage in using those approaches, and they are being explored.
Just to add a few more points to it. So, top-down, the best thing about top-down proteomics is that you get a very high coverage. So if you want to do, as Saki said, that if you want to do some like proteoisoforms where you might get so many of shared peptides, you need high coverage to exactly determine exactly which isoform of the coverage coming from. When I say coverage, think what portion of the protein your peptides align to. Typically, I mean, we prefer 30%. I mean, sometimes just two peptides also enough to cover quantification. But top-down proteomics gives you a high coverage, and with that you can do PTM analysis, we can do isoform analysis, but then that's feasible more at with samples which are less complex. For example, pull down material, pull down of a complex, pull down of a protein. But at a proteome scale, I'll give you some numbers. You know, proteomics community is very obsessed with numbers. We want to see thousands of proteins. So the typical bottom-up proteomics, just with 2 hours of run, 2 or 3 hours of instrument run time, you can get maybe 3,000 to 5,000 proteins very easily. Now, let me compare the top-down proteomics. I haven't done it myself, but I've heard it from the experts. 20 fractions, 2 hours run time each, so 40 hours, and all that for 2,000 proteins. If that tempts you, you can go for it. So definitely not look at it at proteome scale, but yes, at for less complex samples, it's very lucrative to achieve higher coverage. Any more questions?
I guess we can continue Akul permission.
So with that, I'll hand over to Akul, and I would have to head to the airport now.
Same only. Turn this one off. I'll use the.
Okay. This was the sample. Yeah, that's seriously. Okay, so as Kamal mentioned, usually everything he showed is what we do on day one. It takes about, I'd say 2 to 3 hours to do the whole labeling reaction. And then you can take all the PBS and liquid out of the cell pellet and freeze it in liquid nitrogen, keep it in -80 as long as you want. So let's say we did that yesterday. So today we take it out. We take it out of the freezer, we thaw it on ice, and the first thing we do is lyse the cell pellet. Yes. Okay, so this is going to be 2x ripa buffer with protease inhibitors to inhibit any intracellular proteases because you don't want them chewing up your proteins, and EDTA. So we usually add 500 microliters of this into a sample. Add 500 microliters and mix it up and down really well. You should see it clear up the pellet. You might see some gooey substance in there, which will be the DNA, which will be cleared out when we do the sonication the next step. But we can be really vigorous. You don't have to be gentle with the cells. Pipe it up down a lot to get the pellet nice and resuspended and the cells all lysed. And then I usually also sonicate at the It's going to be 10 minutes at the high speed, which is 17,000 feet. On this one at 4 degrees Celsius. So while that's spinning, we're going to set up our column. Just turn that off. You can pretend that's spinning while it's off. So, great. So now we're going to get our column. We're going to prepare our column, and we're going to add 100 microliters of this high capacity nutravided resin to it. So these are the nutravided beads that are going to bind the biotin proteins. They tend to settle, so shake it up really well. So at this point, we put this column onto our vacuum manifold, which I have a picture of here. We'd attach the column with the beads in it to the vacuum manifold, which we don't have here right now. And then we'd add chip. And we're going to add 3 milliliters of this wash buffer just to equilibrate the beads into the same solution that the cells are in, just to make sure they're in the same condition. So you add this. Normally it'll flow through. I'll just try to push it through.
What is that? Key.
You can see the buffer flowing over the like that. You're going to do that 3 times. Add 3 milliliters just to make sure the beads are in the same buffer. Yes, I think it was just up on the screen. So now we're going to take the column, cap the bottom of it, make sure you have it nice and tight so nothing goes through it. And we're going to take our lysate, pretend that's nice and clear and settled at the bottom. And make sure you don't, when you move this lysate to the beads, make sure you don't disturb the bottom to prevent getting any of that debris. Take the lysate, add the 2 beads. And then you're going to cap the, you're going to take this cap and add it to the top of the beads like this. Now your lysate is mixed with the beads, and you're going to let that incubate together for 2 hours in the end-to-end rotor at 4 degrees Celsius. So this gives time for the biotinylated proteins to bind to the beads, spinning like this. So let's pretend it's been 2 hours. Our lysate is finished incubating. You're going to put the column and uncap it and put it back on the vacuum manifold, as seen in this photo here. As you can see, here's a close-up of how it looks. So you can put this, and then when you turn on the vacuum, all the solution that's in here is going to go flow through the column. Now all our proteins are attached to the beads. They're no longer in solution, so you don't have to worry about the flow through. That's junk. And now we have 3 wash buffers that we wash the beads with to wash away any non-specific intracellular proteins that may have bound to the beads due to non-specific interactions. So the first wash buffer is this 1X RIPA, the same lysis buffer we used, and EDTA.
Not any other
That's the lysis buffer we used in the lab since I came there. Kamal, is there a reason? Kamal might know better than me about the reason to use RIPA specifically.
Yeah, why you don't want to use RIPA? I second that answer. Okay, so for this case, I know our mass spec columns, they don't like SGS, but in this case, it's fine because we're lysing the cells and we pull down through with the help of beads, and then the beads undergo very vigorous washing. You'll see like there are 3 wash buffers, and each sample is subjected to 50 ml of each buffer, so 150 ml per sample, right? And these buffers are very stringent. I mean, buffer 2 has like 5 molar, 10 buffer 30 is 5 molar, and something like that. Or even more. This is pretty stringent washing, so you pretty much get rid of all the issues. And we have used it over the, I guess, by this time, like maybe 5-7 years, and we more than that. And we don't see any issues with the column.
For the device is better also.
Yeah, so we use 2X RIPA. We could use guanine hydrochloride. I haven't tried that. But sorry, I don't remember. I think there is one reason which I'm not able to get now. Why we went from guanine hydrochloride.
I suppose the determination must be doing much better for membrane acidity.
Yeah, that could be it.
So membrane proteins is difficult to extract, and if you don't have very strong surfactants in there, you might not be able to see them at all. It's really difficult to extract membrane proteins are. And so they are basically lysing the whole cell. If it's not lysed properly, then you are expecting, and not going to expect the membrane proteins to stick on the beads. It's adapted to it. You know, if you methanol chloroform extract and the thing, if you can go only at protein level, I've seen the membrane domain just crunching down. So that's always, and that's the reason. If you extract it properly, you can't go into the transmembrane region because the protease, extent of protease digestion there becomes difficult. And if you don't, then you anyhow don't get it. So both ways it's difficult for them.
So you need very rigorous lysis. And also, like, it's not 1X RIPA, it's 2X RIPA. Yeah.
Okay, so we've just flowed over, pretend we put 5 milliliters of wash buffer 1, flowed that over the beads. That should remove any non-specific proteins. Now we're going to continue and do that with wash buffer 2 as well. So wash buffer 2 is basically PBS with added sodium chloride. So basically the wash buffers are moving closer from the lysis buffer to the digestion buffer. So wash buffer 2 is kind of a neutral buffer. PBS, that's the transition between the 3 buffers. So again, imagine I've taken 5 milliliters of wash buffer 2, put it on the beads, 2 milliliters at a time in the column, turn on the vacuum, and that 5 milliliters again has flowed all the way over the beads to wash away any more non-specific proteins. And finally, we have wash buffer 3, which is 2 molar urea and 150 or 50 millimolar ammonium bicarbonate. The reason we use urea is to denature all the proteins so they're accessible to the trypsin that we're going to add next for the digestion. And the ammonium bicarbonate makes it a basic solution that's also good for the trypsin digestion. So again, we flowed over 5 milliliters of wash buffer 3. And after allow the wash buffer to completely flow through, so only the beads are left. And at this point, you're going to take the beads in your reducing and alkylating reagent that, as you explain later, you're going to take it into a separate tube and then continue with the rest of the protocol as with normal global proteomics. So these two steps that Kamal and I demonstrated are really what's unique about cell surface proteomics. The rest of the protocol is identical to your normal proteomics processing. Alkylating and reducing, followed by trypsin digestion, and then desalting of the peptides. So there's a little bit left in my presentation. So there's a couple of optional steps that we personally don't do in our laboratory, but you could do if you wanted more data or if you had a specific experiment type that you need this kind of data for. So something that's included in the original Bert Wolshield paper where she describes cell surface culture. Yes. So it's this technique called PNGase release. PNGase is an enzyme that cleaves the end-linked glycosylation pattern. So essentially so as you can see, when we do our normal protocol, we do the on-bead digestion. So we digest the proteins when they're still attached to the nutraminid beads. We take those proteins that have dissociated for our proteomic analysis, and we are left with beads that have a single peptide that actually had the end-linked glycosylation on it still attached to the beads. So normally in our lab, we just toss these out. We don't do anything with this. It's a single peptide. We get enough information from the remaining peptides to do the work we need to. But an option you could do if you wanted information from these peptides as well is you could treat the beads with this PNGase F enzyme that would cleave the end-linked glucose, and you'd be left with these free peptides, which you can then take for as a secondary sample and analyze in the mass spec as well. This would give you these peptides, as you can imagine, are highly specific for cell surface proteins because they were the specific peptides that were glycosylated, so they had to have come from the cell surface proteins. But unlike our normal sample, you only have one peptide per protein, which makes your analysis much less confident in those proteins. It's a much smaller sample, so you're going to identify much fewer proteins, and you need to use twice the mass spec time, which if you don't need this data, it doesn't make sense to use. So since we talked about cell surface proteomics and why we want to do it, I wanted to highlight some applications of where this could be useful, both from within our lab and other groups. As you can imagine, this gives you a wealth of information about the whole cell surface proteome, how the cell interacts with other cells and the environment. You can get structural and functional information about the proteins themselves. You can get information about the cell and its dynamics, how it reacts to the environment, how it reacts to certain treatments and certain drugs. And our lab specifically, it's very useful because cell surface, when you use the cell surface proteome of cancer cells versus healthy tissue, it can highlight novel targets for cancer immunotherapy, as Kamal discussed earlier. And it's very exciting because we use cell surface proteomics, this last paper from Matt Nix and Kamal at our lab. We were able to identify a cell surface protein on B-cell leukemia that might be worked up to a clinical trial. So that's really exciting. So thank you, everyone. I want to thank Arun, our PI, Sanjeeva, for putting this conference and helping us so much throughout his year at the lab last year, and Abhilash, who helped with this presentation and also taught me a lot about proteomics. So, any questions?
Akul or Kamal, so have you tried, so my understanding is in solution digestion is more efficient than on-bead digestion usually. So how would it have been if if you use, even after your enrichment with biotin, short-tablet enrichment post-well, would it have been good if you would have used PNG's F because it would still be acting on deamylating and releasing things, proteins, the glycosylated proteins? Then you would have the whole proteins on, in solution, and then you would be digesting them. Do you feel about that? Anybody? Okay, yeah.
Yeah, I can talk about it. I think that is like, I totally understand what you're saying about doing the PNGH treatment before we do the digestion, just so we get the whole protein and we avoid the on-beat digestion.
Yeah.
It is an extra step, but I think our lab just realized that we had really efficient digestion on bead. The beads are in solution and we get really efficient digestion. That's definitely something you could try if we wanted more data or we didn't want to miss out on that end-link glycosylated protein. Peptide as well.
Yeah, I completely understand that. It's just that the usual trend is that on-beat digestion usually is more difficult as compared to in solution. So it might be a way to maybe increase your coverage or things of that sort.
Right. I think our on-beat digestion isn't too different than in solution because the beads are in solution when we do the digestion. Yeah. So, but yeah, I totally understand what you're saying. Yeah. Thank you.
So I agree with your idea of first diluting with PNGs. But the problem, potential problem that we may get into, it may not be a problem, but what potential problem that we have with PNGs, that's also a protein. That is still being here, and that might be a very high level.
Yes, so that's true. If it's a relatively complex mixture, you are adding one of those. Yeah, that's true.
Order of the mass spec.
Yeah, you're right. You might be affecting. I have been using those things, but I have been usually using them on purified proteins. So even though I add these glycosidases, my purified protein still kind of outcompetes them. But yeah, in a complex mixture, it might make you lose low abundance things.
Even in the membrane protein, there are some proteins which are, highly abundant and some proteins are less abundant. So when you are still, enriching the membrane protein, like some of the proteins which are highly abundant will mask the lower abundant membrane protein. So is there any, how to get those low abundant membrane proteins? Because, they'll get masked anyways.
That's something that is very routinely done for something like plasma proteins, because your serum albumin is so much in high excess, it dominates your mass spectrum. They could be enriched for plasma protein. And something similar happens with the chromatin proteomics, histones proteins proteomics, for the histone proteins. So here it becomes like, do you like a mass spec is, current mass specs are the order of 10 7 or 8. Something like that. Basically, if you can get rid of the top, 90%. See, you
also work, right? Fractionation will also help.
Yeah, fractionation, helps with any kind of, you may increase the coverage in any protein experiment. But it all depends on like how, you can get rid of the top 3-4 proteins with maybe the immunoprecipitation, distributed with the antibodies. So I have to come up with a strategy, how do you do that? Very good to de-enrich for it. But the question is how much would it help? What crop, if until you are not depleting at least 40-50% of the most abundant proteins, I don't think it would be helpful. You just deplete 10, 20, 30%, I don't think it would be helpful. It all depends on how many of the top abundant proteins you want to and you can deplete, and how curious you are about the low abundant one. Basically, cost calculation in terms of resource and time.
We are not getting they are
low abundant targets, again, as I said, the proteomics people, they are very of specific numbers. We want high numbers. We want the word. We feel very proud. We got like 5,000 proteins, 6,000 proteins. We got low abundant proteins. We feel proud of it. But think aside, let's say for immunotherapeutic purpose, immunotherapy. You want to targets which are in high abundance, okay, not just which are in low abundance. Because one of the relapse mechanism with cardiac therapy is that let's say we're targeting CD19. If you treat the patient with CD19, and most of the relapses, what happens next time the relapse of cancer happens, this time your cardiac therapy won't work. Reason being, CD19 is not down. It's not downregulated actively. Because what happens is that you kill all the B cell population, the triple population, the high abundance of CD19. Once they are low abundance, they get spared. They are not killed. And now this population takes over your whole tumor burden. So low abundant proteins, it depends like why is it interesting to you. For immunotherapeutic purposes, like I believe it's not at least at this point. But for me to understand the biology, that might play a crucial role.
It cannot be a very good biomarker. It can help you understand the
Yes.
biology.
Yes, it all depends on your question, like how desperate you are for those low abundant cells and how much time and money you want to spend on de-enriching the top. For something like plasma proteins, you have to get rid of that and put that in order you want to. So here it's not like that. That's not the case.
So thank you, Dr. Kamal and Apple, for walking us through the protocol step by step. That was a very detailed protocol, and so, we have a token of gratitude for Apple Nayak as well. So please give a round of applause. And I request our speakers for the
Well, it was a wonderful session. Thanks, Kamal and Nakul. And I'm sure all the participants will appreciate that, protocol, of course, we can't show the complete way, but at least we got the gist of the chemistry behind it. And Joe protocol paper we have shared with you. I'm sure now you can try, you are motivated enough to try new things, right? It will be pretty new, in fact, thinking, bringing the cross thinking and Proteomics in India. Bringing up our labs, the first one to do it. Right? So please do try. All right, we request all the speakers to come up here and we'll have a good picture. The ground, please come and show. Yeah. Can we shut down the screen?
Please continue.
Be a muscle. Make a bone. Carry nerve signals. DNA tells our cells exactly what to do and when to do it. That's why DNA is often called the blueprint for life. But how do our genes give such orders? Each gene consists of a recipe for a certain protein. Proteins are the machines and workhorses of our body. Proteins trigger cells to perform specific tasks, enabling the cells in your eyes to function differently from the cells in your heart or brain.
Our bodies are made up of about 100 trillion cells, which each contain thousands of different proteins. The collection of proteins within a cell is called the proteome. The proteome is the counterpart of the genome. Our DNA remains the same throughout our lives, but our proteome changes. This explains how the grown-up flying butterfly can become something so distinct from the young crawling caterpillar it once was.
Few proteins work alone. They interact and form relationships with each other, and the nature of these interactions changes over time. You could say proteins have their own social network, just like us. Some proteins have lots of friends and contacts, while other proteins only have a few. Sometimes, a protein will break up an interaction and start looking for a new partner.
With approximately 10 billion protein molecules, one single cell has an interactive network that is as complex as that of all humans currently living on planet Earth. These protein networks determine the health of the cell, and thereby the health of the entire organism they are a part of. So it is easy to understand that the study of these proteins and their relationships is crucial. This study is called proteomics.
Understanding how cells work has made it possible to develop revolutionary drugs that are more effective and have fewer side effects. To detect and identify different kinds of proteins, we measure their unique fingerprints with a technology called mass spectrometry. This technique can determine the identity and, indirectly, the function of proteins.
The process involves shooting charged protein molecules or fragments thereof into a vacuum tube and measuring how long it takes for them to fly to the other end. This flight time depends directly on their mass. Another way we learn about the molecules is by making them collide with gas molecules, which causes them to fragment into smaller species. By measuring the mass of the fragments, we can determine what the protein's building blocks are and can thereby identify it. In one single experiment, we do this for tens of thousands of proteins and roughly 1 million. Huge amount of data we are constantly producing by measuring proteins. Gigabyte after gigabyte. This information helps us to precisely identify proteins and the social networks in which they exist.
But proteins are not unique to humans. Every living species on Earth is built up of proteins. Jellyfish, polar bears, tulips, and even bacteria. For this reason, study of the proteome will ultimately lead to better knowledge, not only of diseases and the effectiveness of drugs, but also of how plants can adapt to dry or salty environments, and how we might eventually be able to combat bacterial resistance. Excitingly, in recent years, scientists have learned how to reprogram the proteome of skin cells in a lab and turn them into stem cells. In other words, scientists are now able to tell cells what to do. For instance, regenerate tissue or repair a damaged organ. New therapies for a healthier society are on the horizon due to the fascinating research of proteomics.
Yeah. So we are actually here. We talked about proteins. We talked about the protein sample preparation part. Today you'll be doing quantification as well as the proteolytic digestion, which can be in solution or in gel digestion. And then moving forward, then you want to separate your peptides which you've obtained from your given sample so that you can reduce the complexity of that given mixture. Think about if the proteomics aim is to look at all the proteins from a given sample. You're looking at one of the tissue sample which may have 5,000 proteins. After digestion, you may get 50,000 peptides or more. All of them you want to separate in a single short mass spec run. That is going to be challenging. You want to reduce that complexity. That's why you use either different type of chromatographic principles or different type of object fractionation or IF principles to separate your complex peptides into smaller simplified mixtures. So this will be the second part. All of this peptide which you have made in the liquid form, or even sometime you want to analyze the solid form, those has to go to the annihilation sources. Followed by you want to separate those complex ions into the mass analyzers. Either you can use one type of mass analyzer to generate MS data, or you can use two different type of mass analyzer, which is known as tandem mass spectrometer, to generate MS data. Hi, welcome to all the participants who are connecting with us today live for the book session. I am Professor Vishwa, a group leader of Proteomics Laboratory and Professor at I have this primarily focusing on proteomics-based applications to investigate different types of clinical forms. As you can see, those are points here. I'm trying to show you some of the technology we use in our lab for the clinical proteomics. So what we are carrying nearby is one of the next editions. And I will explain you shortly that when this SARS-CoV virus started showing its impact and COVID-19 on the pandemic, it was needed to
Can we take it in the studio?
And tell us that what we see So, can we detect viral presence? Because of the virus, so first of all, we try to look for all All right, microphone, please. Yes, sir. All right, so now we have reached to the final step of sample prep. We have seen two different ways. One was the whole cell lysis, second is the enrichment for cell surface proteome. But either way of doing solution digestion now, right? So mainly you do afternoon so that you can wait for the system digestion to happen overnight. So now we are going to show very briefly the steps involved in doing the in-solution digestion. Of course, we can do in gel in a similar way. Only additional part over there is you have to remove the stain from your gel pieces. The multiple washes has to be done in the beginning with ammonium bicarbonate, and then you follow the same reduction oxidation and crystallization. So Sachin will now walk you through this step for the in-solution digestion.
So you have to unshake.
So, welcome back everyone. So, am I audible? Am I audible? No. Okay. Yeah.
No. This one is. इसमें on है क्या?
So right now, like post the post quantification, we now start with the in-solution digestion. So we usually start with anywhere between 32 micrograms of the protein. Sometimes, depending on the need of the experiment, we can start with slightly higher amounts of the sample. So I have with me a tube containing the sample. So this. If you remember this. During the quantification, the protein is
reduced, followed by alkylation, and then tryptic digestion. So now I'll be adding TCEP. Okay, so the reagent that you choose for this particular step is dependent on the buffer system that you are using to make your sample. Like most labs, they use a urea based buffer as the lysis buffer. Okay, so the concentration of urea usually is around 8 molars if tissue is the sample of interest, around 6 molars for any plasma based sample. Okay, so prior to your digestion, you need to ensure that the final concentration of urea comes down to as low as 1 molar, because trypsin has very low activity at higher concentrations of urea. Okay. Also, if urea is present in the buffer, you need to take care that you don't heat the sample, right? Which is why we are using TCEP, which works at RT, at room temperature. Otherwise, the most commonly used agent for reduction is DTT. But as you all know, for DTT to act, you'll have to heat the sample to around 90 degrees. But since there's urea in your buffer, you cannot use DTT directly, right? This particular incubation of reduction is for 1 hour, okay, and this typically happens at around 20 millimolar of TCP. The strength of TCP that you use is around 20 millimolar. And the next step is the alkylation. So the alkylation here is being done using IAA, that is iodoacetamide. And yeah, so iodoacetamide that you use is usually twice the strength of the TCP that was used. So if you're using a 20 millimolar TCEP, the IAA would be somewhere close to 40 millimolar. Okay, and this has to be incubated at 37 degrees in dark. Okay, so this usually is anywhere between 10 to 30 minutes, the step. Okay, and after this, you need to ensure that prior to addition of your trypsin, you check the pH. Okay, so the optimal activity of trypsin happens at around 8, pH of around 8 plus or minus 0.5. So it's advisable that you check the pH, even though you have checked the pH at every other step, or you may or may not have checked the pH at the steps before. But make sure that you do that before addition of trypsin. Then most labs around the world, they work with a trypsin is to protein ratio of 1:50, meaning that for every 50 micrograms of protein that is present in the sample, you use 1 microgram of trypsin. Okay, typically in our lab, we use around 1:30 as well. There have been protocols where we've used 1:30 as well. And also, I mean, there are 2 different vendors of trypsin. You can use either of them. We prefer the thermo trypsin. There are labs which also use the promega trypsin, which is a much more costly variant of trypsin. So after this, you incubate your sample for 16 hours. There are different labs which change the protocol. I mean, they'll be probably incubating it for different times. And after the end of 16 hours, you have digested peptides. These peptides are now to be desalted. And so desalting typically can be done with in-house zip tips, or you can use SEX fractionation. There are also reverse phase fractionation kits that are available, high pH reverse phase fractionation kits. So, and then setback cartridges. So there are different varieties of consumables available for that. We won't recommend any particular this thing for we won't recommend any particular product, but it is totally dependent on your budget and the application. So after that, your samples are now ready. You may quantify your samples. So we use what is called the scopes method in our lab. You can look up the main paper, the original paper. I think it was published in the 60s. Yeah, right. So after quantification, you can inject the sample into the mass spectrometer. So this is just a quick walkthrough through the protocol for digestion. Thank you.
So again, like, there are many details. How much amount of protein should start for digestion, right? What will be in like substrate ratio to be kept? Sometimes only one enzyme, sometimes a cocktail of multiple enzymes. Right? Many of these things you have to play with it to get the most efficient digestion. Then only you get a lot of peptides from the muscle. And before injecting, you have to do the cleaning of the peptide to remove the salt. Again, there are multiple options available for it. If we have some time, we'll show tomorrow. And also quantification peptide. In mass spec, the most important part over there, how much you inject in the instrument finally or the column, how much peptide is loaded. Because if you are doing quantitative proteomics, you have to rely on 1 microgram of your control and 1 microgram of the treatment. Then only you're going to look at abundance, right? So again, at one level, you have done protein quantification, which will help you to decide how much you're going to digest. Let's mean now you have taken 100 microgram of protein from each condition to digest. But in digestion, you will see there are a series of pivoting, right? TCP, you have the IA, trypsin, each is a washing step. You lose a lot of peptide. Major challenge what we see, like, you will lose almost 30 to 40%, if not more, in the process. So if you start very less, you will get less recovery. So again, these things will only come. It's a very straightforward protocol, but you have to do it. And you will fail for sure in the beginning, but 3 times when you do it, you will find it very straightforward. Just I have to be better to recover my peptides. Many of them are going solution form. Right? So these are the basic step involved. Let's now move on to the actual next talk. I look any specific burning question. If you have anything, we can interrupt. Otherwise, let's continue the talk. And tomorrow, we'll again take it from here. Let's assume now you have kept the sample for digestion. Tomorrow you have peptides, which you will inject in the mass spec. We will see the live run and also will do the analysis, right? Okay, so let's now move on to the next session, 2 more lectures. So
we have another engineering dignitary in the field of proteomics. We have Dr. Sabyasachi Babu, who is a staff scientist in Professor John Lee's lab in California, and a lab that designing and testing human and non-human primate trials. In addition, he collaborates extensively with scientists on evaluating protein turnover, protein-protein interactions, and post-translational modifications on proteins across the range of applications in protein quality control, vaccine design, subcellular biology, and animal physiology. So he has presented his work at prestigious international conferences such as Keystone Symposia, UFO Congress, and US Chapter, American Society for Mass Spectrometry, ASMS, annual meetings. Recently, he has been honored with Young Investigator Award from the Scripps for his contributions towards HIV vaccine development. So, with the round of applause, we welcome Dr. Sabyasachi.
Thank you. Thank you for the kind introduction, and thank you for having me here and for all the hospitality. If I can see my screen, can start. everything is a bit slow, but it is going to come to pace.
Just do you have C-type over here?
No, I don't have a C-type.
Can you just clear desktop? So yeah, I think let's open this one.
Yes.
Yeah.
Okay, share.
I can share this one.
Should I share desktop? Yeah.
This one.
This one.
Yeah. Yeah, entire screen. This one screen. Yes. Now I think it will. Yeah, make
Use
duplicate.
Yeah.
Yeah.
It will come.
Okay.
Yes, there you go.
Yeah. Now I will connect this one. Okay. Now it is. Yes. That's one.
Just click it. This.
Yes, so I think this is not coming here. Yeah,
No.
Okay. Is there a microphone?
Thank you. And finally, it's up there. So nice. I can check. Moves. Does it move? Now it moved. Yes, it is moving. Yes, it's moving. And good. So Sylvia has introduced me. I've come here to speak about some basics which I thought might be interesting to you, and explain some of the work which I have done in the past 3 or 4 years, where I have tried to play with whatever are the conventional ways of doing, sample preparation, and so you have been hearing how urea is being used to denature, and how there is reduction of disulfide bonds, alkylation, and trypsin usage, very specific enzyme which cleaves at arginine and glycine at their C termini. And then you go ahead and shoot out these peptides into the mass spec instruments and get your data. I'm going to tell how I have tried to play adventurous with these things, and thankfully I have succeeded in quite a few places. But it has its own process where it would work and where it might not. So going a bit back, this is my PhD. I joined Professor Peter Cook's lab, and there I was. I joined in there because Peter is this maverick who was trying to tell something different. If somebody has been following transcription, he was trying to tell that the RNA polymerase 2, which makes your transcripts RNA from DNA, the RNA polymerases, they chug along like engines on railway tracks. That's the usual, trend or thought which people have. But he was trying to suggest, no, that's not the case. RNA polymerase 2, they come together dynamically, forming what he calls a transcription factory.
and the chromatin loops through. And there are many reasons to believe that it took, I don't know how many decades to convince people. People are convinced now, but they don't like to speak about that still. But anyways, that's what attracted me to Peter's lab. But Peter offered me a different project. And so it, well, I took it on. I can call myself stupid or fortunate, but it was about even more controversial thing. Since the discovery of protein synthesis itself, there has been a controversy which is going on whether there is some translation of protein synthesis going on in the nucleus. I suppose it died a natural death in late 70s when introns were discovered. And you would think that introns are there inside the nucleus. And if ribosomes are actually translating inside the nucleus, you would have introns translated, and that's really, really bad. But over time, there were different reasons, including RNA quality control checks, which came up as arguments, and Peter's group saw that there was some protein synthesis going on because he's looking for sites of synthesis, whether it's transcription or replications and protein synthesis, and they published it in Science in 2001. I joined him in 2008 and tried to follow up the same project, which had died down because people were dead against this. And after 12 rejections, I finally got it published somewhere. Anyway, so I'm trying to make a background of how I entered proteomics and how I've been enjoying it. So you see here, this is nucleus, and this is a labeling of newly synthesized protein. I can go into details, but rather keep it short. And you see that after a 5-second pulse, this is really difficult to do. And I realized that it was very simple to do when I actually attempted it. Nobody tried this. And so at about 5 seconds, you would see that these nucleus, they are glowing up. That means there is some protein synthesis signal coming up. These are 5-second pulses because the speed of translation is 5 amino acids per second. And so if you are labeling for anything, say 20 or 30 seconds, you are going to have people question you because most of what you're seeing might not be the site of synthesis but somewhere else. But we saw that inside the nucleus at 5 seconds, and that was some of the evidence. We had more evidence come up for this, including some of this, where it was even more controversial to see that the half-life of these proteins, they might not be proteins after all, they might be just abortive translation. And whatever was made was, the half-life was less than a minute. That was too much for people to take in. So anyways, we did get published, but I was thinking, well, can I actually go and try to find these aborted peptides using. I wanted to go to a lab where they have their own mass spec, they can play with them, not just for facilities. And I joined John. I've not done that project till now because I got involved in many more interesting things, and this was much more riskier, and I needed much more time. Anyway, so I started off with John, and now I'm a staff scientist with him. I've been there for about 6 years. And John's fame comes from when he started off sometimes in the 90s when the genome project was on its way, and there were some genomes already done, partly done, and they were available for people to use. And he was thinking, well, if we can have databases and theoretical peptides and we can compare them with what we acquire on mass spec, we can most probably be able to do a much better job. And the sequest algorithm came in 1994. And as I was reading somewhere, he was struggling publishing that paper. And finally he did. And this is history, as they say, because one of the most cited papers you would see in the field.
what he went on further is to what Sanjeeva was mentioning and many others have mentioned is the lab tried to fractionate the complex mixture of peptides. So complex is a very is a keyword here because a lot of people until then were actually doing much simpler mixtures, and that's why they could do it on a mass spec. And these database-based system like what the sequest algorithm was capable of doing very complex mixture, but fractionating and getting what we call the breadth or depth was difficult to achieve. And what was done in this application is you see this is a reverse phase resin. It's usually a C18 resin, and your peptides they actually stick here by the attribute of being hydrophobic or hydrophilic. And that's the usual resin which most of us use in our proteomics approaches. But what was done is he put a strong cation exchange here and then a reverse phase. So you see, this is the part which everybody uses. What was put before that was this strong cation exchange, which is going to bind your peptides by the attribute of charge. And this is just a loading zone. So you push your peptides in aqueous solution, low pH aqueous solution onto this resin, and most of your peptides stick. And once you start doing, so that you add a bit of salt, I'm sorry. So once you have the peptide stuck and you start once you start the HPLC, you get the peptides off and you stick them here by charge, and then you add salt and packs of peptides based on what the charge specifications are will come out, and then they are going to stick here, and from here there is more of organic solvents like acetonitrile which are going to elute off the peptides. So basically there was like a two-dimensional fractionation online. When I say online, basically we are pushing in the peptides and it is going through, and here is your mass spec. So it's going in without you doing a physical process and then trying to load the peptides. It increased the depth or breadth of the number of proteins that were identified in those times. Hugely, times are changing. What the numbers that were big then are no more big now, but still. And with all these things coming, shotgun proteomics as a terminology got to be used more often, and the whole human genome was already published, and these genomes now could be used. So the idea here is that we are using a theoretical database here to search for what we are getting on the mass spectrometer. So what's not in your database, you're never going to find it. So you have to be sure that you have a database which includes what you are looking for. If you are trying to search in the wrong database, even if you have that in your sample, you don't find it and so it has its own complexity. Sometimes a lot of people say, like whether it's the streptavidin, beat, concentration, enrichment, people use database, they don't put the streptavidin or evidence sequence in their database. That basically means that so your peptides are being misassigned because there's just too much of those peptides coming from that one abundant protein. And you have told the system, the algorithm, that that protein is not there. So there can be a lot of misassignment. So you should take care that once you are using a database system to do these kind of experiments, you must make sure if you have a highly abundant protein which you are expecting in your sample, you must include that in your databases. At least it will be properly assigned. Okay, I'm shifting gears. I'm trying to get into application, and before I start the application, I want to give you a glimpse of what the biology looks like. So this is HIV envelope. So you have a virus, and many of the viruses have these lipid membrane, and there are these. So these lipid membranes are hiding everything which is inside the virus, basically hiding from the immune system. But there are these proteins sticking out. They have dual role in the sense that they are the ones who are attacking the host, as well as they are the ones which are recognized because they are easily recognized given their proteins. So this is the HIV spike protein. Spike has become very popular nowadays. So it is called envelope protein, and it's abbreviated as ENV. Many people call it just ON. And it is very highly glycosylated and glycosylated. You have heard this term in just the previous conversations. These blue bubbles you see, they are all the glycosylations. And you see that the polypeptide region, the red region is highly variable. HIV mutates very quickly and within the same patient. And then there is what is called non-conserved, that is maybe not as variable. And then there is what is called conserved, which is highly conserved. So what's happening here is the N-glycosylation is shielding these very conserved epitopes. And that means that the variable regions are more available, and that is changing. So this spike is basically not allowing itself to be recognized by the immune system. This is the side view, and you see that these lichens are dynamic. They are not static. They are just moving around, and in that movement, it's very difficult for the immune system to actually identify a target, identify the virus. And it has two parts, which is GP120 and GP41, which is the membrane-bound region. I work with a consortium, a famous consortium based at Scripps. It is called SHAWD. It's a consortium for HIV/AIDS vaccine development. And what I do is I try to analyze the glycan shield for them on the different vaccine candidates they are making. And so at this point of time, they are giving me highly purified, recombinant, highly purified proteins. Over the last decade or so, this vaccine technology in HIV has taken leaps and bounds. So they have come up with very good antigens. That's a whole different scope of talk as to how things have happened in that field. But I get immunogens which are highly capable, but there are many candidates and there is a lot of this glycosylation on them. And people want to know how the glycosylation changes and how it is affecting the vaccine design. So that's how I can give an analogy, it's like a sugar-coated like spearhead. So you are basically hiding behind the shield. And when you get the chance, you poke the host and attack. But these spears itself, if they are recognized, that's the way immune system is going to fight back. And HIV is not the only one. These all viral spike proteins, they have loads of N-glycans on them. And so the cellular process where, the spike protein is synthesized and in the endoplasmic reticulum, here, these glycans are put on, and what is put on is called high mannose containing, so much higher amount of mannoses. And then as they go through endoplasmic reticulum and into the Golgi complex, they can be processed further. All this depends on how accessible are the sites of glycosylation. It's relatively very specific, one of the few things which are very specific in biology. There are exceptions, but not it's not as well evidenced. So at these glycosylation sites, these N-glycans are being put in. So that means that if there is a glycosylation
at co-translational or post-translational level, these sites were accessible for these enzymes which are putting on the glycans. And even during the processing, it's the same thing. If something is not accessible, you can't process it further. So the amount of complex glycans compared to a high mannose glycans is telling you about a site's accessibility, that part of the protein's accessibility to the solvent and the environment outside. So it's giving you a structural aspect as well. So there are traditional glycoproteomics methods and glycomics. These are two words where glycomics means that you're just analyzing the glycans themselves, while glycoproteomics is you are also trying to see where exactly the glycan is attached to the protein. And here, the traditional glycoproteomics, it follows exactly the same protocol. You take the protein, you put a protease there, digest it, and throw it into the instrument. Now, all of us who have seen the mass spectra know that it is complex. And if you add on top of that these variable trees with different masses, and we don't know what kind of glycan tree is there. It can be any combination. It is not a static mass. So it can be any combination, and it becomes very complicated to actually find what glycans are there. So there are softwares coming up, and they have come up. There is something called Bionic, which is being used by most people. But even then, there is a lot of scope of improvement, and it's a very complicated algorithm and calculation to actually find the entire glycan structure and tell what kind of glycans are there. So what happened, Jim Paulson, who is a very famous glycobiologist based at Scripps, he was working with this group of people doing HIV vaccine design, and he turned to John Yates' lab because he had an idea of using some glycosidases to maybe simplify the methodology. So what happens is all these, so this is processing, the color is not properly visible, but what I'm trying to show is this region until this. So what happens, these circle out mannoses, depending on this situation, this particular enzyme EndoH, it can actually cleave off at this region, leaving this glignac, this blue box, one glignac. And so all those glycans which are high mannose, they will be cleaved off by this enzyme. And then there is a second step where the rest of the complex glycans, these glycans cannot be cleaved off by EndoH. But PNGS-F can actually cleave off everything. But because we have already cleaved off this portion, PNGS-F can't act on these glignacs which are left. And rather, wherever the whole trees are left, it is going to cleave off at a different place, basically remove the whole glycan and deamidate this asparagine. And so basically you can have a different mass modification marker, signature mass here for these glycans and for these glycans. Now these are static masses. And if you throw these peptides, if you are after doing this, or after making peptides and applying these enzymes, you have now peptides with just simple static mass modification, and you can do a usual proteomic pipeline search and find out where complex glycans are present and where high mannose glycans are present. So there were two publications then in 2017, and then the protocol was published again in 18. And this is a very interesting paradigm to take along. And I came in just after these things happened, and I was told that though it's a very good way to do things, it's still very cumbersome, complicated. And is there a way to, just simplify it? So before I go into how things were simplified,
I'm going to give you an overview of how proteomics is done, just assuming that there are some who would take this as new information, and I'm not trying to offend anyone who knows better than me here. So let's assume this is a protein with amino acid sequence. There are two ways we can put this sample into the instrument. One is it goes directly into the instrument and gets fragmented. And this is what we had a discussion about top-down proteomics. I will not deal with it now. What we are going to deal with is bottom-up proteomics, which is where this protein is taken and it is denatured, maybe urea, guanidinium hydrochloride. It's basically opening up the protein, helping the protease get more access. Then we are using the reducing reagent PSEP, which we were just talking about. It reduces the disulfide bonds, but they are reversible, so they can come back. And so we alkylate them. So basically put a chemical moiety where the disulfide bonds are broken so that they can't come together again. And this is when we actually digest them with trypsin. And trypsin digests after, lysines and arginines. And it creates these peptides, which are very specific because, there's a specific site of cleavage. And post-translational modifications can be present, for example, say, a hypothetical phosphorylation on this serine, and you see that that will be present on this peptide. And once these peptides are measured by the mass spectrometer at MS1 level, we further fragment it in the collision cell, and then you get fragments of this particular peptide or this particular peptide. And these fragments are the MS2 or the tandem mass spectra, which helps us sequence. And so if there is a PTM, post-translational modification here, it also gives us the scope to identify these post-translational modifications at their site of attachment. I'm going to go to the glycosylation story a bit later again, and it's one of the post-translational modification, and that is how you would identify it. Remember that there will be just one mass there as against the whole glycan tree, because we have cleaved off the glycans and just left a mass signature. Another story on the sideline, which you would like to listen to, is these peptides are using liquid chromatography. They are loaded onto these columns with C18, which is the reverse phase, or the strong cation exchange and reverse phase, which is called the mud pit. And that is where online these peptides are fractionated. If you put in all the peptides together, there is much lower chance for the mass spec, much lesser opportunity for it to, get the depth you want. So if you basically separate them into packs depending upon their properties, peptides would come out, and then there is electrospray ionization. So there is a huge voltage applied between the mass spectrometer and this tape from which your peptides dissolved in the solvents are coming out. And because as they are coming into atmospheric pressure, they are drying out, they are getting smaller and smaller. The positive charges on them are intensifying, and basically ions get into the mass spec, not just by just pushing, but rather there is a vacuum here, so it's sucking in. And once this gets into, the story begins from there on within the mass spectrometer. Oh, did I miss something? No. So, as you will realize that this is a very ideal situation where the whole protein, if I may, was completely taken care of. There was a peptide for every amino acid shown here. That's not always the case because trypsin cleaves at specific amino acids. So if it's a large stretch without any arginine and lysine, you might not have that peptide showing up. So we miss at many a time peptides, with trypsin. And said that please keep that in mind because that's what is going to be discussed later on when I'm trying to discuss how I've changed things in my sample preparation method. And once these fragments, once they are graphed on here, that is what it looks like. This is the MS. So this is the fragment of a peptide. And the database I was talking about, this database, you tell the database, hey, database, can you, in silico computation, can you use trypsin and tell me what peptides will be generated from this database? And the computation gives you that. And then you ask the computer, hey, how would the mass spectra look like? And that's how it looks like. That's a theoretical one. So what you're doing is you're comparing this with these theoretical spectra. So it's a cross correlation. It's a matching of pattern. And the better the match is, a higher scoring will be given. And these scores determine a peptide spectrum match. And that is the basis of sequest. And that is how this is one of the first computational algorithms to come up with these ideas. And then these are still the basis of many of the algorithms being used today. If you go to a proteome discoverer, the HTC Quest is based upon that. If you go to Bruker's Newcomb Pacer instrument, it's the same ProLucid search engine is based on this one. So that is how a mass spectra, the identification of peptides is done theoretically. There are many more steps to follow to do quality control, but that's the basic concept. And now I'm trying to now go back to the glycosylation problem. And here is my glycoprotein at the base, and I have dissolved it into my favorite solution, which I'm going to tell what it is. And I am imagining a super protease. One. I want just one. I don't want multiple. So when mudpit started off and the depth of proteome was a big challenge, John's lab came up with what he calls a triple digestion. So it's a combination of different proteases in a certain way. And that was a way in which he was able to increase coverage per protein. So when I say coverage, sequence coverage, it basically means how many of the amino acids have been accounted for by mass spectrometers for particular protein. So it can be a 20% sequence coverage, it could be 5% sequence coverage, it could be 80% sequence coverage. It will also depend on how long a protein is. Think about 2000 amino acid protein, think about a 200 amino acid protein, the sequence coverage could be very different. And the peptides evidencing for that sequence coverage can also be less or more. So the previous method which Jim Paulson and John Yates' lab had come up with was using this triple digestion method. And it was cumbersome just because there are too many enzymes being used. And we were always thinking, hey, if we want to increase the sensitivity of this method, there is no way we can do that. We need just too much material. Other thing is, there are just too many steps. And it used to take about 7 days. I'm going to discuss a bit in the next slide as to how difficult it was making life. But anyway, so now this is imaginative. I thought imaginative, but you have a super protease and that would digest very well. And then you have to de-kill it because a super protease, you have to take it out because if you leave it, it is most probably going to chew off everything. And then if I use the glycosidases, which I was talking about, and let's imagine that it's a day's work. And we have mass spectrometers to work the night. And guess what? With this thing, it actually was true, and you could get 99% sequence coverage. So
you get rid of all that mind-boggling, complex glycan structures and things that you have to calculate. The computation algorithms have been struggling with, and you still get data, which is what the vaccine and developers are looking for. Sensitivity increased 180 times over our previous method, and the LCMS time decreased 24 times. And ask all people who are maintaining mass spec instruments how much that matters. So just to give a comparative thing about what that existent method looked like and how much we improved upon it, is there are just multiple tube changes, buffer exchanges, and filters, and lyophilization. And so the sample loss was huge. And we had now a one tube single pot method, and we just had one lyophilization step and no buffer exchange except for just, getting the protein from whatever it was provided us in. And so these multiple proteases were gotten rid of, and I used just one, that is proteinase K. You might be, many people who are doing RNA and DNA biology might be aware of using proteinase K to remove all the proteins that you have in the sample. It's a very aggressive protease. It is supposed to, if you just leave it, it most probably cleaves everything to dipeptides, basically. But I found a way to just give it suboptimal conditions, so it creates this situation where you're still waiting for your digestion most probably until tomorrow. I get it done in 30 minutes. So 30 to 50 micrograms was starting material. People were using actually about 90. For many cases, I have come across people who have been leaders in this field, and they were talking about, people ask us for 1 milligram of protein. That's just too much to give. And I said, well, I can do that for you at 0.51 microgram. So that was a huge leap in terms of sensitivity, because there are many preparations which are in experimental stage, and they just can't get that huge amount. And then the runtime, of course, decreased hugely, as I said. And then, as you will realize, this is never a single person doing things. You can think of doing things, but you always need help. And we can't do anything without computational biologists, especially in this field, of course. So I have taken help from my colleague now, who basically created an algorithm after we quantified on our pipeline. I'll talk a bit more about quantification later on. And he created, we had what calculation goes on, we had in mind, but there was no automation. That basically means that somebody would be sitting in front of a computer looking at Excel sheets for hours together, and that's just for one sample. So all that effort which has been done here to simplify things doesn't help, because the data analysis, post-acquisition and post-quantification is just too much to handle for one sample. So he made it all simple. There is human error gone, human effort gone. I'm basically waiting for a data which I can do it in just a couple of days, and somebody is waiting for a clinical trial to get done. So that is how much a computational, simple calculation automation can help. So bench to result time went from 7 days to 2 days. While I'm absent, somebody was actually doing this protocol for me, and she was complaining, oh, this is long. I was like, oh yeah, sure. You should have tried the previous protocol. But it gives you about 95 to 99% sequence covered and all N-glycosylation sites covered. So as I go through a bit more, you will realize why this is so important, why sequence coverage is so important. In proteomics, as Kamal had been mentioning, that we all are after numbers in terms of how many proteins we have identified. But when we look at how many proteins we have identified with more than maybe 3 peptides, you will find lesser. And think about trying to cover the whole sequence of each protein. So it's just too big a thing to achieve. Said that, I'm not claiming here that a whole complex mixture, I can try to deal with this and get that kind of sequence coverage. But for a single protein where it can be really important, these are some of this approach can be very, very useful. You get a whole sequence covered almost 100%. And that's very useful because you don't know where your post-translational modification is. You might be missing out on your post-translational modifications, which are functionally very important just because you couldn't cover the whole sequence. So sensitive, quick, easy, and reproducible. So just the strategy to summarize, this has been published in 2021, and we got a methods in zymology book chapter detailing things in elaboration. And so the situation on glycosylation site is that either the glycosylation site is unoccupied, no glycans are present. It could be the complex or high mannose, which I had mentioned, and depends on the structure and number of mannoses. So here I have my protein, spike protein, dissolved in my buffer, which is actually just 100 millimolar ammonium acetate. There's no buffering. I am not adding any urea. So that's a take-home message. You add urea, you have to dissolve. You have to remove things. I don't want to remove things because that's loss prone. I have just 100 millimolar ammonium acetate. I add the T-cell, which is the reducing agent. I alkylate, and my sample is ready to go. So this is how hypothetically these glycosylation sites will be looking like, and they would have these either high mannose or complex glycans, or nothing. So high mannose, complex, unoccupied. I add NOH. What it does, it removes these glycans and leaves a plus 2O3 mass modification. And after that, I kill my proteinase K at another level of NOH. This NOH step is most probably redundant, but I still use it to maybe remove glycans on the surface so as to provide more access to proteinase K to this highly glycosylated and well-folded protein. It has a melting temperature of almost above 65 degrees Celsius always. And then I add PNGs-F, and if you add PNGs-F, it actually converts an asparagine to aspartic acid. But in terms of mass spectrometry, we can say that it's a +1. But if you do this reaction in O18 water, because when PNGs-F deamidates, it puts on an OH. And if you have O18 rather than O16, you have additional +2 mass added. So +1, +2, +3 mass added here. This is good because when you have just +1 mass added, it becomes more difficult to resolve and more difficult to be confident about that mass modification. So this is the strategy, and then it goes into LC-MS systems. And then it's from here, it's just a normal proteomic pipeline looking for a constant mass modification of either +3, 0, or +203, and we have the information we need. Okay. And so with these mass modifications, you identify. Nice. So this is what these glycosylation pattern looks like. About 30 glycosylation sites, unoccupied, complex, high mannose, and I compared it with the previous method. Looks very similar. And this is what I was trying to tell you, that just one amino acid and you have this amount of evidence. So these are the number of peptides that I have identified. And that means your confidence in terms of that PTM being actually present there increases hugely because your sampling is huge. And so the statistical confidence in your identification is huge. Again, it gives you the sampling to actually make proportions. So because that means that even a small amount of something else can be accounted for. And so your proportions will be very confident. And that is how the proportions would look like. And you can actually per peptide graphs would look like this. So you have very high confidence on your data. I'm going to skip some things because this is Salva who designed the downstream analytical platform. And this is Jolene who helps us with all the MS runs. So nothing would have been possible without these two guys. I'm not going to go through the summary again, but this makes things much easier. And the result is that about 100 diverse immunogens in over a year, about 6 immunogens a month. That includes HIV, SARS-CoV-2, influenza, Ebola in single or multimeric forms as nanoparticles. I have done a study where all the SARS-CoV-2 spikes for different variants have been accounted for with Andrew Watt's lab. And I could find that there were specific glycosylation changes at, from complex to high mannose at these crucial, mechanistically crucial sites here at 165, 343, and N616. I'm going to give some basics which I might have already given you during some of your sessions this afternoon when you were there for practice. And so this is your usual MS1 spectra, and you will have your isotopes, naturally abundant isotopes, say C12 is about 99%, but there can be C13, which is 1%. So when you see your peptide being measured, M over Z, you can have these isotopic peaks, different isotopic peaks, depending upon whether your peptides have one isotope or the other. And usually you want to simplify your life by taking the monoisotopic peak, which is basically the peak that constitutes only the most abundant. It is not always the highest peak because with one peptide, there are hundreds of carbon atoms, maybe, or hundreds of hydrogen atoms. And that means that there is always a chance of the less abundant atoms being there. So said that, that's how on an M over Z MS1 graph, the difference between your isotopic peaks would tell you the charge. So there is 549.1, 549.6, the difference is 0.5. So M over charge 2 is going to give you a 0.5. So that's how you determine the Z. The mass spec always gives you mass over charge. And that's a platform saying how all the peptides get in when they are pushing through the LC system, and you get an MS1 graph. And the MS2 graph is generated by selecting one of those most abundant ones, going into a collision chamber where it collides with a lot of gas like nitrogen, and then it gets fragmented because of the crowding, generating these Y and B ions that you see as an MS over MS graph. And that is cross-correlated with a theoretical thing coming from the database. And that's how you sequence your peptides. And the quantitation which I used for my glycosylation is the label-free quantitation. It can come in two flavors. You can either take a spectral counting, which is the MS2 level. You want to check how many, so these are two experiments, A and B, how many spectral counts you got for, say, the same peptide in two different experiments. So same peptide, two different experiments, how much you gain. And there is the other one, which is supposedly more accurate, than which I use, is area under the curve. So this is the MS1 level. So A and B experiments, same peptide, what is the peak area? And once you define what is a peak, you can actually do a quantitation. And that is how it looks like. It's a retention time on this axis, and you will have different in an M over Z axis here, you get different abundant peaks.
and that is how your chromatogram comes from. That's how your chromatogram comes from. That's how your MS1, looks like, and that is where that's how you get from detection time to MS1 graphs and to MS2 graphs, and this is how the label-free quantitation is done. And the other way which Sanjeeva was talking this morning is using labeling, which is say tandem mass tags. These are isobaric. Isobaric means these tags, if they are attached to a peptide, those peptides are going to weigh the same irrespective of which tag is attached. There are say a choice of 2 tags, 6 tags, 11 tags, 10 tags, 16, and now 18. So 18 tags, all the peptide, a same peptide will have the same mass whether it attaches to tag 1, 2, 3, 4, or until 18. And what happens is only when they fragment do you see the reporter ions and the reporter ions intensity. So there is a mass normalizer which makes sure that all of them are the same mass, and this is a reporter, and this is the mass which becomes different. Only after fragmentation do you get these reporter ions. They are different in masses, and their intensity is going to tell you what is the abundance of that particular peptide. So that's how labeled experiments work. The advantage is, of course, that you can multiplex your experiments now. There were 2 conditions in the previous thing. There can be more, but that basically means that too many experiments. And here, all of them are done. They are all labeled with different labels and then pulled together. And you run just one. But there is a clause. There are too many peptides, so we usually fractionate them under high pH and make say about 8 fractions or sometimes even more. There is a kit available, and then push them into these mass spectrometers, and so you have a huge depth, and at the same time you have a labeled quantification, and you just had one run. That basically means that you normalized lots of things, and for the idea that why you should be taking account of what amount of protein you are starting with, you have to mix them all the same amount of protein to make sure that you start with 1:1 in total protein amount so that you can actually quantitate the differences. So I'm just showing you an example. I mean, this is unpublished data. This is going for a grant or something. And you see that, there were different conditions here. This is a whole mouse experiment. Sham means that nothing was done to this fellow. There is a transgenic and wild type. So there is being a condition created and a transgenic mice is created so that that condition can be rescued. And so that is the denervation. That's the condition created. A muscle is denervated. And the comparison is between transgenic and wild type. Then there is a wild type and there is a denervation versus a control. And you see that these are volcano plots. And this particular line tells you everything above it is statistically significant. And here it tells you about fold change. And we have used the false discovery rate here, which is another level of stringency, and only those things which are above that FDR, false discovery rate, 5% false discovery rate you see in bright red here or bright blue here. So depending on how stringent you want to be, these volcano plots, these are basically fold changes. So we are comparing between 2 conditions, and this is the whole proteome. And I did the same thing by enriching for the same sample, but I enriched it for phosphorylation. So if you take just the whole lysate, here and look for phosphorylation, they are like a needle in haystack. So you use enrichment columns. Kits are available, whether titanium dioxide or nickel NTA, and you enrich for them, and you do the same analysis. You have to start with quite a sufficient amount of material to be able to do these kind of experiments. And guess what? Most of these candidates which you get, they are expectedly in
the mitochondrial oxidation system, which explains or validates the hypothesis which these collaborators are working with. So these are kind of data you can generate very accurate quantitation and fold changes. Here, even a small fold change can be confidently commented upon, because in a label-free experiment, these small changes might not be, you might not be that confident. But here, given that you can apply the statistics and say that there is a false discovery rate cutoff, there is a statistical significance cutoff, these might be very bright candidates for further evaluation. Without further evaluation, and they will still be a hypothesis. Oops. And I would like to thank all those involved. What does it say? All those involved here. John, of course, for all the support. Jim has been a constant support. Jim Paulson, all through. And so Bill Schiff, he is now with Moderna. He is one of the prominent fellows who has been designing different HIV vaccines. Andrew Ward for the SARS-CoV-2 project. And Brooker, who have been very helpful with the IP2 pipeline. They have bought our pipeline, Proteomics pipeline now. And for the phosphoproteomics UC San Diego collaborators, these are the people who make this all possible with their money. Thank you. I'm happy to take questions, and I'm sorry I think I went with myself.
Maybe, another speaker is waiting from New York early morning, so we can take the questions at the end of this session. Let's thank once again Dr. Sattarshi.
Thank you, Dr. Sattarshi, for
Thank you so much.
Informative session. Please accept this token of
Oh,
gratitude.
thank you so much.
So, last talk we have Dr. online, and he is going to talk about some more sample preparation strategies. Dr. John Wilson did his PhD in biological sciences with an emphasis in chemical biology and microbial pathogenesis from the University.
Hi guys, I don't know if you can hear me. Hello? Hello, I can't hear. Hi, I hear something now. Can you guys hear me?
Yeah, we can hear you, sir.
Oh, excellent. Hello, everyone.
Yeah. So I think you can share your screen.
I certainly can. Good evening to you guys, and it's the morning here. Can everybody see this?
Yes, sir. We can hear. We can hear, and it's the slides are visible.
Excellent. So I'm going to set a timer. I'll try to stick to exactly 30 minutes. I will start with a teeny bit of background on the company and me, only to put people in context. Is there a way that I can see the audience while presenting? I guess not. That's all right. If you got any questions, guys, please jump in, and I really do mean that. So Protify is a company that was started out of Cold Spring Harbor Laboratory, and we're scientists. So my co-founder Daryl is one of the 4 founders of Proteomics, and he invented isobaric labeling and mascot. And he's a really unusual guy because he went back and forth between academia and industry. And giving you a little background here, because if you really want to make an impact, you got to think, what problems can I solve so that I make the most bang for the buck? And we'll get to that. So we chose to do things that are impactful for the community. About myself, I did a bunch of undergrad degrees. I've been doing protein analysis since I was 17, did 2 dissertations as an undergrad in proteomics, dissertation at the Rockefeller University in proteomics, and so I've been doing that for a long time. The goal of these technologies, and I'll get into S-trapping very shortly here, is that we'd like to make this thing. Does anybody know what this is here? I'm unable to hear anybody. Is it silenced again?
I think the most of the answers are no.
Ah, okay, thank you. So this device is a fictitious device out of a science fiction program called Star Trek, and it would scan somebody, and it would just go doo doo doo, and it would tell you, it's science fiction, right? So it would tell you what's ailing the person, or what alien parasite they got. And that's really the goal, right? To take these technologies and be able to do something like early detection or clinical chemistry replacement for those things that we do right now. And to start, you have to work at the very beginning, right? So in sample prep, and you guys know this, we start with this beautifully organized cell, and the first thing we do is destroy it, right? So we lyse it, and then we make our lives even more complicated by digesting into peptides, and we're attempting to reassemble this back into the whole to get back to this beautiful beginning. Now, there's technical reasons for that, but it's problematic, very problematic, in fact. And when you look at the overall variability in proteomics, the vast majority comes from sample prep. And that's actually pretty understandable, because we have water-soluble proteins, mostly, and lipid-soluble proteins, mostly. And the physiochemical properties of those are very, very different. So how do you get one protocol that handles all of the different proteins of our body, or whatever? And I want to highlight that the stuff that we spin out as a pellet, it's not uninteresting. It's just not very soluble, right? So you can think about the kitchen. If you put a piece of meat in water, it doesn't dissolve. If you put a bone in water, it doesn't dissolve. And yet our muscles and bones are extremely important to our ability to live. So we have to keep in mind that just because something is not particularly soluble, it doesn't mean it's unimportant. And actually, that was my graduate work. I discovered that histones are lipidated. And the reason people hadn't found that is they spun out their pellet and then threw it out. And when you hang on lipids to something, even something very soluble like a histone, it goes away. The importance of solubility and just the very beginning of the process cannot be underestimated. So in pancreas, if you use a buffer like ammonium bicarbonate and not a detergent like STS, you actually get 0 membrane proteins. I redid this. This is work that is from my own hands, and I thought, wait a minute, this can't be. But no, it's real. And the point is that this is a very significant hole in biology. Obviously, all nutrition crosses membranes, all signals cross membranes. It's just extraordinarily important. In the brain, it fell by half, and in the pancreas, I was just utterly shocked. And just keep that in mind. So the purpose of this experiment was not a tremendous number of proteins. It was to say, as a function of lysis technique, what were the effects on cytoplasmic membrane and nucleolar proteins? And you can see that everything went down, but membrane went to 0. So just keep this in mind, guys. Membranes are very important. Membrane proteins are very important. And if you don't do it right, you will see 0. What Protify has work to do, and we're going to zoom in on the S-trap and sample prep, is to create solutions for this whole range of the process. So, you start somewhere with, could be a blood draw, could be a biopsy, could be an FFPE piece, formalin fixed, paraffin embedded. I'll get to that in a second. And you got to do all of these different steps to get somehow to meaning. And there is no ground truth and no harmonization. And what we set about to do is to fix it. Because if one lab uses RIPA and another uses tween and another uses urea, those three answers, coming back to this sort of thing, look like three different cell lines. And we can't have that if we want to actually take something, take something like this and make a clinical impact. So the S-trap. Give me a rough percentage about how many people have used an S-trap in this class. Any? Can't hear anything. Well, regardless, the S-trap is at its core a balance, or a bridge, I should say, between the fact that STS, sodium dodecyl sulfate, which is used in SVS page, it works really, really well, and it's completely incompatible with a mass spectrometer. So what the S-trap does, in essence, is allows us to use STS to handle and solubilize and manipulate proteins, but then get rid of it so that we can go onto a mass spectrometer. It is a plate, either 96 well or 384 well, that comes in various sizes as spin columns as well. These are functionally equivalent and simply allow us to work with different quantities of protein from micrograms, even submicrograms, up to milligrams. And STS, in addition to being a very strong detergent that solubilizes membrane proteins and everything else, has a unique ability, which is it coats all of your tubes, not your small quantity of protein. And it allows you to then handle these things in a way that is reproducible, that's one protocol, and that's KISS, you know, keep it simple. Does anybody have any questions up to this point?
Is there any question? No, John, you can.
Awesome. I'm just looking at my timer here. Okay, so the way the S-trap works is you start with a sample, which you've dissolved in STS. To give you an idea about how powerful STS is as a protein solvent, you can take 5% STS and put a mouse heart, a whole mouse heart in 5% STS, and with sonic energy, you can get a complete liquid with no pellet. So that gives you an idea of how really amazingly powerful this is. You can do the same thing with bone once you've demineralized it. And so now we have access to, well, I just talked about bones and muscle. We have access to those things. So you dissolve your protein. You reduce and alkylate here, and you want to do it in the presence of STS because things precipitate in the lack of STS when you manipulate them with reduction alkylation. You then add a binding buffer. You acidify it. You put it on the column. Well, you acidify, then you add the binding buffer. You put it on the column, then you wash it. The protein is trapped on the column. This is a spin column, but it might as well be a plate. And it's fully denatured here. Talking about the denaturation, SDS is denaturing, reduction and alkylation is denaturing. The acid that comes after it, dropping the pH to 1, is denaturing. And the binding buffer is actually methanolic. So we have 4 separate mechanisms of really tweaking the protein structure. And that's very important because when you really turn a protein inside out, and this has been designed to turn all proteins inside out, which is why there's no less than 4 separate mechanisms, you will allow a protease, trypsin or whatever, to get in there and get to the interior portions of a protein. You actually get much better sequence coverage, even from something like serum, because now the protease has access to all of those areas of the protein that otherwise would have been on the interior or somehow sterically hindered or otherwise hindered. So on the column, you have the protease. You digest it. You really don't need more than an hour at 47. And then you get your digested peptides. So there's a lot of people S-trapping. 1,320 results on Google Scholar as of yesterday. And I don't want to belabor this because it works really well. This conclusion has been found time and time again, and this is on urine. And why does it do that? This is on urine, and there are other techniques that you can compare to. And in general, this is what people find. There are people who spoof it, and I know how they spoof it, and they're like, oh, our thing works better. All right, whatever. In general, and I'll just give you these slides briefly here. The S-trap system is robust, it's reproducible, and it's pretty rapid. And those are the attributes that we need to have so that we can really advance this field. And what it means is you don't require a lot of training. You don't require optimization of everything, which really slows us down. And you can actually have a throughput in the lab that allows you to make progress in your biological question rather than just figuring out what went wrong. So the Venn diagram is the biggest, and that's all there is to it. So here's the conclusion on urine, outperform these other things. Not only reproducible, but also can be applied to perform high throughput proteomics as for urine using a 96-well plate. Workflow can be implemented for future clinical studies. That's pretty much what people find. This is my buddy Yashai, and this is a while ago, and he gets 1,200 proteins from 500 cells. This is now going to go. This is going down and continues to go down. And again, this is an unoptimized thing. He just threw 500 cells in this thing and got really good results. This is my buddy Yiming at Impossible Foods, actually. And I think I got some other slides here. Let's see. This is the most important one. So Impossible Foods is a very interesting use case because it's food. So they're not sample limited, but they have difficult things. It's, take something like tofu and try to dissolve it. And again, you're not going to get it to dissolve very well. The key is that in his comparisons, using proteins of disparate sources from disparate organisms that are of agricultural significance, we had the largest scores and the largest number of peptide counts. And I mean, I don't really want to belabor this because this is what people have found. Like I said, there are people who disagree with these conclusions. That's okay, but there's a reason we have that Google Scholar. So best performance with largest number of protein IDs with reproducibility generates additional samples within minutes. I mean, it is what it is. There's another interesting study down here, and it's milk fat globule membranes. So once again, membranes, right? Again, applied to food products. I didn't intentionally bias it in this direction, but it's very, very interesting because these are things which, again, they impact our health. And again, we're talking about membranes. And when you compare it to a whole bunch of different other techniques, we've got the largest number of proteins and peptides. And again, this is kind of boring. Are there any questions up to this point?
Is there any question? No, John.
Super cool. So who knows what FFP is?
FFP samples.
So like 5%, 10%, 20%? How many people know FFP?
I think 5%.
5%. Oh no, we got to teach you guys. All right, cool. So FFPE means formalin fixed paraffin embedded. And this is super important, guys, because this is the kind of archive you have for formalin fixed paraffin embedded samples. And this is what happens when a pathologist takes a sample of a person, a biopsy. This is what happens. So actually, I will come to this. This is what happens. Well, it's the reverse of this, actually. Because this is the undoing of it. So let's work backwards. We have a piece of tissue. What we do is we put it in formalin, which is the F. Why formalin? Because it's a cross-linker. It is literally molecular staples all over the tissue. Why do you do that? Well, think of something like, I don't know, a piece of intestine or whatever. It's gooey and wiggly. How are you going to actually make a slide out of a gooey, wiggly piece? Like, it's not going to happen. If you freeze it and you try to slice, it's just going to crack. So we've got to staple it together, which is cross-linking. And that's the fixed part. So formalin fixed. Then now you've got a gooey thing turned to a more rigid thing, but it's sitting in an aqueous solution. What a pathologist wants to do is take this sample of a human and make a very thin slice of it and then put it on a slide. So it needs some support. So we're going to add wax, paraffin, to these samples, but we're actually going to replace all of the water with wax. And so now we'll come to this. So we're going to work in reverse. So you take your sample and you put it in a dehydration media like ethanol or something like that, and you slowly, again, this is working backwards, you slowly go back and back and back until the thing is dehydrated, and then you add paraffin. Now this is going in the opposite direction, because once we have this chunk, it's very stable. It is cross-linked, there's no water in it, and it's room temperature stable. So these archives are massive. I mean massive. Like think of a soccer stadium, that kind of size. And these are each drawers filled with all of these hundreds of samples. And in each of these is like, I don't know, 10 of these little blocks. So because this is such a rich resource, because often these people have associated medical records that are indicative of a course of disease or the efficacy of a treatment or something like that, we really want to be able to tap into it. And paraffin is not compatible with a mass spectrometer, and cross-linking everywhere is not compatible with a mass spectrometer. So currently what people have done, well, now they use our stuff, but in the past, before our stuff, you would get rid of the paraffin with xylene, then you would remove the xylene with ethanol, and again, this is the reverse of how you add paraffin. Then you make it more and more aqueous, and eventually you get to a paraffin sample, and then somehow you decross-link and digest, and somehow, or sorry, decross-link, somehow you clean it up, somehow you digest, and then you're going to clean up this digestion as well. There has been no consensus of what to do, and this mirrors exactly what happens in general sample prep. Do you use urea? Do you use quantity? Do you use detergents? Do you know something else? So our goal from the beginning to the end has been this harmonization. I want every one of you guys, without a bunch of training, without a bunch of headaches, without a bunch of diagnosis, to be able to take a sample and take a kit and put them together and get good results. And that's what we got here. So limiting ourselves to FFPE, we did the one thing that should have been done and wasn't done to my knowledge outside literature. We took a sample of a human, a piece of liver, and we chopped it in half. And half of it went into liquid nitrogen, and the other half went through the entire FFP embedding process. And this is all automated, as it has to be for clinical stuff. Then we compared various techniques of taking the FFPE thing and undoing it, trying to match the fresh or flash frozen exactly. So we want this thing to look like this thing. And in the end, we developed a program called Hypersol, which is basically use SDS, sonicate and heat, and use an S-trap. And the results have been pretty astonishing. And I want you guys to remember this. So S-trap in general is just a really robust sample prep system, and people use it all over the world for all kinds of things. The most interesting sample I heard yesterday was sea urchin spines, right? That's an unusual sample. And if you did not have the S-trap, if that lab didn't have the S-trap, they would have had to do a whole set of protocol optimizations to be able to handle this. Rather than that, which could take weeks or months, then they just dropped it in S-trap, and they're proceeding very well. So in the same way for FFPE, we want you guys to be able to access this, because these are incredible resources, hundreds of millions of samples, and room temperature stable. I'm going to reiterate that. So FFPE, you guys got to know about that. It's an incredible sample source, and we need to be able to handle it. So point number one about Hypersol, our protocol to get flash frozen results out of FFPE. The yield is huge, right? So this is all standard techniques, and we're getting like, I don't know, 2 to 3x, something like that back, which of course allows you to do things with samples like laser capture microdissection. As a different illustration of that, we get 200%. Most importantly, for something that has been through the process of cross-linking and then embedding in paraffin and then undoing all of that, our ID rates are virtually identical for peptides and proteins. And arguably just as important, maybe even more important, the correlations that we have between quantifications of this Hypersol protocol, where the sample has been through all of these manipulations, and flash frozen tissue is it's 0.9 something. I think the average was 0.936, and that's just, it's the best that's in the literature. Which means now we can apply our techniques to actual clinical questions. And I'm spending time on this because this really is an incredible source of information, well, source of sample to get information. And we wanted to actually demonstrate that this works. So these are 3 different cancers which are histological mimics. So they look identical in the microscope. And these are actually corresponding to malignant peripheral nerve sheath tumor, synovial sarcoma, and metastatic melanoma. And they look the same. So while they look the same, their treatments can be different. And so we would really like a set of biomarkers that a pathologist can use on a slide to distinguish them. So we needed biomarkers. We did this protocol, and we discovered them. We discovered not only proteins that are known as biomarkers, like S100, but entirely new proteins. And so coming back to the overall strategy, you start with the FFP sample, you solubilize it, you decross-link it, and then you S-trap it. So in essence, this is the same protocol as the S-trap, just with the decross-linking step using thermal decross-linking. And you get really, really good results. And again, this is the intent. You dissolve an SDS, you don't need xylene and toxic stuff. You get quantifications IDs that are nearly identical. This can only happen with a system that you don't have to tweak. So with regards to FFP, and this is extensible to all samples, because we now have control over our sample prep, we know when a sample is not good versus when we have an error in sample preparation. And this has been very, very difficult to disentangle, coming back to that original pie chart. So our conclusion was we anticipate this workflow will assist usher in a new area of clinical proteomics because we, again, have hundreds of millions of samples, and I'll stand by that. So questions to Hypersol, first of all.
Any question regarding a hypersol?
Or anything else. Guys, you're killing me. I'm just like pontificating.
So John, we'll take all the questions at the end. So
okay, cool. So new developments in the S-trap processing system. And once again, the S-trap is this. You solubilize, reduce alkylate, denature, bind, wash, and digest. And that's it. Like, that's it. Oh, I'll add one thing. This is a solid phase cartridge, right? So there are people who use beads, there are people who do other things. Regardless of how you do it or what system, the degrees of freedom in a solid phase system is vastly reduced. And that's really, really important because, again, we want to kiss it, keep it simple. As few variables, as few points where things could go awry, that's exactly what we're looking for. So our standard protocol, and you guys know this, is you have a sample, you ice it. We like the Pixel, which is a megasonicator. You do a protein assay, you prepare it, and you concentrate it. We have done our best to eliminate all of these steps that we can. And the new S-trap Turbo takes this derivatization density and increases it dramatically, which means our trap, which is physically like you can see it right in the products that people buy right now, it turns into this much, much thinner thing. It's like 20 times less volume. And that's the key, because rather than preparing a sample and eluting it, and then you have to concentrate on a speed vac, you just elute and shoot, because this volume is so much less. You just take this, elute it, and that's your digestion, meaning your elution. And what we found is that from 1 to 100 micrograms, we get about 80 to 90% off in the first fraction. You can get another, I don't know, 20, 10%, something like that, in further elutions. And this is pretty stable. So what you can do is simply, for things that are unlimited, not sample limited, not unlimited, but not sample limited, serum, for example, or when you have enough tissue, you use an S-trap Turbo, which eliminates this concentration step and saves you, it might be hours or it might be overnight. And speed vacuuming is notorious for having some tube that doesn't go down, and I don't know why still, but we eliminate that. And let's come back to this graph here, protein assay. So typically we use BCA, which is why it's these colors here, because it's compatible with SDS. And I'd like to eliminate that as well. So a couple years ago now, I was I'm not supposed to be in the lab, but I was in the lab because everything was failing. I mean, everything was failing. The turbo pump failed, the plate reader failed, the vacuum pump on the speed vac failed, like everything was failing. So everybody's working late, like we're sleeping in lab. And of course it's before ASMS, right? So suddenly I realized, I was like, well, you know, every column, think about organic chemistry or whatever, it loads from the top, right? So there's binding sites at the top of a column, and as you saturate those, it goes down further, and that's true of C18 or an S-trap, right? I was like, well, you know, the problem with all these protein assays, including fluorescence, is that you've got contaminants. But the S-trap, it eliminates all those contaminants. So proteins fluoresce in the UV when excited in the UV with tyrosine, tryptophan, and phenylalanine residues. I thought, you know, well, what if we excite from the top and measure emission from the top? And lo and behold, it works. This is a protein assay. So the amount of fluorescence is proportionate to the amount of protein. And we would expect a hook because as these residues get further and further into the trap, the UV doesn't get in. And if it does get in, it can't get out. So we expect a hook, and that's exactly what we observe. And go ahead.
Sorry to interrupt. 5 minutes more you have.
Yep, I've got a timer here. Thank you. So, so in essence, this is a direct inline quantification that is non-destructive, and it works really well. It is equivalent to a BSA assay, but rather than doing this whole series of manipulations and colorimetric determination, you just take a plate, wherever a plate is, or you take a plate and you put it on a plate reader. So this protocol, this is a tube, but it could be a plate. This is unchanged, other than right before you digest, you do a protein assay, and for free, you get an exact quantification of your protein. And this is really, really important because rather than at the end trying to manipulate and mix 2.5 microliters with 17.5 and whatever to match concentrations, you simply adjust your auto-inject volume. So once again, coming back to this, and this is really key, guys, the more steps you can eliminate in any process, the simpler you can make it, the more robust it is and the less variable it is. And that's exactly what we've been working for. Now, developments that are under wraps is the side trap, where you have a sample and you get all the different classes of biomolecules, including lipids and metabolites and DNA and RNA and glycans and peptides. So you'll see that coming out. And we have a new device that takes a protein and just chops it up everywhere, giving you a complete protein coverage. So BSA is a very standard model protein, and normally you get 10, 20, 30 peptides, whatever. But in this case, you get over 2,000 peptides from this one protein, which means that the entire sequence is covered. And that's very important if you want to do de novo sequencing. For pharma, when you guys get into it, we have a kit for MAM, which is multi-attribute method or multi-attribute monitoring. It is LC-MS proteomics applied to biological drugs. And we've got two other things that might be of interest. Trypin is a protease that cuts not after lysine or arginine, but before it, which means all of your positive centers, so the basic center of lysine or arginine and the N-terminus, they're all on the left-hand side of a peptide. So when you break this, all of the ion current wants to stay on one side, which means rather than getting fragments over here, because there's a plus, and fragments over here, because there's a plus, you get just B ions. This is useful because you get an increase in sensitivity. And the reason for that is in a peptide like this, when you have a tryptic peptide, this plus, well, this one, is over here. And when you break it, half of your ions are on the N-terminal side and half are on the C-terminal side. But with trypin, when you break it, you don't lose any of your ions. So you get an increase in sensitivity. You also get an incredible simplification of the spectrum. And we have software that I don't have time to get into, but very shortly, Simplify is as easy to use as Google Flights, and I can show you that later. And it's robust. So I'm going to skip all these, and I'll just continue this. So the S-trap has become very, very popular. It's a bridge between get proteins out of a tissue or whatever and put it into the mass spectrometer with minimum variability, with no protocol development, maximum efficacy and speed. BCA no more is inline, non-destructive, works on an S-trap. The S-trap turbo allows you to eliminate that step of concentration. So again, we're eliminating as many steps as we can. The shredder is something that you use if you need full protein coverage, which cuts everywhere. So you've got all of these overlapping peptides. Trypin is an N-terminal protease that cuts in front of lysine arginine, which gives you an increase in sensitivity as well as a simplified spectrum. MAM is LC-MS proteomics applied to biological drugs and pharma. For those who go get a job, you will eventually run into this. So just keep that in mind, and we got a kit. The key is the S-trap gets rid of not only SDS, but salt and PEG and buffer and all of the other things that can contaminate your mass spec. All of that is in biological drugs. The SciTrap is sample prep for multiomics. Simplify is our tool that allows us to interpret all of these data or any other data. It takes transcriptomics, lipidomics, metabolomics, glycomics, etc. We, in our 384-well plate, are able to reduce the time per sample to minutes per sample. And now the LC, your analyzer becomes limiting. This allows us to do thousands of samples per day, and we have this automated on a TCAN system. And the end game is to put this in actual settings where it connects with people, meaning personalized medicine, precision health, whatever you want to say. We had to develop these solutions because if two labs can't get the same answer, how are you ever going to get this in the clinic? And that's what this is about. So the goal is truly to get to this science fiction point where we have a scanner. And I just want to thank my co-founder, Daryl.
So we have two questions.
Yeah.
Will you tell? Yeah.
Yeah, no, these are the guys I thank, that's all.
Yeah, hi, John. Yeah, nice talk. So I just have this query regarding the use of the FFP samples, right? Have you had the chance to test how well this works across FFP samples around the world? Because the quality of FFP is not consistent across the world. I mean, there would be certain places where the quality is really low. So would that not affect the overall efficiency of your columns? Have you had the chance to test that?
So we had the chance to test age of samples, and up to over 10 years, I think it was 17 or something, it had no degradation of performance. We were able to identify a misdiagnosis because the sample prep was good enough to ascertain that this individual was not corresponding to the diagnosis. We haven't done a worldwide study to directly answer your question, and that would be very interesting to do. And the nice thing about FFP is, if it makes it into formalin without a lot of time between there, that kills the enzymatic activity. So yes, there are differences, but as long as it gets into that formalin in time, you probably are going to have results that are at least going to give you some insight.
Okay. Yeah, and also, would you recommend any a certain modification to be searched for in case of FFP tissues?
The only modification necessary to the protocol is you should read the paper. This is all published. Just search for Wilson hypersol and you'll find it. But Dr. Wojcik did the hands-on stuff. I didn't touch it.
Okay.
So the only modification required is heat to decross-link.
Okay, got it. Yeah. Thank you.
So, Dr. Wilson, can you hear me?
Yes, I can.
This is yeah. So I have used STRAP. It is a wonderful thing, but I have few questions related to it. So number one is like the STRAP binds to all proteins. My question is in terms of selectivity and specificity. I want something to be enriched, like some peptides to be enriched, but in general, it is binding to every bit of the peptide. So is there any modification in which STRAP can help us to enrich some certain sections of the peptides? And number two, if we are traveling to remote areas, okay, and where we want to do something rapid for the proteomic applications or proteomic explanation, STRAP needs a centrifuge. Like, can we?
The audio just cut out. I heard can we?
Yeah. So these two are my questions.
The audio cut out after can we. Could you repeat what happened or what you said after can we?
So can we use SRAP in such a way that it does not require a centrifuge?
Yes, you can. You can do positive pressure or negative pressure. In fact, you can use even a centrifuge. Sorry, a syringe. It goes slow. It's one by one, but you do not need a centrifuge. All you need to do is make sure that the various solutions go from top to bottom, and as long as you make that happen, it doesn't matter how. In fact, if I go do samples myself, I'm always in a hurry. So for micros, I put them in a vacuum manifold. And for washing, I don't even measure. I take a Falcon tube of binding buffer. I just pour it, pour. I go around 3 times. And that uses vacuum to transmit the sample through the column. And it works very quickly. So you can use anything that manages to get solutions from the top through the column.
And do we have some special version of STRAP like which can go for only something else and something else? Because STRAP is specific to all the peptides.
So you'd like to enrich specific peptides from your digestion?
Correct. Initially, so for STRAP is unbiased, correct? It will take all the peptides. It will bind to all the peptides. If I
No, it doesn't bind. Hold on. This is very important. An S-trap binds denatured, undigested protein. It doesn't bind peptides, and that's essential because the whole point was to trap proteins, clean them all the garbage, including SDS, and then let the peptides go. So peptides are supposed to not bind, and they don't bind. There are ways to
enrichment can be done. So if I'm interested in specific set of proteins or peptides.
So the S-trap itself is not designed for enrichment. You can do everything after that, like IMAC or whatever. But the S-trap itself is really designed to take proteins, clean them, and turn them into peptides. So all of those steps would either be before you put it on the column or after you put it on the column. And Sanjeeva's there probably saying it's time to go.
No, I just thought to come and say thanks to you, John, for the wonderful lecture. I'm sure, Argyo has many points to discuss, which we can discuss offline as well, given that session is quite late now. It's wonderful, to use S-trap. We have had good experience using in our own lab. In this workshop, we tried participants to give multiple options, and what you realize that we are giving you different ways of sample processing. And all best practices that you can choose, depending on your context, what can be worked out well. Many difficult sample like FFP, what John was talking, when we had experience, many times other protocol fails, then you have to try out what can work best for you, right? So in this, it's good to be open, and we are trying to expose you with all best possibilities in this workshop. So thank you very much, John, for giving the talk early morning in New York.
Thank you for the
John has
opportunity.
already been to IIT Bombay earlier to one of our workshops, and it was very wonderful session with him. People have actually brought all kind of samples themselves. We asked participants to bring whatever you want to do protein extraction. Somebody brought plant, somebody fungus, somebody tissue, and we tried all of those samples in that workshop. So we had an amazing time with John. So we look forward to see you sometime soon. John and IIT Bombay campus, and thanks for giving the talk. Thank you.
I really look forward to it. Thank you, everybody. And the last note I'll leave you with is this was stress tested by no less than my 8-year-old child at that time. And they got good results. So again, this is designed to be simple. And thank you so much for the opportunity to present to you guys. Please send any questions my way.
Thank you. Bye-bye. All right, so we are almost there to close the session, but before that, we'll take 5 more minutes to do one more guest and
Thank you. So, 7 years ago,
I stood in this very room, well, not exactly 7 years ago. And that occurred because of a mission set up by UK Foreign and Commonwealth Office from the UK and to India. And at that meeting, MS. And as a result of that, a lot of positive things have happened. And it's my great pleasure that we've got Abhishek Jha from the Foreign and Commonwealth, UK Foreign and Commonwealth Office, who's just going to really, I think, say a few words of support and thanks. So, thank you.
We would like to invite Abhishek to talk to the participants, but also after this we have the IT and snacks for all of you. You can interact with him more. Idea is to provide all of you, further opportunities and see if something works out for further exchange within the UK program. So, what do you?
Thank you, sir. Good evening, everyone. Thank you once again for joining the workshop. It's a great pleasure to be back in IIT Bombay. I remember my last visit was in 2015 when we brought Minister Maud, who used to be heading trade and investment at that point of time in the UK government. So it's always a pleasure to be back at one of the top center of excellence on technology in the country. Just want to say thank you, sir, once again for making this bridge of collaboration between UK and India really strong. And if there is anything that can be done from our side at the ministry, at the embassy, please do let us know. And we'll be very happy to collaborate on any areas of collaboration which you might have ideas on with UK. So please feel free to discuss that with me, or anything that we can do to support that from our end. We'll be happy to do that. So thank you once again.
A very quick question for you. Earlier we had written about fellowship, which helped many of the Indian students to go to UK.
Yeah.
Including two of my friends have very positive experience of part of their PhD they pursued there. I just heard that the program got,
Sure.
Yeah. So is there any other scheme you have, where any student can go and exchange programs with you?
So in fact, today, 25th of July, is a launch of our India-UK professional bridge course. You guys can go online and see that, which is kind of a 2-year program for Indian students to go to UK and to enroll in professional courses over there. So that is something to just look out and watch out. I'll share the details with Sir so that you can circulate it in the community. So that program actually is getting launched today. There are some more initiatives which we'll be launching in the forthcoming months. One of them is the Global Talent Network, which is a GTN. So in UK, we are just trying to look for the best of talent across the world to come and work over there. So Global Talent Network is another one, which is a forthcoming one. We have something with IIT Kharagpur where we are doing a collaboration with University of Manchester for PhD programs. So that's a joint PhD in which you do 2.5 years in India and then 2.5 years back in Europe. So we'll be launching similar more initiatives. We are in talks with various institutions. So we'll be just bringing back those details to you and happy to even explore more opportunities with IIT.
All right, great. So that ends the session today. So thank you very much for being very attentive and interactive audience. And of course, nothing in a long day, but it's you're all energetic and so please grab some snacks and you can leave for the day. We will still share some more information on the WhatsApp group or Telegram group where you are. Probably some calculations will be done. Additionally, tomorrow morning, although we have a pretty late time for hours and it used to start workshop at 9. But we realize in the rain, getting everybody on time will be challenging. So do try to come as soon as possible before event 10, like 9:30 or so that we can make more use of time. Right? There are a lot of things to do as you can see. All right, that is the session today. Thank you very much.
Tell something for the participants.